All right. Well, welcome everyone to the third year of the Global Biodiversity Festival. From tonight through to Sunday evening, we'll be celebrating the weird and the wonderful, the challenges life faces, and some good news conservation stories. Our guides will be the scientists, conservationists, and filmmakers on the front lines of studying, documenting, and protecting our planet's biodiversity. So, your hangout, your spot, your base camp all weekend long is the Global BioFest website, globalbiofest.com. You can find the schedule with all the speakers that we have coming up over the weekend. There's an option there to tune in live. You can find the embedded videos where you can tune in live. Of course, if you registered, we'll be sending you all the links that you need to join us as well. And you can get to know some of the speakers a little better on the site as well. So it's a good spot to spend some time and keep updated with what's going on. So my name is Joe Grabowski. I am one of the founders of the Global Biodiversity Festival, and I will be one of your hosts throughout this weekend. I am the founder and director of an organization called Exploring by the Seat of Your Pants. We bring science, exploration, adventure, and conservation live into classrooms across North America and beyond through virtual guest speakers and field trips. On any given month, we could be doing 40, 50, even 60 live events for classrooms. Since 2015, we've hosted well over 3,000 live events, connecting over 500,000 students with scientists and explorers from 95 countries around the world. What we do is and always will be 100% free for classrooms everywhere. You can visit exploringbytheseat.com to see all of the events that we have coming up for classrooms. It's a good time there. And of course, even if you're not a classroom, you can tune into the events live via YouTube if you're just curious. So the Global Biodiversity Festival is a virtual weekend for the general public with a simple goal of shining a spotlight on biodiversity loss. It happens each year around May 22nd, which is the International Day for Biological Diversity. And, I can't think of a better person to get us started tonight. We have my good friend, Pablo Boborglou joining us. And this is a hat trick for him. He's been with us since year one of the Global Biodiversity Festival. He is a marine biologist and a penguin conservationist. He's dedicated over 30 years of his life to the study and protection of penguins. He's the founder and president of the Global Penguin Society, an international science-based conservation coalition that protects the world's penguin species. His conservation work has been highly awarded, being recognized as a National Geographic Explorer, a Whitley Award winner, and a Rolex laureate. And we're going to talk a little bit more about what that means at the end of his presentation. So I'm going to bring Popey in with us right now. Hey, Popey, how you doing today?
Hello, Joe. So I'm so happy to be with you, and thank you so much for inviting me to join you again in this amazing Global Biodiversity Festival.
Of course, of course. And, it is going to be a global festival. We're going to bounce all around the world. So, I'm here in Canada, you're in Argentina. I think it's a great way to get things started.
Excellent. So thank you so much, and I'm going to start sharing so I can start showing what we do to help penguins globally. So just a second there. Okay. So there you go. So thank you so much, Joe, for this nice introduction, and thank you to all the people that are watching, joining us today, and joining us in another fantastic Global Biodiversity Festival. I am thrilled to be with all of you, sharing what we do to help penguins globally. So in this presentation, I want to show you why penguins can be seen as catalysts for global conservation change. I'm going to share with you the main issues, the main conservation issues that penguins have been facing recently, and some of our challenges and also accomplishments. So let's dive into the penguins world. So there are 18 different species of penguins in all the planet, and they breed across islands and continents throughout the southern hemisphere. But penguins are excellent indicators of the health of the oceans, and the oceans are in trouble, and so are penguins. Half of the 18 species of penguins are considered threatened, which means endangered or vulnerable, by the International Union for the Conservation of Nature. Now, I want to show you the world, the planet, from the penguin perspective, which is with Antarctica in its center. So, as you can see, there are penguin colonies in Antarctica, but also in the south of Australia and New Zealand, in South Africa and Namibia, and also in 4 countries in South America. And if you take a look at the yellow arrow, that indicates the place where I am right now talking to you. I am in Patagonia, in the southern part of Argentina, close to Antarctica, and the penguins are very close to where I am right now. I'm basically in penguin land. So, contrary to the common belief that all penguins live on ice and they only live in Antarctica, there are only 4 species of penguins that breed in Antarctica, including these funny Adelie penguins that you can see in the image. But there are other species. Most of the species they breed outside of Antarctica in temperate regions, like this humble penguin that is hugging a cactus in the coastal area of Peru in a desertic area. We even have tropical penguins, like the Galapagos penguins that live in lava caves in the Galapagos Islands in Ecuador. And I don't know if you knew, but there are also penguins that nest in the forest. These are the Fiordland crested penguins that live in the south of the Island of New Zealand. And I don't know if you could find the penguin in the picture easily, but it's in the lower right part and it's hidden there. And it's very difficult to work with them because it's very difficult to find them when you work with them. They live in caves, under logs and trees, in very steep slopes in the fjords of New Zealand. So as you can see, penguins live in a wide variety of habitats and not only on ice. And penguins, they spend most of their lives in the ocean, but also live on land. So they accumulate threats in both environments, and they're very, very sensitive to all the alterations in their habitats, including climate change, fisheries, pollution, and when they come on land, they are affected by human disturbance and the introduction of unfamiliar predators. Climate change in Antarctica is changing the pattern of ice formation and melting of ice, and that affects the quality, but also the availability of habitat that penguins need to eat, but also to breed. But outside of Antarctica, climate change is affecting the availability of food because it changes the distribution of the food in the moments when penguins need the food to be close to the colonies. Because when chicks are small, the food needs to be close to the colonies. But climate change has also been increasing the frequency, the severity, the intensity, and the duration of heat waves. And penguins have a problem with heat waves because they are adapted to live in the ocean or in cold environments. So when it gets very hot, it's difficult for them to dissipate the heat. So they pant like dogs. Their bodies are covered by feathers, so they cannot eliminate the excess of heat. And like, for example, in 2019, in one of our colonies, there was a temperature of 112 degrees Fahrenheit one afternoon, and 240 healthy penguins died in one afternoon when they were trying to reach the coast to refresh themselves. And climate change, they have also been increasing the wildfires. And this is a very big problem for penguins because during their evolution, they have not been exposed to fire. So penguins do not recognize fire as a threat, so they don't run away. They have been seen preening their feathers calmly behind the flames, so they stay there until they die. So this is a new threat, a new concern for penguin conservation. Fisheries are also a problem because they can compete for food with the penguins. And not only because they compete for food, but sometimes penguins, they die because they get entangled in fishing nets during the fishing operations. And unfortunately, there are also problems for penguins when they come on land to build nests. For example, last year, we reported and denounced a case in which a person with a bulldozer destroyed a big part of a penguin colony. He destroyed 22,000 square feet of habitat. He used the bulldozer to build a road of almost 1 mile, removing all the native vegetation and also destroying the burrows where penguins nested. Penguins also nest under the bushes. So as you can see here, this is an image taken from a drone. So you can see the burrows where penguins nest and some of the bushes where penguins nest underneath. And you can clearly see the impact of that road through the middle of the penguin colony. So this is also a big problem related to human disturbance in penguin colonies. But the good news is that there are many ways we can prevent these situations. Right now, we are working in a big, challenging project to create a huge marine protected area of almost 600,000 acres in Patagonia in a place called Punta Ninfas, which is close to the place where I am right now. We want to create the area, and then we will work in a participatory process involving the communities, the landowners, and the governments to make the management plan. This area is amazing, not only because there are penguins, but because there are there's a rich biodiversity, including whales, orcas, dolphins, different species of seabirds, and terrestrial mammals, and unique reptiles. But of course, in this area, there are also many threats. One of the threats is plastic pollution. This is an image of one of the nearby areas. Many of these plastics have come from the fisheries, like those plastic boxes that you can see there, but also a big part comes from reckless people that visit the area. This is a problem for penguins and other wildlife because they get entangled in big pieces of plastics, but sometimes they swallow the plastics, and that can harm them and can even kill them. And also, as I mentioned before, human disturbance is a big problem for penguins and for other wildlife because people now have different kind of vehicles and they can access places that were pristine for many, many years. So the original seed of to protect this area was a colony that we discovered 14 years ago, when we discovered the colony had only 6 pairs of nests, and it was severely threatened by many threats, like human disturbance. People used to go and throw garbage, they used to hunt, they used to take dogs that were harming the penguins. So we protected that area and that colony, and the colony grew from 6 pairs to 3,600 pairs now, thanks to our conservation actions. And now, with this project, we want to expand this area and benefit more wildlife with our conservation model. In other places, through our organization, the Global Penguin Society, we are strengthening our conservation work and research work, like, for example, in New Zealand, studying the Fiordland crested penguin. One of the things we do is we track penguins, and in the next video, you can listen to Ursula. She's one of our Global Penguin Society members in New Zealand, and she's going to share some of the results with us.
So we used satellite trackers to follow their pre-molt dispersal. And we found that birds traveled up to 2,500 kilometers away, covering distances of close to 7,000 kilometers. That's halfway to Antarctica. No other crested penguin species known to travel that far during this time of their annual cycle.
The interesting thing is that this used to be one of the least studied penguins of all. So we tracked them because we thought they were going to forage very close to New Zealand. So this is New Zealand, this is Australia, and Antarctica is down here. So when we tracked them, we were very surprised to see that they traveled almost 7,000 kilometers, halfway to Antarctica, which was really surprising. And this information is critical to protect the areas where they are in the ocean when we don't see them. So we also partner with colleagues in South Africa to gather information, to track penguins and gather information to help stop the plans of the fisheries to eliminate the current protected areas there. And this is very important because the African penguin population collapsed from 2.2 million breeders 100 years ago to only 20,000 now. So that is a drastic decline, and these penguins need our attention. But there is no possible conservation if we don't engage the communities and the children. So we have a huge education program in which we do a lot of activities. Like, for example, we have taken 7,000 kids from developing countries to visit their nearby penguins for the first time. Some of these kids live very close to the colonies, like half an hour drive, but they've never been able to see them and value them. And in the next video, you are going to see some of our education activities in which we take adolescents to remove plastics from penguin colonies when penguins are arriving to nest there.
Hey, Popey, the video just finished playing. It was great.
Thank you. Thank you very much. So, as you can see, penguins are key for ocean conservation because of their ecological role. But it is very important to consider that people have a natural emotional connection with penguins. So, penguins can be used as the perfect tool to inspire changes in the actions and in the choices of businessmen, of decision makers, and the international community. So far, thanks to the work of the Global Penguin Society, we have been able to protect 32 million acres of penguin habitat in the ocean and on land, and we have benefited 2.4 million penguins worldwide. So we have done a lot, but there's still a lot to be done, and we will continue working very, very hard to help the penguins, the people, and the entire planet. So thank you very much, and I'm really happy to answer any questions you might have.
All right, excellent. Well, Popey, thank you so much. I always love seeing the work that you're up to. You get to work with some pretty charismatic species. And I mean, the conservation work speaks for itself. The numbers of penguins increasing, especially in that colony in Argentina. And I often think back to the live events we've done from the colony with the satellite units. And that's such a awesome thing to be able to take students live virtually to the colony.
Exactly, exactly. And those events are amazing because, I mean, children can see live chicks and eggs. And, you know, Joe, one of the most rewarding things of working with penguins is that, for example, we protected that colony and penguins responded very quickly. You know, they could see that that was a safe place and it was healthy for them to come back. So they kept on coming. And at the beginning, there were only 6 pairs. But then, you know, year after year, we have like hundreds of new couples coming in and coming in. So 6 in the origin, now 3,600 pairs. So that's really amazing. And when we go to these colonies, we say we cannot believe the numbers because Fortunately, is every single year is more difficult to count them all.
Yeah. Yeah. And you kind of have a small advantage with the species you're protecting because penguins are so charismatic. Kids love penguins, adults love penguins, whereas some people maybe looking out for invertebrates and things like that have a little bit more of a challenge to get people excited about that species.
Absolutely, absolutely. And we think that of course, we work with a charismatic species, and one of the things about penguins is that they are umbrella species. So they are not only charismatic, but they use the environment in a very wide way. They use vast areas in the ocean. They swim thousands and hundreds of kilometers in the ocean and also on land. So when we protect penguins, we create huge protected areas, and we benefit other species that maybe are not so charismatic, or maybe we don't know enough about them. So through the penguins, as I've shown in the picture, through the penguins, you protect big areas and you protect many other species. For example, here in the place where I am right now, we created the largest UNESCO Biosphere Reserve. And the face of that campaign were the penguins. We protected 20 penguin colonies, but we also benefit 800 marine and terrestrial species. So the penguins are helping their friends as well.
Awesome. Absolutely. Use them to your advantage. They're a great flagship species. So in the chat, I see some people already saying hi. You've got some company in Latin America. We've got people in Quito, in Ecuador, in Argentina, and Costa Rica. So don't be shy. Use that chat sidebar and send us in some questions. One question here, Popey. You showed that great map of the penguins' world perspective, where Antarctica is in the center, and then there's a few other places in the southern hemisphere. So what does a tropical environment need to have in order to have a penguin species? What is it about those few tropical environments that allows them to thrive?
So it's very interesting to compare the life history. I mean, the features of the penguins that live in temperate regions, like closer to Antarctica, where you have very strong and marked seasons, compared to other regions that are closer to the equator, like Galapagos or Peru, where the seasons are not so marked. Like here, for example, I am in Patagonia, penguins have left the building. I mean, because they come only here for the summer to breathe, because the food is here. But when winter comes, the food is no longer here. So penguins leave, they follow the fish, and they go to other latitudes. Like our penguins in Patagonia now are in Brazil, you know? So, and if you think about the penguins that are in the tropics, like the Galapagos penguins, they don't migrate, they stay year long. And instead of having one like breeding season, you have colonies like in Peru, and they are breeding all year long in different moments, because there's different different availability of food, like in different parts of the year. So all the how you say, all the breeding biology differs completely if you compare a penguin living in the tropics and in closer to Antarctica.
All right. Another question here coming in via the chat. Actually, some more greetings too. So Brazil, India, someone else joining us in Quito as well. So it's great to see so many people joining us. And then a question here about, so penguins are distributed in very different parts of the world. So what would you suggest individuals do to help protect the species? Maybe what request do you think they could make to government?
So I think a critical thing, even if you don't live in a coastal area and if you like penguins and oceans, one of the things that I mentioned is plastic pollution. And plastic pollution reflects the way we have impacted this planet. You know, we see it. It's not like climate change that sometimes is difficult to understand or sometimes difficult to measure. Plastic pollution, we see it, but there's a lot. The good news is that there's a lot we can do. I mean, we can start right now. When we are, I mean, every single year, 11,000 tons of plastics end up in the ocean, and that affects wildlife and different organisms. So all of us, I mean, we all have a role to play. And if we stop, if we can avoid single-use plastics right now, at the end of the day, there will be a lot of many pieces of plastics less on our planet. So this is something that we can all do. It's up to us, and in that way we can help the penguins, the oceans, and many other marine wildlife that are affected by plastic pollution.
Yeah, absolutely. Amazing point. And it is something easy for us to do, is to kind of move away from those single-use plastics. You know, the equivalent of a dump truck every minute entering our ocean. The difference that would make for not just the penguins, but many other marine species would be huge.
Yeah, exactly. And apart, yeah, sorry.
No, go ahead.
No, that it's also important because for me, I'm motivated by penguins because my grandmother, when I was small, told me about her visits to see penguins 100 years ago. That's the way I connected to nature. But all of us, we have different motivations. Maybe we are interested in butterflies or in plants or trees, whatever. So whatever we are interested in, we can learn a little bit more, and then we will value those creatures more, and we will do a lot of things to protect them. Even talking about them, if we go to a birthday party or whatever, talk to our peers, talk to our friends about the concerns, because that's a huge role that we can have in the conservation of our planet.
Absolutely, no question. And that's why I love the second video you shared of the next generation getting involved and realizing the importance of protecting not only species but the ecosystem and what it means to us. So amazing work you're doing, and I want to make sure that we share the link to your website. So if anybody wants to visit globalpenguinsociety.org, find out more about the incredible work that Popey is doing along with his team. And then, of course, support in any way that you can, because that's so important for keeping the work going.
Excellent. And apart from that, if people want to join us on Instagram, we are all the time posting things that we are witnessing with penguins live from the field. And so we're very happy if people want to write us. I am the one also answering the questions, so we're happy to answer any questions you might have.
All right, perfect. Well, like I said, Popey, I can't think of anyone better to have kicked us off. It's a hat trick for you. 3 years in a row joining us at the Global Biodiversity Festival. It is so good to see you virtually, and I can't wait to see you in New York in a few weeks. Thank you so much for all the incredible conservation work you do.
No, thank you so much for inviting me. Have a fantastic festival, and I'm sure it will be a complete success, just like the other ones.
All right. Have a great evening. We'll talk to you soon.
Bye. Thank you.
All right. Well, we're moving along. The first event is under our belt. It's so great to see so many people around the world joining in. Someone just said hi from Nigeria. It is great to have speakers from around the world, but it's also great to have audience members from around the world joining us as well. Before we introduce our next speaker, I do want to touch on what I mentioned when I was introducing Popey, and that's the fact that he's a Rolex laureate. So we're absolutely thrilled. This is the second year that the festival has had support from the Rolex Perpetual Planet initiative. So as the 21st century unfolds, exploration for pure discovery has given way to exploration as a means to preserve the natural world. So Rolex continues their incredible legacy by supporting the explorers of today on their new mission to make the planet perpetual. So I want to share a little video clip with you here now before we meet our next speaker. So here we go.
Explorers, adventurers, scientists, the ones who always broadened our horizons. We were at their side when they reached the deepest point in the oceans, the highest summits of the Earth, and both poles. We may think we've seen it all, that the world has its limits after all. But why do explorers, adventurers, scientists continue to venture out there again and again? Certainly not just for the record. So what do they seek, really? To to understand more intimately how complex and delicate our planet is, to document its change and how we can affect it for the better. As long as they need it, we will be at their side. Because today, the real discovery is not so much about finding new lands, it's about looking with new eyes at the marvels of our planet, rekindling our sense of wonder and acting here and now to preserve this pale blue dot and make it perpetual.
All right, so I'm going to share a link here if you want to dig a little bit deeper and find out more about the Perpetual Planet Initiative. And over the last few years with the festival, we've been lucky enough to host well over a dozen Rolex laureates and share the incredible work they're doing that in part is being sponsored by Rolex. Very cool. All right, so here we go. Like I said, I'm here in Canada. We were just visiting Argentina, and now we're going to pop over to the other side of the world because we can do that virtually to Australia, and we're going to spend a little time with Dr. Vanessa Parada. So she is a wildlife scientist, science communicator, and woman in STEM. Her zoological background has taken her around the world for wildlife research. She's pioneering the use of emerging technologies for whale conservation. Her whale research has taken her around the world to places like the Galapagos, Madagascar, Antarctica, and Tonga. In her home country of Australia, she's using drones in unique and creative ways to assess whale health as they move through the environment in a non-invasive way. So I'm going to bring Vanessa in live with us now. Hey,
Hello.
Vanessa, how are you?
I'm very well, thanks, and hello to everyone around the world.
All right. Well, it's great to have you, I guess, coming to us from the future. It's Friday for us. It's Saturday morning for you. So good to see you, and just thrilled to have you sharing a little bit of your story, the work you're doing.
Thank you so much. And I'm so excited to share this with you because there'll be probably people who have not seen our work, but also are not aware of the stuff that happens excitingly down here in Australia. So if you're okay with me, Joe, I'm going to share my screen.
Absolutely.
And we can get into it. So we'll go to my slides. So just one moment here. The work that I'll be sharing today will be about the whale research that I've been doing for many years now. So I'm just going to go to my PowerPoint presentation, and I'm in Chrome, but for some reason it's just not working here.
That's okay. We'll walk you through it. We'll get it up.
Yes.
If you hit the share button in the middle.
Yes, I've got that up ready.
Yeah, and then the share screen
Yes.
button. And then hopefully it's going to give you three options. Do you see the one for entire screen?
Yes, the bottom one that says share screen.
Yeah, and then click on that picture of your screen and the share button should light up.
Okay, and share. Now Chrome has last permissions. Can you see it?
That's okay. We can fix that. Are you on a Mac?
Yes, I am. Sorry for the teething problems. I have been on this platform before.
Yeah, I know. It can just decide sometimes that it wants you to refresh. So if you jump to the apple in the corner.
Yes. My Chrome apple.
Yeah, look for system preferences.
Yes.
And then
bear with us, everyone.
Yeah, no worries at all. This is going to happen a few times this weekend. It's the joy of being live. Select security and privacy.
Okay. One moment.
Yeah.
Yes.
And then you should have a scroll down menu. Look for screen recording.
Okay, one moment.
Yeah.
Yes.
Yeah, click on that and you should be able to put a little tick next to Google Chrome on the right.
Now, unfortunately, it's unhighlighted, so it's very...
That's okay. Just unlock the little lock at the bottom of the window so you can make changes.
Oh, great. We love being on a Mac. Here we go. Okay.
It is definitely easier on a Mac. I do like it when it's a Mac.
Now it says quit and reopen.
Yeah, say yes, and we'll see you in a moment. All right, there we go. So yes, the beauty of being live is that sometimes technology doesn't cooperate. Vanessa has joined us for classroom events where she shared the work she's doing with students. It was only a few months ago, but you can see that the StreamYard program decided it wants her to give permission to share her screen again. So we should see her momentarily, and then we should have no issues getting to dive in a little bit deeper into her research. And you're going to be pretty excited. I think she's got some cool video clips that she shared with students in the past. And it is really a novel way of, a non-invasive way of being with whales and exploring their health. So let's bring Vanessa back in. Here she is.
I'm back.
All right, let's give that screen share a whirl. I bet you we'll have no issues this time around.
Nope, that's right. Thank you for bearing with us, everyone. So hopefully now you should be able to see my screen. Can I just confirm that?
We've got it. Looking good. It's all yours.
All right. Thank you so much for your patience, everyone. Okay, so here we go. The work that I'm talking to you today about is a lot of the whale research that we're doing down here in Australia. But I will point out that I'm talking to you from Sydney in Australia, which is today this is from the future because I'm in Saturday today. So it's very cold here. And right now we're having humpback whales on the east and the west coast of Australia migrate from the cool waters of Antarctica all the way up to their breeding waters in the northern Australian waters. So this is very exciting. And before I do officially start, just letting you know that I'm affiliated with Macquarie University and also the Marine Predator Research Group here in Sydney at Macquarie Uni. And the work that we do in our laboratory led by Professor Harcourt is a variety of things. We just heard about penguins. The lab group also works on penguins, little penguins, the world's smallest penguin, and the team puts trackers on penguins to see where they're going. I work on whales, obviously, and there's sharks, fur seals, you name it. And some of the other work that I'm leading here in Sydney is Wild Sydney Harbour, which is looking at using citizen science to learn more about our locals in the area, such as dolphins, but also this is a fur seal. You may or may not be familiar with fur seals, but this one here likes to hang out at the steps of the Sydney Opera House, so very famous. Okay, well, you're probably wanting to know about my talk title. And as I said before, the humpback whale is the animal that we are now experiencing off our coast, which are a migratory species. And in the southern hemisphere, they do tend to migrate from the cool waters of Antarctica. And thankfully, they go past Sydney. So this is a photo I've taken during research of a northward migrating humpback whale. And in the background there is Sydney, and Macquarie University is a little down the other side. And so this is just to show you that these animals annually come past our coast and in our backyard very close. So we're very lucky. And for those who love Sydney or very aware of Bondi Beach is just down there a little bit, and the animals will literally go past that. So you've got one of the biggest cities in the world and a recovering humpback whale population, which is a good thing. So these animals were hunted to near extinction. And fortunately, since their protection, their numbers are doing quite well, which is not the same story for other whale populations around the world. And these animals do, unfortunately, face a number of additional threats around the world like other whales. So I'll point that out. But when they were in Antarctica, so where they've come from, so over Christmas time, these animals go down to Antarctica to feed and to enjoy the cool waters there where there's a lot of krill. So these animals will predominantly feed on Antarctic krill, but they will also feed on small bait fish. And Antarctic krill are, and I will show you a photo in a moment. There they are, just on your left-hand side there. They're small little crustaceans, probably a couple of centimeters in size, and they can feed on these animals in huge numbers. And research has shown that down in Antarctica, there can be really big swarms, up to 1 kilometer in length. So there's huge numbers of them, which is good. But with climate change, these animals are at risk because if we see a reduction in sea ice, we see a reduction in Antarctic krill, which is bad news for a recovering whale population. So it's really important for us to think about whales as this, as a sort of an umbrella species, we could say, as we heard in the previous talk. And these animals are really important for all of us. And this is why I tie in with the title of my talk as Whales as Important Ocean Monitors, because what happens with whales is that often changes in their population may potentially be reflective of our environments. As I mentioned, Antarctica, soon we'll be going to the tropics. So these animals undertake big migrations from extreme environments each and every year. And so they're seeing the differences and the changes in these extreme environments that we are not necessarily privileged to, because, well, logistically, working in Antarctica is quite challenging. And so whales, in short, play important roles in our ocean because they'll feed in one area, and they'll feed a lot, because we're talking about an animal the size of a school bus, 17 meters in length, and they can feed in one area, and then they'll swim to another. And as they swim to another, they'll poo. And this is a picture of their poo. Their poo breaks up so that, and it can be quite rich in color because of the animals that they're eating. But often when a whale poos, it will disintegrate and just really break up. But when they do poo, they'll feed a whole other part of the marine ecosystem, which is important. And they'll feed those tiny organisms that the krill are eating. So they're able to also move a wide variety of nutrients like iron as well around the ocean. So these animals are really ecosystem engineers of our marine environment. So super important. When I went to Antarctica, I was on a research vessel called the RV Investigator, which is an Australian vessel through the CSIRO. And this is the vessel I was on. It's 96 meters in length. And so that's nearly 100 meters. And so this is an iceberg here just to show you the size of it, which is pretty epic. And you can see it here again. So this research that we were doing was not specifically focused on whale movements, but was looking at seafloor mapping as well as collecting ancient DNA. So this very ship can drill down into the ocean floor and collect almost like a time capsule of previous climate, which is really interesting. So my role on this vessel was to keep an eye out for whales as part of an Australian federal permit as a marine mammal observer to make sure that our activities didn't disrupt the animals down there. And so as a result, I was able to collect a number of observations of these animals feeding. And when they feed, it's amazing. They will open their mouths up. Some whales feed coming vertically up, but some animals will lunge feed where they open their throats kind of like an accordion or a slinky. They open it up really wide, open their mouths, and then they can engulf their krill. I'm talking a big swarm here. And then the water as well. And then they'll use their baleen plates, which are hair-like structures, to filter out that water and then swallow the food. So key points here, these animals will feed in one area and poo in another, moving nutrients around and fertilizing our oceans, which is really important. And some of the other work that I'm very fortunate to do is I'm an ambassador for the Antarctic Science Foundation. And I was privileged to see Antarctica via the air a few months ago. And so I just want to point out that while working on a ship, you see Antarctica from one perspective, but really the opportunity to see this environment is just spectacular through another perspective. And the photo on the right-hand side where you can see the 787 wing, that's where the McMurdo station is. So that's where the Antarctic scientists are taking place. And there's a lot of elephant seals down there. So I just wanted to point that out. Then when the animals are in Antarctica and then they've done what they've had to do, they've put on a lot of weight, they've fed, they've spent their time collecting a lot of fat, putting on a lot of blubber. These animals will then spend a period of time where they don't eat and they migrate. So this here is a photo of a humpback whale migrating past Sydney. And this is a really great opportunity to see some of the throat pleats. So you can see the throat I was talking about, the accordion or the slinky opening up, and the eye is just around here. You can't see it very well, but this is a photo of a humpback whale migrating north past Sydney. And because these animals are migrating past my backyard, essentially, we're able to learn more about them quite in an easy way because, well, they turn up every year, which is good. So when they do go north, where they're going to is the nice warm waters of Queensland. Now, this is the East Australian coast population that I work on. And when they get to the areas like the Great Barrier Reef, it's warm, it's lovely, and they're there to have a good time. I call it the whale disco, where females will give birth, but also males will be singing to attract other whales that haven't had a baby just yet. So they'll be wanting to reproduce as well. So as you can see, in a span of just a few slides, we've gone from an animal that's in Antarctica, migrating along the east coast of Australia to get to the warm waters. So in every single year, these animals, these very mammals, which undertake one of the longest migrations of any animal, or at least any mammal, undertake a huge transition across the marine environment, and really can tell us a lot about the ocean in each year, which is incredible because, again, they're going from such an extreme environment. And so I've actually written about this, and this is the first time I've shared this, but I've written a children's book to document this migration, but also to explore some of the science around it. And it's called The Voyage of Whale and Calf. So I thought I'd put that there for any younger viewers who are interested to learn more. And because these animals undertake this migration, the story that they undertake is one of a narrative that I've just shown you, but also one in which is a scientific story. And so this is where I introduce some of the work that we've done, and we continue to do so. And because the animals, these ecosystem engineers, they're pooing in one area, feeding in one area, pooing in another. And then as they come through the environment, they're actually collecting information at a microscopic level of changes in our ocean that may be happening that we're not necessarily aware of. So one of the ways in which we can access this information is by using new and emerging technologies, which I love. And I love teaming technology, conservation, and wildlife together. So this wonderful collaborative approach to learning more about animals, and in particularly in a non-invasive way. So again, here's a photo of 4 humpback whales migrating off Sydney. I believe this is off Bondi Beach. And on the top you can see a drone. And yes, that does say whales not collecting drone. Now, the work that we've been working on is assessing lung microbiota or bacteria, also which we refer to through science communication as whale snot. So this drone is one of many drones that we've currently got in our toolbox of tools to access whales. And we are collecting that breath or that spray that the animals have. And they've got 2 nostrils. And as they take a breath, you can visually see that plume, which is a mixture of water, but mostly bacteria coming from their lungs, which we can collect to learn more about their internal health. Now, one of the ways we do that is by flying a drone from the back of a boat out to a whale's position. And this drone is loaded with a fresh petri dish on top of it. And that petri dish will remain shut only and will only open until over the whale's position. Now, the great thing about this is that as the whale is swimming 200 meters or more away from the boat in the distance, the whale will have not necessarily any idea that it's happening. And previous research has shown that in all our flights, the whales either knew the drone was there and did absolutely nothing, or the whales had no idea that the drone was there at all. So we're talking about a drone probably the size of a school ruler, maybe a bit smaller and longer in another way. And as the whales come up to take a breath, the camera on the drone enables us to see the whales underwater and just about to surface. And then as the whale goes, that's the information or the biological information that we wish to collect to learn more about them. And this here is a photo of the drone in the air with that. You can see kind of a bit of a spray there, a whale that's just taken a breath, and the drone's video will be filming the whales to work out which whale it's sampled from. And funny enough, you're probably thinking, oh, those whales, they all look really similar. How do they know? Well, the whales do have a lot of different shapes and markings on them to help us go, oh, that's Jack, or that's Fred, or that whale is totally different from that one, which will enable us to work out how many different samples that we collected. So this is just one example of using a tool to access animals in a remote way. Now, what is this actually telling us? Well, keeping in theme with the entire theme of my talk, we've seen that the whales, the roles that they play just naturally and biologically, this here is enabling us to collect microbiota or tiny bacteria, but as well, and in a world first via this method, viruses. And so what we're able to collect is viruses associated with areas down in Antarctica. Now, this is really powerful and really important because the animals, if they're able to collect certain bacteria and certain viruses at certain points in time, may be able to show us changes in the environment over consecutive years. So if we see the presence of certain organisms, that might be reflective of certain temperatures in our ocean. And by thinking of this in another way, we are essentially able to collect some of the world's smallest organisms from some of the biggest. Now, while this is showing us what is likely to be representative of relatively healthy humpback whales, we're able to take this information to compare it with other whale populations around the world, and also compare it with whales that might be sick or injured to look at the changes in their internal health. In addition to this method, using drones to measure the width, the length of a whale is also really important to gain morphometric information so that we can use the internal health information as well as those external factors to put together a piece of this information puzzle to learn more about their health as well, which is really important. And I must point out, keeping an eye on time, is that these animals, this population is recovering quite well, which is good. But a recovery in the population may also mean that at some stage, how long is this population going to keep growing at a certain rate, and at what point will it simply stop growing, or there won't be enough resources to support them? So there are a number of things that us as scientists are thinking about ahead of time, and also in a changing environment. And when I say changing environment, climate change does mean that there's a lot of implications for when and where these animals might go when they migrate. Our research has also shown that these animals are feeding in Australian waters in big numbers, which is the first time seen here just a few years ago, which means that their feeding areas might be extending outside their primary feeding grounds in Antarctica. We knew that opportunistically they would likely feed on their migration, which is kind of the equivalent of you and I going for a road trip and taking a healthy snack. But at the point now where these animals are feeding in huge numbers, that it's becoming more than just that. And this might be reflective of changing ocean temperatures and currents, which is able to support these animals feeding in Australian waters. So as you can imagine, really key information here for us as scientists to investigate further, to learn more about their movements, but also when and where they're likely to be, to help ensure their protection in Australian waters and in other parts of the world, which is key. So a lot of big findings there. Here's another picture of the drone in action. As you can see the dorsal fin of the whale and the nostrils. So the whales have this splash guard, which is sort of it's erect a little bit to enable that the animals can take a breath with minimizing the amount of water that might go down their nose. And we've also adapted this drone application to collecting dolphin snot via a drone in another world first, which is a little bit more tricky in comparison to the humpback whale collection. And I would just highlight that I've spoken more about this in my TEDx talk, so feel free to Google this as well. What whale snot tells us about whales in the ocean. So again, looking at the vehicles of whales is telling us more about the ocean at a microscopic level. And I guess I want to leave on this is, well, what can you do to make a difference? We've seen scientists do things to help whales and help penguins, and that's all wonderful. But what I would like to point out is that you don't have to be a scientist to make big changes. And this is a global talk that we're having right now. There are many actions that you can take to help preserve our marine environment, which I've just explained to you, that we have many really important animals to help ensure that the regulation of our ecosystem is in check, like whales. And our actions on land are really important because any changes or activity that we have here is likely to have flow-on effects in the ocean. Having a discussion like we're doing today, a global discussion, or this evening, and a morning discussion here in Australia, and thinking about the future. I know you'll be hearing a wide variety of talks in this wonderful festival, and it's really key that we think about the organisms that we're working on, but also the bigger picture and where they fit in. So the future thinking is key here. We've now come to the tail end of my talk. Thank you so much for listening. If you'd like further information on any of the work that I'm doing, as well as my team, here it all is. There's social media references, and there's also a website that I do have where you can learn more. And so with that, I'm going to stop sharing my screen and open up for some questions. So, Joe, do we have any questions come through?
I think we do, Vanessa. Thank you so much for a great presentation. And thanks for sharing all your social media handles there, and TikTok. I'll have to check it out. I don't think I've checked out your TikTok account, so I'm going to have to take a look at that.
Only new.
Oh, there we go. All right. Okay. Well, just a few comments here quickly. I want to give a shout out to the UK. I can see we have some viewers. It's after midnight in the UK, so thanks.
Wow.
Thanks for being here with us, and hopefully we see you during the day tomorrow as well, when it's a bit more manageable. Very cool. All right. Well, don't be afraid to use that chat and send in some questions for us. But I have a couple here I wrote down already. So the first question I have here is about the data you're collecting with the drones. What's been the most surprising thing to come back so far?
Oh, I would believe it's the level of complexity. So we're doing next generation sequencing, which is a very sophisticated way of learning about the types of information in whale bacteria, in their snot rather. And so I think being overwhelmed with just how much information or how much variety and diversity of bacteria they're carrying. And if we think about it, we too as mammals carry a huge variety of information biologically. And so trying to interpret or at least understand a starting point to then go onwards and then collecting information over consecutive years to see how that might be changing and how is the environmental differences changing what's likely to be in a whale's population in terms of overall population health. And that's really interesting. And the limitation here is that we obviously can't sample all 40,000 plus humpback whales in this very population. So, it's a bit of give and take. So that's why sampling a few individuals can only tell us a small snapshot of what's going on in the population we're working on.
Yep. All right. So with the drones, was it difficult to get? Did you have to get permission to do it, to fly so close to the humpback whales?
Yes, with any whale research, you need animal ethics and scientific licenses. And in Australia, we're very proud to have regulations through the Civil Aviation Safety Authority, as well as state and government parks, national parks. So there are rules and regulations for flying drones around whales. And the general public here need to stay at least 100 meters away. But also the team that I was working with had huge amounts of drone experience and were able to work really well together to ensure the safety of the animals at all times.
All right, excellent. So another question here in the chat. First of all, saying great efforts. Obviously, the water environment is a technical ecosystem to work on and research. And so the question is about being a female scientist. How, what kind of impacts, anything maybe in the way of the work or anything you've experienced?
Well, it can be tricky because if you want to juggle having a family, working in the field, I mean, it's all a bit of time management and priority, but you can get through. And it does mean that if you're really passionate about something, then you'll pursue that passion. And it also requires a lot of hard work. So I'm not going to sugarcoat it. It does require being flexible with time. Good communication is key, but it is also really rewarding because it's great for young minds to see people in my position and other females in science driving boats and doing science and working in a predominantly male-based environment. And thankfully, I'm being very privileged to work with a great team and will continue to sort of be visible for young audiences and young girls as well, to encourage them to get into STEM and STEAM, to follow their passion into. And I must point out, a lot of the marine science jobs don't even exist just yet because they're the jobs of tomorrow in the future. Yeah. So you just never know what might come up.
Yeah. All right. Very cool. Cool question here. Have you ever sampled the same whale, but in a different season?
No, we haven't. So that's a great question. No, not yet. And there is a very famous whale called Migaloo, the white whale, here in Australian waters. He's completely white. And that would be a great example of sampling the same animal, but we've not seen him in research.
Okay. So I think this is a great question to kind of to end the session on is what's next? So maybe you kind of gave it away, is maybe trying to sample the same whale in another season. But what's next? What are you hoping to accomplish next with this research?
Oh, what's next? That's a great question. Adapting, our drones have been adapted to do different things and ask different questions non-invasively of whales. So I can't give too much away, but everything we do will involve conservation, technology, and learning more about these giants to learn more about our environment, which is so important to all of us, our marine environment.
All right, perfect. So you shared your social before, so those who are tuning in are able to rewind. We will put up the events in a playlist as well, but I will share the Instagram link there if you do want to check out more of Vanessa's work. And congratulations on the book. That's so exciting.
Thank you so much. Thank you.
All right. Well, thank you for joining us, Vanessa from Australia. It was great to have you joining us. It was great to hear about the work you're doing and the cool use of technology.
Thank you so much and have a lovely festival.
All right. Enjoy the rest of your day. Okay, well, we are going to move to a different part of the world now. We're going to go to the United States. I'm going to give a brief introduction for our speaker, and she's waiting behind the scenes. And we have a video that we're going to share before we get a chance to ask some questions. So Victoria Strange is a wildlife biologist with experience conducting surveys for birds, mammals, herpetofauna, and fish throughout the U.S. She currently manages 4 preserves for Palm Beach County in Florida. And we are going to join her in the field tonight, which is pretty darn cool. So I'm going to bring Tori in with us right now for a little greeting. Hey, Tori, how are you?
Hey, Joe, I'm good.
That's awesome. It's so great to have you joining us live tonight. Before I play the video, can you tell us where you are?
Well, right now I'm up in North Palm Beach, but I work all the way from Jupiter down to Lake Worth area down to Delray, spanning the whole Palm Beach County area.
All right. Well, the amazing Kate Bosler, who will be joining us at other points throughout this weekend, filmed a video with you. So what do you say? Should we pop in the video and go from there?
Sure, that sounds great.
All right, so just bear with me for a moment. I'm going to queue up our video here. There it is, and here we go.
My name is Katie Bosler. I am an investigative environmental journalist, and I'm joined today by a speaker I have known for over 10 years, Tori Strange, one of the most dedicated conservationists that I've ever met. And her frog catching skills are yet to be surpassed. Today, she will be taking us into the nature of Florida and Palm Beach County. We'll get a rare look at land that hasn't been developed for some of Florida's quintessential human activities like golfing and yachting. Her reserve shows plants and creatures that are safely tucked away for now. She'll get into a little bit of the challenges that face the area. She's a wildlife biologist, and she's researched all walks and flights of life, from birds, mammals, reptiles, amphibians, and I've even seen her free dive deep into the ocean to learn more about fish and marine mammals. I'm willing to bet that she can hold her breath longer than most people out there. Her graduate work took her into the high altitude of Nicaragua, and she's speaking up to us today in her current role working for Palm Beach County, where she manages 4 preserves. We're about to take an excursion with her where we will learn about scrub habitat, Florida's oldest community type that is facing pressures from surrounding development and other challenges that her and her team work hard every day in the field to address. Tori, thank you so much for joining us today. Can you tell us a little bit about where you are? I see some really cool plants and trees and environment in the background. Are you at one of your sites?
Yes, I am coming to you from Palm Beach County, Florida, in the Southeast Peninsula. We are on a scrub site, Lantana Scrub Natural Area, managed by the Department of Environmental Resources. And here in South Florida, we have no shortage of pressures on biodiversity. Encroaching development, like you can see to the south of us, causes direct habitat loss through land conversion, and basically eliminates functioning native assemblages. Along with that, fragmentation causes a loss of all the sensitive species, such as scrub jays, which family groups require over 20 acres of well-managed habitat to survive. The remaining populations on these isolated fragments are subjected to inbreeding depression, such as is being researched in the Gopher tortoise. And I see one actually over at the fence right now. Yeah.
Not a bad gig hanging out with Gopher tortoises.
No, it's a pretty fun day when it's filled with Gopher tortoises.
Can you tell us a little bit more about what makes this habitat unique? What's it made up of? What's around you?
So I am standing in front of a prickly pear cactus, one of the iconic species of arid ecosystems. It's very well adapted to high periods of drought stress, and it has beautiful yellow flowers that we'll see later on. And so scrub is Florida's oldest natural community, like Katie said, and it's actually like Florida's desert. It occupies high and dry sandy soils that have low nutrient content, and it has a sparse herbaceous cover, open sandy patches, and low shrubs, sometimes with a sand pine or slash pine overstory. So Palm Beach County had the foresight to see that all of the last remaining lands were being developed. So they took the opportunity to save some of these last intact habitats. And since then, they have conserved over 30,000 acres of land. And this is actually our most newly acquired site.
And I see some of that, like sort of, is that sand that you're looking at below you, or are plants able to root in that? Tell us a little bit about that.
Yes, so scrub adapted plants often have really, really long tap roots because the water table is way far below this sandy surface. And then other plants are actually adapted with shallow roots, which can uptake the low nutrient content and organic debris at the top of the soil. And see this crust right here? This is made of organic matter that takes many, many years to form. And it's like a biological crust of microorganisms.
So is that sensitive to people walking around? How do you manage for humans visiting the area?
It is. Yeah, we try to keep them on the trails because it is very sensitive to disturbance and it takes a really, really long time to form. And so actually we do have some another really cool species that sits on the surface of the sand. It's called lichen. And it's actually a symbiotic relationship between algae and fungus. The fungus provides the structure and the algae provides the working parts. So this right here is a type of cladonia. It's called a puffball or reindeer lichen. This right here is called a cigar ash. And then this is another cladonia species.
Wow, incredible. Are they just on the surface, just lying on top of it?
Yeah, they're just laying on the surface. They don't have any roots. They absorb everything directly through their outer covering, which is not very thick. And this actually makes them prone to absorbing toxins. So they're used as a biological indicator of air quality a lot.
And so tell us about that. What is the air quality like there? Is it healthier than surrounding areas because it has these plants?
So we are affected by roadways such as I-95, and I've seen the effects of that. In the portions of my sites that are closer to I-95, I tend to see less lichens, and the further away you get from I-95 in the traffic, the more you'll see lichens.
Are there animal species that are dependent or able to exist there because of things like the lichen? I know something you pay a lot of attention to is how the species are interacting with one another and how they make possible habitat for all kinds of creatures who come to visit them and depend on them.
Yeah, so it actually can be browsed by some animals, and it is allelopathic, so it creates plant inhibitive substances that help maintain those open spaces,
And
which
so
is really cool.
you pointed to a gopher tortoise earlier. I don't get to see those every day. Can you maybe head in that direction and tell us a little bit about those cool creatures?
Yeah, we could see if the gopher tortoise is still over here. He was over by the fence line earlier. So,
and they're native to Florida, is that right?
Yeah, so gopher tortoises are an endangered species. They live about 40 to 60 years. They are actually a keystone species because they dig burrows in which up to 360 other species of animals live in. including a lot that can't dig their own burrows. So here we actually have one. As you can see, they have really strong forearms that they use as like shovels to dig their burrows, and they are protected by very thick scales on their arms and head.
Oh, wow. Oh,
And the top portion of their shell is called a carapace, and the bottom is called a plastron. And males have a concavity in their plastron, and females have a flat plastron.
so are we looking at a female or a male?
I'm not sure because I haven't picked them up. We technically aren't supposed.
Oh, look at them go. And so about how many live on this site? Do they interact with one another?
They're mostly solitary. They spend about 80% of their time in their burrows. During mating season, though, they do become more active with each other. They interact more. The males have aggressive behavior towards each other, and the males follow the females around and harass them. And so they do interact with each other. Sometimes I'll see two in one burrow, but that's rare. They usually are off by themselves, though. We have about 6 gopher tortoises on this site, which is a pretty low population. Another one of my sites has over 100.
Wow. And what kind of food are they after?
They're after all the herbaceous species, so they can eat up to 300 species of plants. They like wiregrass, andropogon. They actually can eat prickly pear, cactus, and gopher apple leaves. So they're pretty versatile in their food habits.
In what kind of ways are you managing for them? What challenges do they face?
So in Florida, we have another issue with biodiversity we have is that we don't have fire on the landscape like we used to. So our understory becomes very overgrown, and it's hard for gopher tortoise to navigate this, and it's hard for herbaceous species to be able to germinate because they're shaded out. So on small urban areas like this, if we can't do prescribed burning because we're surrounded by a bunch of smoke sensitive areas, we do mechanical fuel reduction. And so we will go and chop the understory vegetation to create more of those open, low shrubby spaces so that herbaceous species can proliferate more, and so that gopher tortoises and other animals will have more food sources.
And so the tool of using fire for the habitat, is that something that you yourself are directly involved in?
Yes, yeah, I am on the burn crew, and we try to get as many burn days as possible on our sites, but we're spread pretty thin, and there's a lot of high priority places, like larger pieces of land that are flatwoods that are more adapted to a higher fire frequency. So those need to be burned like every 2 to 3 years. Whereas scrub is a little bit longer of a fire return interval of anywhere from 5 to 40 years, depending on the understory vegetation.
So I know that invasive species is something that Florida deals a lot with. Of course, the pythons and the Everglades famously comes to mind. Do invasives affect your site? And if so, how do you manage for them?
Yeah, invasives are another big issue in South Florida, both plants and animals. We have less exotics at scrub sites because they have a harder time gaining a foothold, but we still have them. And during the growing season, a large percentage of our time is dedicated to eradicating or keeping these species at bay. We do can removal and herbicide applications usually.
And so a lot of these species are unique to me. I'm just looking at these big bulbous plants and bushes behind us. Is there anything you can point out and show us some cool leaves or flowers, or what do you got going on back there?
For sure. So we have actually at our feet, we have the Curtis's milkweed right here. And it's about to bloom. You can see it's in inflorescences right now. It's a rare Florida endemic. They can live up to 25 years. Insect larvae use it as shelter and food, such as the queen butterfly. It's pollinated by skippers and hair streaks, which are actually having a difficult time these days finding these scattered, isolated populations. You can hear the gopher tortoise crashing through the softball meadow.
He's got big plans for the morning. So endemic means that a species only exists in that region. How much on the site is endemic?
So I would say we have at least 10 endemic species on this site, if not more, but I can count at least 10.
Are there any plants that you have, I'm not saying you have favorites or play favorites, but anything that you're always excited to see when you're out on your sites?
Yeah, I mean, I'm always excited to see the Lechia serenua, which is right over here.
I'm impressed by all the taxonomy you have memorized.
Here's a little tiny one. And then we have a bigger one over here. It is a really cool species that we don't know a lot about yet. It's a rare endemic as well. It's state threatened. It has really fuzzy leaves. It's highly adapted to fire and germinates post-fire very readily. It has really cool red and yellow flowers in the spring. So here's another one. So its common name is nodding pinweed.
Very cool.
Yeah, it's one of my favorites.
And what about in terms of things that are wiggling around out there? What kind of reptiles do you see? Amphibians, smaller animals?
I see a lot of snakes. So we have a ton of black racers on the site. We have some other, we have lizards. We have native green anoles. We just saw one earlier. And we have the 6-line race runners, which is a really, really fast skink-like creature. And then we have a bunch of exotic lizards too, the curly-tailed lizard and the Cuban brown anole, and green iguanas.
I've seen those little curly tails. They sure are fast darting around.
Yes.
So Tori, go ahead.
Oh, so then I just wanted to show you another species right over here. This is called gopher apple, and it's a low-growing clonal species. Only the very tips of it are above ground. Most of it's underground. And its fruits are eaten by gopher tortoises. And it's pollinated by a lot of Lepidoptera species.
Wow, pretty.
And so then I want to take you guys over here to show you a few more species.
We have a few minutes here to check those out, and then I want to ask you about your public education efforts, because I know that's unique to the sites you manage, is that people from the surrounding areas are allowed on and can be a bit of a refuge for people. So would love to end on that.
Yes. Okay, that sounds great. All right, so now we're coming up to an unnatural berm. this was the result of spoil piles made decades ago. So it's one of the scars of human disturbance on this site. And here you can see we have this interesting orange vine. It's called love vine, and it's a native species. It acts as a nuisance sometimes in the absence of fire, but it actually has a suppressive effect on the oaks. So it does help to keep them back sometimes because they do get overgrown.
Well, love is messy and complicated.
Yeah. Oh yeah, and here's the prickly pear flower with a little bee all up in it.
Aw. Well, I think that's a beautiful last shot to end on. Tori, if you want to just maybe wrap up with sharing something about the public education element of the site, that would be great.
Okay. So we have regular events on our sites. Every year we actually have a natural areas festival. It's been postponed because due to COVID the past few years, but we have a whole week long of events where we just try to hit as many sites as possible. And we take them in the wetlands, we take them in the uplands, we take them in the intercoastal to see islands that we've restored, and we have a lot of different types of education for children, adults, any age group. So definitely look us up if you're down in Palm Beach County.
Well, we'll put links in the chat for people to be able to follow along and see. I know your site has an Instagram with pictures of all of your wonderful creatures. We definitely want to keep tabs on that Gopher tortoise and see what they're up to day to day. So Tori Strange, thank you so much for joining us. It's been a real pleasure.
Of course. Yeah. And just to end, I want to show you one more cool species that's iconic in Florida. We have the air plants. This species is Tillandsia utriculata, and it can live up to 20 years old. And in it, it collects water for dry periods, and frogs, snakes, arthropods, centipedes all use this as a water source when it's really, really dry out.
All right. Thank you, air plant. We love to see it. All right, Tori. Thank you.
Of course. So good to be with you.
All right. What a great clip. I'm going to bring Tori back in with us now. Tori, that was so cool. Thank you for that amazing walkthrough of that ecosystem.
Of course. I was glad to share it.
So are you in that ecosystem right now?
No, I'm actually up in marsh, like intertidal area right now.
Yeah. Can we do a little pan around, see where you are?
Yeah.
Let me make that full screen here. There we go.
So I have mangroves and palm trees behind me right now.
All right. Very cool. Well, that was our first Gopher tortoise ever joining the Global Biodiversity Festival. So that was pretty cool. I'm glad we were able to see that. And what I really found fascinating was, talking about it being a keystone species and the burrows that they dig being used by so many other animals in the habitat. It reminds me, we just did a live event for students from the American Prairie Reserve. And we learned about how the prairie dogs do the same thing, the keystone species out in that prairie environment. So it's so neat to see the way that different animals are kind of adapted to the same role in different ecosystems.
Yeah, like analogously. Yeah.
Very cool. All right. Yeah. Well, to those tuning in, don't forget, you can share some more questions in the chat. We'll keep an eye out for them, but I've grabbed a few here to start. And so the first one is about ecosystems in Florida in general. So I think when a lot of people think about Florida, they probably think about the Everglades, maybe the springs, maybe the marine environment. Do you think, scrub? Do you think because is it tougher to convince people to want to conserve and pay attention to that ecosystem?
Absolutely. Yeah, I think it is too. Until people actually get on the ground and visit it and they figure out how enchanting it really is, a lot of people have hesitancies about going out there because they don't know it. There's a lot of thorny plants. It's very hot because the sand is reflecting the sun. So there's definitely a lot of things that keep people from the scrub, but once they get in there, they definitely see how magical it is. And it's such a great place to develop because it's high and dry, very well drained. You don't have to do a lot of land conversion to build upon it. So people kind of just say, well, it's empty space, not being used. So that's kind of the mindset that gets taken when they take the scrub and develop it.
Yeah, that makes sense. I mean, anything that makes the development easier always makes it more of a target. So
Exactly.
you may have mentioned this in the video, but about what percent of scrub is protected in Florida?
I read about 30%. But that is what remained after a lot of it had already been taken.
Yeah.
So I've also heard the statistic that only 5% of scrub habitat that used to exist actually remains.
Okay. All right.
So very small portion.
No, definitely, absolutely. Your role in the preserves do you find it's a big education role where you're trying to share, like you did with us today, the endemic species, the relationships, the gopher tortoise? Do you find that's a big part of your role?
I do. It's not only a role that I like doing because I think it's important, but it's also a required role. We do have the responsibility in as land managers to spend a proportion of our time educating the public. So when I see them on my sites and they ask me questions, I'll engage with them. And then we also have like special events where I try to cover as many topics as possible, and try to endear people to these species that they might otherwise be kind of hesitant around. And like one of my coworkers and I have really started this bug bio blitz, where we try to hit as many of our sites as possible and catalog the the invertebrate species that we see. And we get people there that would never have touched an insect before that. And we get them catching them and asking all sorts of questions, wanting to identify them to the species as best as possible. So I think it's really, really good way of getting people on board. And I mean, the citizens are the ones that are supporting these bills that are getting passed to give us the funds to be able to sustain these lands. So we need to get everyone on board with that because we want their vote.
Of course. Yeah, and I love that, getting people excited about the invertebrates. That's always an important thing to do.
Yeah, it's a little thing.
Awesome. Well, Tori, I can't believe how fast half an hour flew by. That was such a great tour of the ecosystem. Maybe just squeeze in, because you mentioned that these habitats are fragmented. Is there some effort underway to try and maybe create corridors between the scrub habitats?
There are some more inland, further inland, where there's larger swaths, like the Lake Wales region. When you're out on the coast where I am, it's done. The land between the fragments is so heavily converted and impermeable. There's no passageway any longer. So we just try to do our best to retain what's on there and, do our best to manage the land so that they can survive in what is left. Yeah.
Well, Tori, obviously a huge shout out to Kate for being a great host for that recording. Thank you for getting out into the field and taking us into that ecosystem, because, pictures are great, but seeing it kind of firsthand is one step higher, right? So thank you so much for doing that for us. Thank you for joining for some Q&A and being outside to do it again, because that's so important.
Of course. I'm happy to do it.
All right.
Yeah, well, thank you, Joe.
Tori, thank you so much. Have a great rest of the weekend. And hopefully we talk again soon.
All right, great. Sounds good.
All right. Thanks, Tori. Okay, well, what a great start to the festival, from Argentina to Australia to the United States. And now we're going to bring in another one of our hosts who will be joining us through different portions of the weekend. I'm going to bring in Lizzie Rosenberger here. Lizzie is an explorer. She is an amazing educator. And Lizzie, is this year 3 joining us, or is it 2 years that you've joined us?
I think this is year 2, Joe.
Okay. All right. Very cool. Well, last year was our marathon, 80 plus hours of broadcasting. So we were thrilled to have you joining us, and it's great to see you again and have you joining us. Lizzie, maybe just take a quick few seconds and introduce yourself a little bit.
Sure. So thanks, Joe. I'm super excited to be back with the Global BioFest this year. I first met Joe through Exploring by the Seat of Your Pants as an educator. So I teach science in the Boston area in Massachusetts, and also worked to bring an Antarctic expedition into classrooms before I moved up to Boston. And that's how I found out about Exploring by the Seat of Your Pants, and met Joe, and have been bringing students through to different experiences with Joe ever since then, which has been amazing. And every time the students get to go online with Exploring by the Seat of their Pants, they get very, very excited. So as an educator, this is one of my favorite things to do. And as in my personal life, I also love exploring as well.
All right. Very cool. Well, Lizzie, I'm going to disappear for a little while. You have a great triple header of events ahead of you. Have fun. And we'll see you soon.
Great. Thanks, Joe. So coming up next, we have a really exciting speaker joining us. We have Archana Anand, and Archana is an environmental microbiologist, and she primarily is working in the interface between environmental engineering and coastal marine biology. I'm going to bring you on in here so we can all see you. Hello, welcome to the Global Biodiversity.
Hi, Lizzie. Thank you for having me.
Yeah, it's wonderful to have you aboard. So we also hear that you're intrigued by aquatic microbes that drive several vital processes in the land and the ocean. So we're really excited to have you on board with us today and hear a little bit more about your work.
Of course.
So
Likewise.
I'm going to bring in your screen share for us now.
I just started the slideshow. Can you see it?
Yes, it looks perfect.
Okay.
All right, I'll let you take it away from here. And if those of you who are on our live stream, please feel free to add questions into the chat and we can get those answered at the end.
Thank you, Lizzie. Well, hi, everyone. Welcome to The Path of a Flush, Sewage Can Tell Stories. Before I begin, I want to thank the event organizer, fellow explorer and friend Joe, for inviting me. Now, can anyone recognize where this picture was taken? This is Marina Beach, very close to my childhood home. I was born and raised in Madras in India. I come from a family of teachers, and naturally the passion for education was instilled in me from a very young age. I spent most of my adult life in Singapore and Hong Kong, and I've now been in the San Francisco Bay Area for about 2 years now. So here's an overview of what I'd like to share with you today. First, I want to take you through my science journey from engineer to biologist. And next, I want to talk about how I spent the valuable years of the pandemic listening to stories from wastewater. And of course, what all that means for global biodiversity. I'm a mechanical engineer by training. and I graduated in 2007 with a degree from the National University of Singapore. I worked for a few years and traveled widely, but I wasn't very sure about what I was doing. And after a few visits to Africa, the Himalayas, this was with my partner and fiance at the time, I wanted to learn more about the environment and use education to conserve it. So I did the first thing that I thought might bring me closer to my goal, and I enrolled in a master's in environmental science at the University of Hong Kong. Now, this was where I met Professor David Baker. This is David on the floor. And I was intrigued by the work the Baker Lab was doing, looking at the impacts of human inputs on coral reef ecosystems. So we had a meeting, and I asked if I can do a project for my dissertation. David, who ended up becoming my PhD advisor, was skeptical because I was an engineer and he was a marine biologist. So we decided that a good starting point and middle ground would be chemistry. I was thrilled. The catch, I had to work on wastewater. The lab had just been successful getting some funding to look at nitrogen impacts on the marine environment. And they needed someone with a blend of quantitative skills and marine biology, basically me, to look into this, an interest in these fields. And so it began. Now, let me give you some context on why all this matters. The United Nations Environment Program estimates that over 8 million tons of marine litter reaches the oceans every year. Now, just 10 rivers carry over 90% of the global litter into the oceans. 8 of these rivers are in Asia. Everyone is talking about plastic. We've banned the use of plastic straws. This is great. But what about sewage? Why aren't we talking about sewage? Do you know where your sewage goes? Sewage is rich in nutrients, such as nitrogen and phosphorus. Now, nutrients are important, but when in excess, they can accelerate the growth of some microbes, and these microbes begin to respire rapidly and deplete oxygen concentrations in the ocean. This can have detrimental cascading effects on the life and the health of the ocean. Hong Kong is no exception to this. The city has a population density of over 7,000 people per square kilometer of land. Compare this with, say, Iceland, which has a population density of only 3 people per square kilometer of land. In fact, Hong Kong dumps over 1,000 Olympic-sized swimming pools equivalent of treated sewage into the ocean every day. Consequently, the impact of urbanization on the marine environment is stark. But why does this interest me? The reason this interests me is that despite all these environmental stresses, the marine environment is rich in life. Did you know that Hong Kong has many more hard corals than you can find even in the Caribbean? So what was the big question that kept me up at night? How does nitrogen pollution affect the ocean? And I split this big question into 2 smaller research questions. How does it affect life? And how does it affect health? Now, this was also when I got an amazing opportunity to lead a large collaborative effort with research teams from the University of Hong Kong and the Smithsonian Institution in the United States. But for all this to materialize, I needed an important skill: scuba diving. Now, this was hard work, but I got certified in open water, advanced open water, rescue diving, and scientific diving. And to date, these have been some of my best lived experiences. Now, coming back to the project itself, did you know that over 60% of life in the ocean is either unknown, unnamed, or invisible to the naked eye? And yet contributes to majority of the nutrient cycling on Earth. So to study these tiny critters, I used structures that were called autonomous reef monitoring structures, ARMS, or mini ocean hotels. And these mini ocean condominiums or hotels are about 9 inches in height. They are made of PVC plastic. They have gaps in between for water to flow through freely. And I scuba dive. I deploy these in the ocean floor for about 1 to 2 years, after which I collect and identify everything that has colonized the plates using photography as well as their DNA signatures. Now here's a video of me with my dive buddy Mike in Hawaii retrieving an ocean arms. And you can see we've placed a white bucket, secured it with bungee cords on the arms. And here on the right, you can also see another unit that's still soaking in the water while we retrieve this one on the left. The reason we're securing it with a white bucket is because we are interested in not just what is on the arms, but also what is in the water that is immediately surrounding the arms plates as well. Now, arms retrieval and processing by itself is a huge team effort because it allows for the involvement of so many students. I think my screen is frozen. Oh, okay, yeah. So you can have many students being involved, and not just students, but also the general public as well. So here I want to show a video of our lab retrieving the ocean arms and processing them in a huge citizen science effort where we're trying to build a genetic library of Hong Kong's marine environment. I hope you enjoyed that. This is one of my all-time favorite slides. It shows some of the organisms that we found on the arms from Port Island in Hong Kong. And interestingly, we found a higher abundance of pathogens in sites that had more nitrogen pollution. And we're still working through the data, and we have a lot of interesting things to report as well. Now, around this time was also when I had my first baby. This is Advik. He is now 5 years old, and he loves the water. Back to science again. Now, I had learned something about life in the ocean itself. So it was time to look at the health of the ocean. But before we talk about that, let's think about why the health of the ocean is important. Say we jump into the ocean and we see no sharks or any big fish for that matter. Is this a sign of a healthy ocean? Now, this actually might be an indicator that there aren't enough predators to keep the herbivores in check, which will either directly or indirectly affect several other levels in a food web in the ocean. So, long story short, I decided to look at predation in Hong Kong. And to do this, I used a tool called the squid pop. This essentially refers to a piece of squid, dried squid, that is attached to a metal stake and serves as bait for fish. I deploy them using scuba diving, and I leave them there for an hour, after which I return to look at the extent to which the fish have consumed the bait. And that will give me a sense of how healthy the ecosystem is. Another thing that we looked at was decomposition, essentially how quickly and also how effectively microbes are consuming organic matter. These are the scavengers. in the ocean, and they also play a very, very important role storing carbon. So to do this, we used tea bags, the ones that you used to drink, green tea and rooibos tea bags. This is me with my dive buddy Taehoon and also lab mate. So we are burying tea bags. We leave them there for 3 months in the sediment, after which we come back and then look at how much the microbes have consumed the tea leaves. We simply weigh them and look at the mass loss. And interestingly, we did find sites that have more nitrogen pollution. We found that those sites did have much higher rates of decomposition and poorer rates of carbon storage capacity. Now, coming to mid-2020, I moved to the Bay Area right in the middle of the pandemic. And shortly after that, I had my second baby. This is Adit, and he's a pandemic baby. That's why he has a onesie that says, "My parents did not practice social distancing." He's also a dancer. And so this brings me to the last part of my talk. The pandemic was in full swing, and I happened to be at the right place at the right time. So owing to my experience with wastewater and also looking at microbes in the environment, I got sucked into some amazing projects. And this was at Stanford University. So here, I'm now going to be spending the last few minutes talking about wastewater-based epidemiology. So to begin, can anyone recognize this person? This is Jon Snow, not from the Game of Thrones. He is a British physician. In August 1854, there was a cholera outbreak that occurred in Soho. At that time, it was assumed that cholera was airborne. But Dr. Snow did not accept this theory. He argued, in fact, that it entered the body through the mouth. And he was convinced that sewage that was contaminating the river was the source. So after carefully investigating, he plotted the cases indicated in red on a map with pumps, water pumps in the area indicated in blue. And he was able to identify a water pump on Broadwick Street to be the source of the contaminated water. He had the handle of the pump removed, and the cases of cholera began to diminish. This theory, of course, was not accepted until the 1860s, but today he's considered to be one of the founders of epidemiology, which essentially refers to the study of diseases in populations. Now, disease monitoring in wastewater took off since then, and this has been used to gauge aspects of public health like narcotics usage. In fact, in India, the government officials could see the poliovirus re-emerge. They managed to contain the outbreak 90 days before it appeared, because it appeared 90 days before that in wastewater. So this kind of an application has accelerated interest in wastewater-based epidemiology, and the pandemic has continued to do that. Why? Because we see SARS-CoV-2 genes. SARS-CoV-2 is the virus that causes COVID-19, and it's detected in the feces of many individuals. Now, this is amazing because you can track the virus in the wastewater at least 1 to 2 weeks before an infected individual begins to develop symptoms, or he or she approaches a hospital, or perhaps a hospital begins to see a patient as an ICU admission. So, but when the pandemic began, we weren't sure about this. We weren't sure about how to use this tool effectively. So at Stanford, the team that I was involved in began to look at two big things. The first was, can we correlate the cases that we see in the hospitals to the concentrations we're seeing in the wastewater from large sewer sheds, from huge treatment plants? The answer to this was yes. We looked at sewage treatment plants from the East Coast, the West Coast, as well as the Midwest United States, covering about 9 million people. The next thing that we did was we also realized that smaller workplaces, and universities had to close because they could not detect or contain this higher spread of infection. So we looked at sampling wastewater in dormitories, and we realized that it could also be an effective tool to catch cases even when the sewer shed contains fewer individuals than large treatment plants that have millions of residents contributing wastewater into them. So with that, that was the beginning of everything that I did in terms of wastewater. And to end this presentation, I want to talk about what I'm doing right now. At the moment, I'm a postdoctoral researcher at Columbia University. And here I'm looking at not just SARS-CoV-2, but also other pathogens in the wastewater. So imagine if you were in an airport, you can see a dashboard with different flights that helps you get a snapshot of what flights are going where. So the idea is for us to build a dashboard in maybe a university campus and get a snapshot of community health by having a list of different pathogens that are present in the sewer network. So that's what I'm working on right now. And fast forward to 2022, we got a puppy who we fondly call Taco. And I'm very grateful for my wonderful, wonderful family. Of course, this journey has not been problem-free. I've had several hurdles, both personal and professional, but I'm super grateful that the science continued to grow through all this. And I'm here today sharing this journey with all of you. So with that, thank you so much for lending me your ears, and I'm happy to take any questions.
Wonderful. That was so fascinating. I loved hearing your whole journey from when you started all the way back as an engineer and how you got to where you are today. A couple of questions that I've got for you. I'm super curious, just based on what you're working on today with your postdoc. You mentioned that it is kind of could be a snapshot of community health at a campus level, but would that ever then be able to be extrapolated to community health beyond a campus? So for a city or a town or a different type of community?
Thanks for that. Yes, that is the idea, for us to pilot this in a campus and look at a couple of pathogens like SARS-CoV-2, influenza, or maybe some food pathogens, and then see if we could do the same thing across a city, say, like New York City. And the idea would be, yes, for us to do that not just efficiently, but also quickly.
And how long do you think that this kind of initial test phase of the project is going to take?
Well, we just got funding for the project. So it's at a very nascent stage right now. And we're gathering resources, potentially recruiting team members to get involved. And we've got initial funding for a year. So we hope to make some progress within the year and then see what to do thereafter.
Exciting. We'll have to stay tuned to see any publications that you guys come out with. I think that'll be really informative and wonderful on so many different levels. The video that you shared with us about your work in Hong Kong and involving the citizen scientists and the community, how, like you said, we would see how intrigued everyone was and how engaged they are. How do you go about kind of publicizing or advertising for your citizen science projects? And how many people would you say that you got to help out with that project?
Thanks for that. That was actually one of the fun aspects of my PhD, because we got to interact with so many people and they had wonderful questions. So at first, the government of Hong Kong is extremely cooperative, and we have a very good relationship with the government officials. So most of the time when we have a project like this, we reach out to them, not just for permits, but also to share what we plan to do. And they often circulate this information. They put out posters that they would let the general public know. In addition to that, we also have our own network. So we do advertise within the Department of Biological Sciences, and we have relationships with the Hong Kong International School or other schools. So the teachers would often reach out to us if they would like some students to join, and for that effort to be part of some kind of teaching module in their classroom. So the video that you saw is actually involving about, I think, 30 students from the Harbor School, and they're all middle school students.
Oh, that's wonderful. I'm sure it just sparks their interests immediately into what they can do with themselves in the future as well.
Yes, exactly.
Kind of along that same lines, all the different sampling techniques that you have. So the hotel, the arms, and then the squid bot and the tea bags, are those things that you and your fellow scientists come up with on your own, or are those traditional sampling techniques that are widely used with everyone? Or kind of how do you choose how you sample what you're looking for?
The short answer to that is it's a combination of both. So the arms is something that originated here. I think it was NOAA that came up with the unit itself. At first, it was made of concrete, and then it was worked on, and then Smithsonian Institution took it up to come up with a standardized model that could be reproduced, perhaps in other factories across the world. So any data that you generate from one arms can be compared with one from an arms somewhere else as well. So the squid pops and the tea bags, the squid pops were also sort of part of like a protocol that we worked with the Smithsonian Institution. The tea bags we did kind of improvise, so it's something that was new and has not been used in the marine environment. So that was something that I worked on and came up with.
It must be so fun to kind of come up with your own inventions of how to sample for exactly the data that you're hoping to collect as well.
Yes, it was used in, the tea bags were used by researchers in mangroves and other on land, but not so much in the ocean. So that was the tricky part.
Yeah. And would you, I know that you kind of mentioned that we don't really talk about wastewater and sewage very often, but how important it is for our Earth and for our globe. So what would you kind of say to people about how they can get involved or how to learn more about what we can do to help or what we need to be more aware of?
Oh, thank you for that. Well, I think the first thing to do, I tell my students, is to find out where your sewage goes. Do you know where your pipe goes? For them to just track it. And more often than not, we don't know whether you have a septic tank at home, is there a treatment plant, or is it just going to the river or the ocean? To just be educated about what happens after you flush the toilet.
Yeah.
And once that happens, I think individuals tend to get more curious about what gets flushed down as well. And you tend to become more aware. So that's the first thing in terms of awareness. Otherwise, I think in general, to be more involved with maybe beach cleanups, or perhaps talk to an NGO nearby or a university, see if you would like to be involved, because we often need help from the public, and the public that actually visit these areas know much more than we do because they've lived there for so many years. So it's just about connecting with your nearest, I think, science group or maybe an organization. Yeah, and seeing how you can help.
Awesome. I think that's great. And what a great piece of advice that we should all, I challenge everyone who's watching to go and find out, where does your sewage go? Where does that pipe lead to? And then learn more. Thank you so much. This has been a fast half an hour, but a very, very fascinating one. And so wonderful to hear about your journey and where you are today, and see the personal touches of your family as well. That was wonderful to see. So thank you so much for joining us today at the Global BioFest, and we'll stay tuned to see about a year from now what happens with your current research.
Thank you so much, Lizzie.
Yeah, have a great night.
You too. Bye.
Bye. Wow, that was so wonderful. I feel like I learned so much more myself. So our next speaker that we have coming up is, we're taking a different turn from sewage and wastewater, and we are heading to the world of photography. So we have Pedro Peloso, who is an award-winning field biologist and wildlife photographer for a passion for wildlife and wild places. He, through his work, has helped discover and help name over 30 new species of animals, most of which are part of the Amazonian frog. So hopefully, I think we might see a couple of pictures of some of those. Pedro is also the founder and director of the Documenting Threatened Species Project, which is a bold initiative that combines science, art, and storytelling to promote conservation of endangered amphibian species in Brazil specifically. So I think today we're going to hear some of the stories and images of just that, what he's doing to help document and protect biodiversity in the rainforests of Brazil. So with that, I'm going to go ahead and bring in Pedro. Hello, Pedro.
Thank you, Lizzie. Can you hear me all right?
Yes, we can hear you loud and clear.
Awesome.
Welcome to the Global BioFest.
Thanks for inviting me. You and Joe.
Yeah, we're thrilled to have you today and to see some of your work and learn a little bit more about what you do.
Cool. Awesome. Thank you.
Yeah.
I'm ready. Yeah.
Great. I'm going to go ahead and pull in your presentation and I'll let you take it away.
Perfect. Awesome. So thank you very much for the very kind introduction. And as you said, I am a researcher and a conservation photographist. So it's good that in some expeditions, I get to be a biologist with a camera. But in some other expeditions, I am a photographer with a background in biology, and that helps me a lot to understand the animals and to photograph them in different ways and get to know their behavior a little better. So I try to do a sweep throughout my career, and I'll try to do that very fast because it's almost 20 years working on tropical forests, and this is where I started. So I've traveled mostly throughout South America, but also Southeast Asia. And these are some of the places that I've been to: Atlantic forest, isolated islands in the Atlantic Ocean, the high Andes, where the Amazon actually starts, the first forest of the Amazon in the eastern slope of the Andes. But my biggest passion is actually the Amazon, and I've worked in the Amazon for about 15 years. Ish, a little bit less than that, but that's where I've concentrated most of my effort. And throughout these expeditions, I've had really close encounters with a lot of very cool and rare and endangered animals. And this is one very good example where I go after the animals and then try to be in a place where I can get photographs, such as this one of this Andean bear, the only bear species in South America that's now endangered with extinction. My initial passion was with reptiles. I wanted to work with reptiles, but at some point in my career, I switched over to frogs. But and more, I got interested in showing different angles and different images of rare animals. So this is a very rare pit viper, only known from a single island in Brazil. This is another example where I put myself in situations and in places where I can get photographs of very, very rare animals. So this is the world's rarest toucan. It's only known from very few localities in Peru. It's highly endangered. There are very, very few pictures of these toucan available in the internet, and I was able to spend a couple of days with it in Peru. And because I always had a camera and I share some of my images in meetings and in talks and in social media, I've learned that depending on the picture and depending on the story you attach to that picture, you can really grab the attention of people to the animal or to the situation and stuff like that. So I've been using my photography to actually highlight the animals and highlight these conservation stories or research discoveries and stuff like that. But as you said, most of my work is concentrated with amphibians, and that is now my biggest passion. So there are around 8,500 species of amphibians in the world, many different forms, colors. There are all sorts of varieties of amphibians. Most of the amphibians are frogs. In South America, we have very few species of salamanders. I'm sure in Europe and North America, there are lots of them. But I've been traveling a lot and enough so I can see all of this diversity of amphibians, and what I get to do now is just share these images with people so that people know about this diversity. Because if I just say there's 8,000 different species, that's only a number. But if I attach pictures and different pictures of these animals, and then people can start to relate with these animals a little bit more. And even like very common species like a common toad, if I show it in a different angle with different lighting, so it brings a different sense of beauty to these animals, and people can start to care about them a little bit more. I try to also document behavior, so this is useful for research. Many colleagues have used my photos and my images for research in natural history and ecology and taxonomy as well. And I'm lucky because I am in Brazil. So there are over 1,000 species of amphibians in Brazil, which means that sort of like 1 out of every 8 species occur in Brazil, and that's the most in the world. Roughly, we have 4 times more species of amphibians in Brazil than we have in the whole United States. So that's a huge diversity, and that's also a lot of opportunities. And I've been photographing and documenting these animals for quite a while, and I have collected images of very cool animals. And there are some species in Brazil that are very unique and that people do not know about. This is a very cool example. This is the only species of amphibian that relies on vegetables for a huge part of its life. So it actually eats fruits. So for part of the year when insects are not that abundant and there are these like tiny red fruits that are available, this animal eats fruits. So that's the only known amphibian that eats fruits for as a primary source of its energy for a big part of the year. This is another very cool animal, and it's allegedly capable of injecting venom into predators or prey. It has never been observed in nature, but it has all the mechanisms to be able to do that. Sort of like a venomous spider, venomous snake, but in a very different mechanism. So all amphibians have venoms in their skins, but this one is highly toxic, and it does have a mechanism to actually inject that venom. And this is known only from a Brazilian species. But the thing is, working on a very diverse environment means that a lot of it is still unknown. So there was actually a recent estimate that about 50% of all amphibians may still be unnamed, which means these are species that are not yet fully recognized by science. And this has a lot of implications that I will explain a little bit down the line. And that same paper that brought out this estimate, they pointed out some places where most of the future species are likely to be discovered. And Brazil is like this huge place with tons of species to still be discovered in the future. And as you said in the introduction, I was lucky, and I've been working with several colleagues, and I have participated, either led or collaborated in the description of over 30 new species of animals, mostly frogs, but also some lizards, most recently a bird. And you will hear about a praying mantis, I think, tomorrow, and that we're collaborating on that too. So that's a lot of animals. And this is just some of the examples. This is a tiny frog from the Amazon that we named after our colleagues, the Kayapó indigenous people in southern Amazonia. This is a tree frog that we named after Ozzy Osbourne, the famous musician, because it has a very high-pitched vocalization that remembers a bat, and there's the whole story about Ozzy Osbourne biting the head of a bat, and that's the association there. And this is the most beautiful frog I've seen in Brazil, and this was also a new species that we named after a mythological character in Brazil. the Mapinguari, and it probably is the legend of the Mapinguari is probably based on very ancient sightings of giant sloths, and that's a big legend in the Amazon, and that frog is from the Amazon, so we just associated the name. So we can get pretty creative with species names. But the one of the main issues and things about taxonomy is that all of that takes a lot of time. So on average, it takes 20 years between the time a species is discovered and when it gets its actual name. So that estimate, let's say we know that 50% of all diversity of frogs is still unnamed. And if we started now, it would take probably 20 years to name all of that. And the reasons is that many of these species require a lot of work to be described and named. So some of them we can do it only by morphology if they're very distinct, but for some of these species we need additional work like anatomical work or comparing vocalizations or doing genetic studies. And we all know that a lot can happen in 20 years. So I'll just show a few pictures of complete destruction of natural environments. This is in the arc of deforestation in the Amazon. That's a lot of deforestation, and the border there, the river there is the limit of an indigenous territory. This is this deforestation in the Atlantic forest, same situation, forest is gone. This is illegal gold mining in southern Amazonia as well, near that other photo of the indigenous territory. This is salt mining on the coast. So a lot of the coastal species are also threatened because of this and because of expansion of urban areas. Mining on mountains. On top of that mountain, there's a species that lives only on the top of these mountains, and its habitat is being reduced. And the Amazon is now being reduced to these small islands. So I'll show you another example of a species we discovered and just recently named, which is Adenomera phonotrichus from southern Amazonia. This is where the species was discovered, and if you look at the arrow in the center of the image, that's the patch of forest where we found that frog. But that was the forest 30 years ago when I was born in 1985, and this is how the forest looks now. So it's pretty easy to see that most of the habitat of that species is gone, and it's very likely that this species will be listed as endangered with extinction in the very near future. So the biggest message that I want to live now with you is that we are losing species before we get to know them, and this is a fact. Because if it takes on average 20 years for us to know to name these species, it means that we can take too long, and in 20 years we can have a whole lot of forests destroyed. And if these species have very narrow distributions, and it is the case with many of these species in Brazil, they have like only known from a very narrow patch of forest or a single locality, we are going to lose these species. And frogs are amphibians in general are a lot more sensitive than other animals. 40% of all the amphibians are threatened with extinction in some level. And just to be clear here, what I'm talking about threatened is what the International Union for Conservation of Nature, IUCN, use these categories. And we can have species that are not endangered, not threatened, or they're extinct, or they're in these three different categories that scientists use to categorize them. I'm sure most of you know this, but some people may not. So these are the main categories of endangered species. We currently have only 42 species included in the Brazilian red list based on those criteria. So we have 41 threatened species, one that is officially considered extinct, and at least 167 species, that's almost 200 species that we don't even have enough data to know if they're threatened or not. And that's a problem because a lot of these data deficient species are also endangered. There's a very good correlation that these species are going to be endangered. And these numbers are certainly outdated. So the new list that should have been published already, but the government is taking too much to approve and to publish this list. The list has been done since 2018. There are at least 15 species that are possibly extinct. And the list of endangered is a lot bigger than these 42 that are officially protected by law. So that's how I envisioned and created the Documenting Threatened Species project, which is this project that I'm involved now that pretty much combines all of this background that I have in amphibian biology and photography. So I created a project, and our idea is to go to all the places where these endangered species are and have them documented in photos and videos and study them and document the threats to these species and so on and so forth. And so far we have conducted 8 expeditions, and this is sort of how they're distributed in Brazil. And you all know that Brazil is huge. So getting a project and getting all the money and time to go and look for all these species is a huge and monumental effort. But we've already conducted 8 expeditions and documented 14 species, 14 or 15. I forgot the exact number, but it's a lot of species. And I'll just quickly show some of these pictures, and these are all very rare, highly endangered animals. This is critically endangered. This is one of the rarest species in Brazil. It's only known from a single river, a single small river in a single island in the Atlantic, and it's a very rare species. It's very highly endangered. This is another critically endangered species, also only known from a single river. This one in southern Brazil, and you can see the river is there, and there's a little bit of forest, and the species is there, but you can see there's already deforestation in the middle of the area there. And this is an example of how this all sums up a little bit of my career and how I got involved with all this. So this is a species I discovered when I was an undergrad student in Brazil in 2005. And it took a while for us to name it. It didn't take the whole 20 years, but it almost took a decade for us to be able to actually describe and name it. And we named it in 2012. And in 2014, it was already included in the Brazilian list as critically endangered with extinction. Just a few more examples. And these are all images that we collected for this project. All very rare. There are very, very few images available of all these species. So that's another thing. We're starting to work with conservation. I would Google and look for images of these species, and there were none or very few good images that we can use in books and publications. So that was one of the drives that I had to go to the field and photograph all these species. And in addition to just photographing and having this database of images, we're also doing a lot of stuff. When we go there, we do some research. So we take swabs, we look for infectious diseases, and we have students that are working on different species that we already documented. We do a lot of communication. So there's a lot of press. There's a lot of press that people do on our project. We write small articles, and we also write op-eds and stuff like that to actually put out the images. And now these new images, these creative artistic images, are associated with stories, and we've been able to highlight a little bit more about this species. And one of the coolest aspects of the project is that we're partnering with artists to have a bigger conservation impact. So the two on the right there with Bia Mauricio and Biome Conservation, we're actually having products that when we sell these products, some of the money is converted either to the DOTS project, my project, or directly to people and scientists working with a particular species. And one final very important aspect of what we're doing is education. So we try to, as much as possible, get especially kids involved. So we have people going to schools and giving talks and getting kids involved. We've promoted bio blitzes and I've getting dots involved in many different aspects of education in general. And we're partnering. There's a lot of cool stuff that's probably going to come out this second semester of the year and next year, including books and musics and stuff like that. But that's all a surprise, and it'll come out soon enough. And the reason I do this is so that I can come back to the Amazon and have these frogs protected, and I need the environment to be there for our future generations. With that, I end my participation, and thank you very, very much.
Thank you so much, Pedro. That was really beautiful to see so many of your images, and just amazing. I can see exactly why you've fallen in love with the frogs, as well as the other amphibians as well. There's a question that has come in on the chat for you that I also was wondering, especially when you can see on your finger some of the super small frogs around the Brazilian coin, how small that frog was. So the question is, how do you find these tiny frogs? It must be really hard to spot them when you're within their surroundings. So how do you actually find them?
It's different for every frog. Well, it's different for every frog, but a lot of them we hear them. So I've been literally frustrated for 3-4 hours hearing something, and I know it's there, and it just basically. Most of these animals are there at night, so it's very old-fashioned flashlight looking for them. But when they're calling is a lot easier because you know they're there. But it's also frustrating when you don't find them. And many times you're looking down and they're up, or you're looking on the leaf litter and they're burrowed inside soil and stuff like that. But every frog is different. And the tiny ones, you just need to like kneel and just search for them for maybe a couple hours, and you'll eventually find one.
You must really love searching in the leaf litter.
Oh yeah, I love it. Yep. And you find bugs, and you find cockroaches and spiders, and you find all kinds of cool animals too.
Yeah, so it's not just the frogs. Another question that came in from our Instagram feed is there a frog species that you really want to see but you haven't yet? And if so, what does it look like?
There are a few. So there are these marsupial frogs that carry the babies on their backs, and I know where they are. I've been to an area where they were there, but I never got to see them in the wild. So these marsupial frogs with actually carrying babies. I've seen them carrying the eggs, and I've seen the males and females, but not with the babies. But I want to see those with the babies. That's the one I'm missing. A lot of these frogs for the DOTS projects, a lot of these are frogs that I've wanted to see for a long, long time. Like the one that ate fruits. This was on my list for years, and then this project allowed me to go there and actually see that. So that's a quite cool aspect of this project too.
And how many people are working with you on your project? So I know you said you have had 8 expeditions so far, but how many people actually go out into the field with you?
It also varies a lot. So the core team of the DOTS project is around 8 people. Some people are on and off more involved. 3 of us are always involved since the inception of the project. But I go in all of the expeditions when we're searching for a frog for the first time or doing the actual documentation and photos. But for example, we have a team working on the monitoring of one of these species in the Cerrado. So there's like 5 or 6 people involved in just that aspect of the project. And whenever I want to go, let's say I'll do an expedition in December and I want to see one frog. So I'll contact people there, local people and say, how many people can we bring? How much budget do we have? So it varies a lot. So we can have like 2 people up to 10 people. We've had an expedition to one island where we were, I think, 8 people. And all of these expeditions have somebody that's already working with the frog. So I don't want to just go there. Oh, I'm the photographer. I'm going to go there, find it by myself and photograph. Because one of the things I want to do too is highlight people that are already working with these animals. But it varies. It varies from very few to as much as we can allow to bring in the car or boat or the budget allows for.
And how long do you usually spend in the field? If you're spending hours looking for a tiny frog, is it a couple of days, a couple of weeks, a couple of months?
Yeah, we can have long conversations about how crazy these expeditions for this project are. So the first expedition it was pretty much a tryout. That we had the idea, we didn't know, we don't know if this is going to work or not. So we planned to see 5 species, 5 different localities in 4 days. And it was crazy. Just like drive, try to find the species, photos, and then drive, and do the same thing for 5 days. We were exhausted by the end of it, but it was nice. And we didn't have money because we didn't have. Now we have money for some expeditions. Now it's getting better, but the first ones were just sleeping in very bad hotels or in other people's couches and stuff. But we usually plan for 2 days per species.
Oh wow!
Because the thing with these species is that they're endangered, but people know they're there. So it's not that they are rare where they occur. They only occur in 1 or 2 places. But usually because they're endangered, people know where they are. There's at least 1 or 2 people already working there. So that's why I contact the specialist and I say, no, it's in this creek. This is the GPS coordinates. We just have to go there and look, we're going to find. And for the documentation aspect of it, we just need 1 individual. If we find 1 individual, we can do magic and have like 100 photos and videos and whatever we can from that 1 frog. So we plan for 2 days, but we usually have multiple species per trip. So if we find it in the first day, cool, we can have 3 days for the next, and that's how we sort of roll. Unless it's like a very remote place that we have to plan a little bit more. So we went to that island where that species is in the island. So we were there for 4 days because we had to guarantee, because it's a lot more money to go there and we have to have success in that single trip. Some of these places we can come back and stuff.
Yeah. You have a lot of shout outs on our YouTube channel of how wonderful your work is, the beautifulness of your images, and just wishing you strength to continue your project in the current Brazilian science situation. And there's another question, and I might pronounce the name of this fungus wrong, so please forgive me. But the question is, has the citrid fungus or DB affected most species in the Amazon, and also have the scientists managed to control it?
So BD is not as common in the Amazon, and it's actually more problematic in the Atlantic forest, and there is solid evidence that some of these species that probably are extinct were killed by the fungus. So that's a problem. And one of the things that we do is whatever place we go, we try to swab and see if the fungus is there. So far, we haven't had a lot of species with the fungus, but some, yes. But it seems like some species can handle it and they're stable, even if they're infected. But there are some species that are probably gone, and it was because of that. But that was like some decades ago. But they're gone now. It's too late. So this is an aspect that we're dealing with, yeah.
And on your website, just to give us a quick overview of the website, if we go to visit your website, can you tell us what the name is so we can search it later? And then what will we find both your beautiful images as well as the research you've conducted and what your maybe places you're hoping to go next?
Yeah, so my website is my name altogether, pedropeloso.com. And there you'll find a mixture of a lot of things from research. You can download all the papers that I've been involved. I've been trying to upload it, more things and update it, but mostly research, some photos, some curiosities, all the people that work with me, like students. I have a bunch of graduate students that are amazing. They do most of the hard work now that I've done in the past. They do it now. And there's a link to the Documenting Threatened Species project that is a bigger independent website where you can find more like direct details on the endangered species and more photos about them. But not all the information is there. We're renewing the website and uploading all the photos, but we have 10 to 12 species already there.
Awesome. Well, thank you so much, Pedro. This was wonderful to have you be part of the Global BioFest and learn more about amphibians in Brazil, specifically the frogs, and that we need to find even more and try and protect them all. So
Yes.
Thank you so much for sharing all of your beautiful work and research with us today.
Thank you, Lizzie, very, very much. And thank you, Joe. This is an amazing festival. I'm looking forward for all the talks for today, tomorrow, and the day after that.
Yeah, they're going to be amazing. All right. Thanks, Pedro.
All right. Bye-bye. Thank you.
Bye. Wow. Yet another amazing talk and hearing about all of those fantastic frogs. There's some that were so small. So I just wanted to give a quick shout out to some of our sponsors. I'm going to bring in a video from Lenovo that is one of our festival sponsors this year. Awesome. So Lenovo, definitely check out Lenovo. Wonderful organization. So for our next speaker, we have got Chloe Boot, who is a coral scientist and a PhD graduate from James Cook University in Australia. She's originally from the UK and volunteered in aquariums and teaching reef ecology in Honduras before her PhD. So Chloe is going to be joining us, I believe, from Australia today, and we'll bring Chloe in to confirm. Are you in Australia?
Yes, I am in Australia. I was going to say good morning from Australia, although I think it's your nighttime now. But yes.
Yes, it's nighttime for me, but it's tomorrow where you are.
Yeah, well, thanks for having me so late.
Yeah, we're very excited to have you today and hear more about all of your work with coral reefs. So with that, I will bring in your screen, which looks good to go, and I'll let you take it away.
Okay, well, thank you. And firstly, I just want to say a big thank you for having me. My name's Chloe, and I work at the University of Technology, Sydney. And I work with Emma Camp, who was meant to present today, but unfortunately got COVID. So I'm a last minute fill-in, so I hope this is okay for you all. But today I'll be talking to you about what I did on my PhD, which was with James Cook University and the Australian Research Council Centre of Excellence for Coral Reef Studies. Now, firstly, a little about me. I did my undergrad in the UK at Portsmouth, and I managed to get funding to go do an honours project out in Indonesia, where I was looking at the heat stress of Porites lutea, a particular coral species, which was a really exciting experience because I had to learn to basically build all my own equipment because they don't have a lot out on a remote island in Indonesia. And this went really well. And following this, I went to do my master's at University of Essex. Again, I did a little trip to Indonesia, but I also did a project on looking at heat and light stress response in a coral called Aquapora digitifera. And this is where I started getting into more genetics because I'm really obsessed with genes and microbiology, and I think it's all really cool. And And then again, following my master's, I went out to Honduras, where I worked as a reef ecology lecturer, teaching students how to do reef science. And also then I went to both the London Zoo and Horniman Museum and Gardens in London, and I volunteered as an aquarist, so working in an aquarium, and also doing outreach and talking to people about conservation. Now, following this, I was very lucky to get a scholarship to James Cook University. So I packed my bags and I moved to the other side of the world, just like that. Just thought it'd be a good decision. And I went out and I got to do all my field work in Orpheus Island. So this here is Orpheus Island, absolute beautiful spot of the world with amazing reefs. And I got to study these, and they've got fantastic facilities. So I could do a PhD looking at coral spawning and developmental biology. And then also a big thing of mine, like looking into coral bleaching, and again, that genetics and microbiology. So currently, so I completed this in February, and currently I'm working as a research assistant at the University of Technology, Sydney. And here I get to do cool things like reef carbonate budget surveys, where I get to like survey the reef and see really cool fish. And I also get to do some stuff with the Coral Nurture Program, which I believe Emma spoke to you guys about last year, outplanting coral fragments on the reef, trying to regrow the reef, with lots of amazing people for different tourism operators out there. And I also get to do some really cool stuff in the lab. So yes, so that's a little about my background and my journey. And then now I suppose I'm just going to talk to you about my PhD research, get into the nitty gritty of the science I do. So firstly, reefs, coral reefs, they're super important, beautiful ecosystems and so biodiverse. They support thousands and thousands of marine species. They support us with coastal protection, food, livelihood, and they also do an amazing amount for nutrient cycling in the environment, which is really important, particularly in today's climate. And they also have some cool products on them for biotechnology, so looking into medicine and future products and all this nifty kind of stuff. And they also, though, unfortunately, are threatened quite severely for numerous different threats. So we have local threats like over-exploitation, oh God, I can't say the word, so like overfishing and pollution runoff and invasive species. And then we also have climate issues like ocean deoxygenation, ocean acidification, and also ocean warming, which has always been a big thing that I've been concerned and want to learn more about. So this beautiful reef here in Indonesia, and then unfortunately we're seeing this rising ocean temperature, which is leading to large scale bleaching across the globe. So here is a picture that Greg Torder took during one of the JCU bleaching surveys in 2017. This is me just swimming over the reefs at Orpheus that bleached then. And it's really sad to see when you're like, love this ecosystem, that you get these beautiful, colorful reefs, and then suddenly all the color's gone and they're not very healthy. And it's funny, this image has been snapped up by a lot of media people. I see it on articles all the time. I'm like, oh, it's me. But yeah, so we get these reefs and they bleach when they get too hot. And unfortunately, if they can't recover from the bleaching, we actually see that the corals will die off and we lose this reef. We lose all this biodiversity, all these big, beautiful structures. They're gone and they'll degrade down. And we no longer have all these amazing, ecosystem functions that we get from reefs. So my thing was like, oh, why is this happening? What is that genetic mechanism, or what microbes are involved in this bleaching response? And there's lots and lots of theories about it. But one of the things that I was really interested in is the fact that corals have two tissue layers. So us as humans and other mammals and we have three tissue layers, but corals, because they're less evolved, they have their outer skin layer, their ectoderm, so that's their outside layer, and then they have their internal layer, which is their endoderm, and this is separated by a sort of liquid barrier, the mesoglia. But the really important thing is that these Symbiodiniaceae or zooxanthellae or the photosymbionts, they live within this endoderm. So they live in the inner tissue layer, and they live in symbiosis with the coral, and together they are really important for coral health functioning. And the coral pass products to the algae, the Symbiodiniaceae, and the Symbiodiniaceae pass stuff back to the coral. So what we have when the corals experience heat, unfortunately, they get really stressed and they start to produce toxins that are really bad to be in their cells. And then, so from this, the Symbiodiniaceae are then either expelled out of the coral or they are engulfed. And we don't particularly know exactly if it's the host that kicks this off, the coral, or if it's the algae. And we really wanted to look into this endoderm region because it contains the Symbiodiniaceae. So we're like, well, this must be the point where bleaching might happen. But it's really hard to study in typical coral models that we have. So the photo I took of Acropora millipora, again on Orpheus Island. Now this is the thing, so colonial corals, or most normal corals that you see on the reef, they're made up of thousands of tiny, tiny little individual corals living together as one big colony. So kind of like a beehive, like there's all these tiny, tiny little polyps, corals living together, and they're working together, and that's how they are. So it's really hard to look at the endoderm when it's so, so, so small. So we had this idea of creating a new novel coral model, and this is what we proposed. This is Heliofungia actiniformis. Now this here, it looks like a sea anemone. It looks like a giant sea anemone, but it's not. It's actually a coral. So this is where this unique thing comes from. It's good to be unique. It's good to be weird. This coral is just, you know, it's got these big tentacles. It's large. And the really interesting thing is that because it is so big, this is here an adult, about the size of a dinner plate. It's bigger than my head. And it's one large solitary coral. So as I said before, with the polyps that are all interconnected, they're all living together, they're really small. This is actually just one huge coral polyp. And the other thing that's cool is it's not attached to the reef. So most corals are attached, they're stuck on the reef, but mushroom corals, which Heliofungia belong to, can actually move around the reef really, really slowly. And then Heliofungia is very unique within mushroom corals because it's got these big tentacles, but it also has one really big mouth that is so large, it can actually eat fish. So this is a photo taken, I think, from National Geographic in 1973. This is a tiny little damselfish having a really bad day because it's about to be eaten by a Heliofungia. So yeah, that's all what my PhD was about. For the first time in coral biology, we could physically separate this ectoderm and this endoderm, which was methodology taken from one of my supervisors, Aurélie Moyer, that she did on anemones, and we applied it to Heliofungia. Also, whilst working on it, I realized I could look at different regions. So I could look at the mucus, which is this outside layer on the coral, like this secreted slime all over the coral. I could swab it and have a look at the different microbes going on there. And then also I could look at that ectoderm, the outer skin layer, and the mesoglea is that liquid in between. And then we have the endoderm with the Symbiodiniaceae living within it. I also realized during my PhD that Heliofungia are unique as they have nematocyst bundles or acrospheres. So this is all the little stinging cells that live in corals and jellyfish. Rather than being throughout the coral, they're actually all isolated to these little tips of the coral. When you go near it, it's like a medusa head, like they all come towards you to like sting you with them. And also I realized that I could actually sample the gut because it's so large. And finally, another thing that comes from using a big coral is that it's really good for microscope work. So this is a microscope image. I think easiest thing just to take from it is that this is one of the tentacles. This is a slice of the tentacle skin. And what we can see is it's really clear. This is the ectoderm, this outer layer. We've got this mesoglea in between, this like liquid barrier. And then the endoderm here, this inner layer, which is packed full of Symbiodiniaceae. So those tiny little algae photosymbionts that live in the coral. They live here in the endoderm. And it's really good because you get such good resolution because with all the tiny coral, you know, the colonial corals, it's all so tight and smushed together, it's really hard to see. So what did I do? I had a non-destructive method of sampling. I'd collect my corals and I had multiple sampling points from 2016 to 2018. And I would go collect my corals and I'd snip off a few of the tentacles, which sounds a bit mean, but it's kind of like cutting their hair. Like they're used to losing them in the wild. It's not a big deal for them. And I would take their coral tentacles and I would dissect it into the different tissue regions and I'd look for microbiome and host genetics. This is me just doing my little dissections here. And then we also have me doing my mouth swabs. It's really funny because, yeah, you could cut the tentacles off the coral and they weren't bothered. They're like, yeah, whatever, do that all the time. Stick a swab in an animal's mouth? No, they did not like that. They like physically looked like they were having a sulk afterwards. And then they would recover, and I'd put them back on the reef, and I'd go get them next time. So the microbiome results, just showing you some of my science here. The microbiome is essentially all the microbes that live in a certain area. So like humans, we have our gut microbiome and our skin microbiome, and these are all really important for our health. It's been shown that corals have a microbiome and that they also contain microbes that could be really good for their health or really bad for their health. But again, it's like an overall coral microbiome. We don't know where specifically these microbes live, and therefore it's very hard to know their function. So I wanted to look at where my bacteria lived, and I showed here, does the microbiome differ between the tissue regions? And over here on the left, we have the alpha diversity, which is the number of different ASVs or bacterial potential taxa living in that region. So the mucus here has a much higher diversity in the red group, and the ectoderm has a much tighter, smaller community, and then the endoderm is somewhere in the middle. And then we have the beta diversity here on the right, which is really interesting because it's not just the number of species present, but then the abundance of that species. So you could have 5 of this bacteria and 5 of that bacteria and 5 of this one. And this makes up the community composition. And what we can use these plots for is essentially if the groups don't overlap, we know that there's something different. We know that the community composition is different, and then I can look into what is different and what species are different. And again, we see the ectoderm here, this blue one is very tight-knit, very close community. It's not very diverse. There's just like maybe one dominant bacteria. But then the endoderm in gray, it's a lot more diverse as well, with the mucus in red. All these little points are a different sample. And this was really exciting for my PhD. I was like, this is amazing. I'm massively biased, but I was so excited. But I thought, is this just a fluke? Is this a one-time thing? And it wasn't. This is across my 6 sampling points, and we saw the same pattern. We saw the alpha diversity, this much higher diverse mucus, and really like closed off ectoderm. And again, in the plots, we saw that the ectoderm remained quite niche, with the endoderm close by, but with some variation. And the mucus just had a really diverse and really variant community, which is not surprising considering the mucus is outside of the coral and exposed to the seawater. So the main takeaways from that was I saw a significant difference in microbial communities. So the ectoderm was its own community, and then the endoderm and the acrosphere, that nematocyst bundle. I didn't show them in the plots because I only added them in 2017, but they had a very similar microbiome to the endoderm, but this was still very different to the other regions. And then the mucus and gut had very diverse and very different microbiomes to the other regions, but again, are much more diverse because they're these semi-open systems and they have the seawater. Another paper that's coming out of my PhD was when I specifically looked at the bleached microbiome and how the bleached tissue regions differed from the healthy, and they showed a really big difference. And the bleached microbiome is really cool because we saw a lot of microbes that we know are to do with disease and being unhealthy, and the healthy ones showed a lot of microbes that are more related to probiotics and being a healthy animal. So it's really interesting to see, oh, this microbe lives in the endoderm, which is a really important region for bleaching, and it's really helping the coral's health. So maybe if we give this coral more of this microbe, and like, you know, like a probiotic pill or whatever, like a lot of researchers are working on these days, then this could help their health overall and help them survive bleaching. And also just some cool images. So I did fish, not the cool things that swim in the sea, but actually fluorescent in situ hybridization, which is a way of just like validating my sequencing data. Like, I'm not making this up, guys. Like, these are some pictures. So I looked at, so this is just a couple from the ectoderm, and I use these fluorescent probes to tag bacteria. This is a general bacterial probe, but I also use species-specific probes. And then I could find these bacterial aggregates, these big fluffy clusters in the skin, and be like, oh, this is where it lives, and this matches my data. And again, you get this great resolution because here at 100 times, you can actually see like the little fluffy bits that are the different bacteria living all together in the coral. and again shown to be like specific to the ectoderm. And then finally, guys, my genetic results, just another picture of Orpheus, so beautiful. So similar idea here. The main question was, when Heliofungia is bleached, does it have a different genetic response to when it is unbleached? So unhealthy, healthy. And again, going back to that tissue region idea, is I really wanted to know, is the endoderm going to have the biggest like change in gene response because it's got the Symbiodiniaceae and then it doesn't have the Symbiodiniaceae? And that's what we showed, that the endoderm did have the greatest gene response. Long story short, I did some maths, hate maths, but I managed it. And we looked at which regions, so the ectoderm versus the endoderm, comparing the bleached and unbleached groups, like which genes like had the most and the most extreme response, and then which region did that relate to. And what we wanted to see was here, this endoderm group, the main thing to take from this plot is it is a higher value, showing the greater change in gene expression, and it also has a much wider variation. So like 0 would be the normal, genes don't change expression. What we want to see is lots of genes doing a really extreme change in the endoderm. This was also really useful, this interaction value, because when you get gene expression data, you get thousands of genes. Like I'm talking 30,000 genes. And I'm like, well, I don't know which ones are important. But using this sort of maths to this equation to work out which were the most important genes when bleached and unbleached in the different tissue regions helped us identify the key genes related to this loss, this process of bleaching, which came back with 156 significantly differentially expressed genes, which is a lot easier to work on than 30,000. And it was really interesting. The main thing to take from this is just that they had different expression patterns. So we could see the genes that were only expressed in the bleached ectoderm or only in the bleached endoderm, or if they were across them in multiple regions and multiple areas, or if they were really expressed when the coral was healthy. And this helped us understand the function of the genes because we knew their region and when they bleached or when they weren't bleached. So for example, one of the really exciting groups here is the genes that were expressed at a really high level in the bleached endoderm, so that region that contains the Symbiodiniaceae. And what we see, this is just an example of some of the genes, but like I said, like they're just really highly expressed in the bleached endoderm. And these genes are related to things like immune response. So there's a lot of research on immune response and the loss of Symbiodiniaceae, because these guys might remove the symbionts during bleaching. And we saw anti-apoptosis. So apoptosis is quite a sad process that when corals and cells in an animal are too stressed and they can't handle life, they basically give up and they die. And there we saw genes in the endoderm that were really anti-apoptosis. So they were trying to stop the cells from dying, which is massively important because these are the kind of genes that can help us know what, corals that bleach and die, they don't have enough of this gene. And this might be something that can help corals in the future. We also saw the genes that removed harmful products or toxins. So again, that heat stress on that coral, the coral's really stressed. It's like feeling quite toxic. And these genes are the ones that like, okay, let's take this poison like out of your body. Let's get rid of this. Let's make you healthy, trying to survive because Heliofungia is good at surviving bleaching. And finally, the return of homeostasis. So what genes we need to see to get the coral back to functioning, normal functioning, getting its color back, feeling healthy, feeling good and living and continuing to be this biodiverse ecosystem. So yeah, overall guys, my tissue specific analysis can give us a better understanding of coral biology in normal and stress situations. And also it's really important thing that we can localize this, genetic or microbial response, seeing what is happening in what area when it's bleached or healthy. And then just for fun, as I didn't talk about developmental biology, but here is a really cool video of my corals spawning in 2017. So this is a female coral. These are all eggs. My corals have split sexes. And this is about 1 a.m. and all of us very excited to see this happening because it's such a large scale, because it's such a huge mouth and it basically just vomited out all its babies. So yeah, thank you for listening, everyone. I hope you enjoyed that. And yeah, any questions?
Great. Thank you so much, Chloe. That was really fascinating. A lot of really detailed work that you have done on the corals with your PhD. I'm wondering, kind of with all of the work that you've done and the data you've collected, what do we do now? So we've kind of seen kind of their difference in the bleach and the non-bleached corals, and I understand that like in a lab, you can kind of simulate how you get healthy corals and bring these bleached corals to be healthy again. But how would that translate into the ocean?
Oh, that's a lot. There's a lot of ways. I suppose there's also the genetic way and there's the microbiome way. So there's a lot of great research being done that's looking at, these microbes that are really good for coral health, and essentially like looking at if we can then like put this into the water column, more of these healthy microbes, so that when the coral is stressed, that then they sort of have these microbes to fall back on and they can help them survive. And then also a lot done with like breeding, like baby corals being exposed to, again, the microbes. So when they grow up, they've got it in their microbiome, like we do as babies, we get from our mother's milk or whatever to like survive and be hardier when we're older. The genetic front is more interesting, I suppose, in the fact that there's all these things to do with epigenetics and assisted evolution. So again, either the breeding the genes into the corals or even trying to genetically modify the corals. But there's a lot of ethical stuff that comes with that, because obviously, modifying an animal. But it's all about trying to target what the process is that causes the bleaching, so that then we have an idea of how we can stop it. I don't know if I answered your question or if I just went off on that. But yes, implementing it into more corals, using this model organism so we know what happens in the colony corals, that obviously we can't look at that tissue region, and then hopefully be able to understand more about it and see it into the ocean.
Yeah, excellent. Do you see kind of the diversity in the mucus and the ectoderm and the endoderm? Is that in those microbiomes? Is that similar in the species that you studied in other species, or are those totally different amongst coral species, or do we not know?
We don't know. This is the first time it's been done with corals. There's some anemones and Nematocella and Hydra that have looked at particular tissue regions, and giant clams as well have shown. So the ectoderm is basically dominated by one species in Heliofungia, and that's seen similarly in other marine inverts like clams and Hydra and anemones. But for the actual tissue regions in comparison to coral, there isn't a comparison. So, actually, no, there are some comparisons with the coral mucus, because obviously that's the outside stuff so you can study that in corals, and that's really similar to what we see in other corals. But yeah, looking at the guts and the tissue layers themselves hasn't been done before, but it's just very exciting, particularly in the gut, that we had quite a few species that are well known for digestion in fish and reef fish, and so I just got really excited because I was like, oh, it's actually got like a digestive microbiome. But yeah, okay, I'm trying not to be a nerd right now.
No, that's what this is for. This is the perfect platform.
Yeah,
cool things in the gut of corals. So, I mean, it is fascinating.
Oh, thanks. I think so.
Yeah. So the last we've time for one more question, which is what's next for you? What are you working on currently, or what is next that you're studying?
Next, well, at the moment, yeah, I'm working with Emma, and I'm working on quite a few interesting projects with the elementome, which I think she's going to talk to about today, how the elements impacts the coral, its genetics and its microbiome. And then also again, I'm looking at other coral species and their genetic response to bleaching, and also her super corals, so ones that work in the mangroves and the reef. At the moment, I'm just trying to finish up and publish my PhD papers so that I can get some postdoc funding and hopefully start a full-time project. So at the moment, I'm just working on a lot of cool, interesting projects. So yeah.
Great. Well, thank you so much. And thank you for sharing all of your work with us today at the
Thank
Global
you.
BioFest. And I hope you have a wonderful day over in Australia.
Thank you. I will. Thank you so much for having me. This has been fantastic.
Absolutely. We'll see you soon.
See ya.
All right. Well, that was wonderful. I'm going to go ahead now and bring in our next host that is coming on board with us. We have Jessie, who is the VP of Education at Exploring by the Seat of Your Pants Extraordinaire.
I'll be here all weekend. It's so nice to see you, Lizzie, again. It's been since the last BioFest. And it's so nice to have amazing programs to kick off our festival together. Wow. Oh.
Yeah, we've had some wonderful, wonderful presentations, and I'm excited to see our next one. We're headed back to Brazil, I hear.
We are. It's been a very Brazilian day today. I know we've gone from Argentina to Australia. I have my passport. I don't even need it because I get to travel virtually with everyone today, and it's been such a spectacular ride. But a lot of Brazil focus and some great Brazilian friends staying around in our chat today to ask some great questions. So I hope we get an awesome Q&A as we dive in with our next speaker.
Yes, absolutely. Well, I'll let you take it from here, Jessie, and I'll see you tomorrow.
Thank you so much, Lizzie, and have a wonderful evening. As Lizzie said, we are heading back to Brazil in a minute, and so we are going to dive in with our next speaker in just a second, and he really embodies everything that we're trying to emphasize with this Global Biodiversity Festival. We are about 7, 8, 9 broadcasts into over 60 programs over the next 3 days. I hope you guys get the chance to tune in live for the recorded programs after. It's really going to be a special expedition around the globe to all the continents to hear some really amazing and inspiring conservation stories. Now, growing up as a boy, David Attenborough is my hero, and one of my very favorite books of his is Adventures of a Young Naturalist. So in it, he details some of his expeditions, but one that particularly stuck with me was a journey to South America to go find the giant armadillo. For those who haven't seen a giant armadillo, I know we're going to see it in this talk coming up in a minute, but look it up. It's just the most spectacular and unique and incredible species. And today, we are joined by Arnaud Debier. His work has been featured by PBS, BBC, National Geographic, and more. And he brings this global expertise, having worked across South America and in Nepal, and for the last 19 years in Brazil, working to save giant anteaters and armadillos. Today, we're going to focus on the latter. And I'm so excited to bring him in to share his story and hopefully inspire you guys to take some great conservation action wherever you might be joining from around the world. So Arnaud, thank you so much for coming in today, and welcome to the program. Welcome to the BioFest.
Hi, Jesse. Hi. Thank you so much for having me.
Yeah, so great to have you here. I know we were testing screen share in the back. If you want to bring us in and dive into the talk, I'm excited to hear from you, and hopefully we get some great questions right after you're done.
Perfect. So let's dive in.
Fantastic. I love your backdrop, by the way. I'm becoming an aficionado of backgrounds in people's houses, and it's very elegant. I like the dream catchers.
Okay, here we are. So hello, everybody. My name is Arnaud Debier, and I'm the founder of a small Brazilian NGO called the Wild Animal Conservation Institute. And tonight, I will be talking about giant armadillo conservation in Brazil through our Giant Armadillo Conservation Program. Now, I'm originally from France. I've lived half of my life in the United States and France. And I had already been living for 8 years in Brazil, and I had never seen a giant armadillo. And this was a species that I always dreamed about seeing, just kind of like Jesse introduced in the book about David Attenborough. This is a very elusive, rare species that very few people get the chance to see. And so I was determined to try to, just get an image of one. And so so 12 years ago, I set out in the field where I was living in the Pantanal and put some camera traps up. And this is when I got my first image of a giant armadillo. So this was in July 2010. And I got this big male giant armadillo leaving its burrow at the right at the cusp when the night was starting. And this picture really changed my life. I was absolutely shocked to see this species. Just the sheer size of it. Giant armadillos are 5 feet from the tip of their nose to the tip of the tails. They weigh about 80 pounds. And I just couldn't believe that this species existed. And, this picture changed my life. And when I got this picture, I decided to just dedicate myself to understanding and then conserving and trying to save this species. Giant armadillos are distributed throughout South America. They are found from Venezuela to northern Argentina, basically everything east of the Andes. They are the largest of the armadillos. They are, as I said, they can weigh about 80 pounds. They're about the size of a Labrador retriever. They're characterized by this big armor, and these, on the third digit, these really large claws that are bigger than the claws of a polar bear. They can reach over 15 centimeters. And they use these big claws to rip open ants and termite mounds, which is what what they feed on. And they use their back paws to kind of, like us to scoop sand out. So, I started the project now 12 years ago in 2010 from that right image. And now we have programs in different areas of Brazil and even Argentina. We work in the Pantanal, Cerrado, Atlantic Forest, and the Argentinian Chaco. And I'm going to start focusing with our work in the Pantanal because that's really where we started. That's where that first image was taken. But it's also kind of like our laboratory. This is where we learned the basics about giant armadillo biology, ecology, health, genetics, and their interaction with the other animals. For those of you who don't know, the Brazilian Pantanal is located in the in the center of the South American continent. 95% of the Pantanal is privately owned and divided into these really large cattle ranches where traditional grazing is practiced. And this is, the Pantanal is actually one of the few is one of the few examples of the sustainable use of a tropical ecosystem. And until recently, wildlife was pretty well protected there. That's probably due to the extreme droughts, extreme flooding, which means that, besides cattle ranching, not much else can be done. Well, as I mentioned, I started by placing a few camera traps, and that's actually how we made our first discovery, which is that other species use giant armadillo burrows. Here we have a six-banded armadillo using a giant armadillo burrow, a southern tamanduá using a giant armadillo burrow, a naguti munching a seed in a giant armadillo burrow. And what we found is that giant armadillos are ecosystem engineers, and this means that they modify resources in the ecosystem, and the resource here is they provide these homes, these burrows, these deep burrows. They can be up as deep as 5 meters. They have this large mouth, about 45 centimeters, and basically the giant armadillo spends all day deep underground. They go out at night for a few hours, on average 5 hours, to forage on ants and termites, and then often enough they will dig a new burrow. And on average, they dig a new burrow every 2-3 days, and then they abandon these burrows. And these burrows are then used by lots of other species. We've documented over 70 species using giant armadillo burrows. And you can see this little guti is munching a seed. It's 41 degrees Celsius outside, but inside the burrow, it's a cool 25 degrees Celsius. So giant armadillo burrows offer a refuge against temperature. Here you have a crab-eating fox, ocelot, tyra, lots of other species. Collar peccary, it's a refuge against extreme temperatures, but also a refuge against predators. And not only the mammals, but also reptiles, your tegu lizards, tortoise, lizards. And of course, it attracts the predators. So we have all this life happening around the giant armadillo burrow. Here's this red-legged Siriema looking to see if there's not a lizard or a snake that it can feed on. And it's not just the burrow, but also the sand mound, which provides food. There's lots of invertebrates, even amphibians, and animals go there to seek food or as a place to rest. So we were talking about David Attenborough here. We actually had a documentary made in 2017 about hotel armadillo, describing this process, which was narrated by giant armadillos. And so this is one of the fascinating facts about this species. Here's a species we knew almost nothing about, and yet they provide homes for other species. But of course, we had to capture giant armadillos, and up until now, in the past 12 years, we've captured 36 giant armadillos. And we placed telemetry devices on them and have been able to follow them and understand better about their ecology. They have exclusive home ranges that are 25 square kilometers. And just as an example, the largest terrestrial mammal we have here in Brazil, the lowland tapir, has a home range of 5 kilometers. So giant armadillos, 25 square kilometers, 5 times larger than a tapir home range. we've shown that they select forested habitats mostly. And that is especially important for reproduction. Females reproduce and have their pups in the forested areas. And the first few years of the pup's life are spent in the forested areas. So forested areas are key for giant armadillo survival. We also learned about their reproduction. Almost nothing was known about giant armadillo reproduction before this project started. And what we found is that their gestation period is 5 months. They have only one pup at a time with the interbirth rate of 3 years. And giant armadillo mothers are very. But of course, we don't only do science and study the animals. We also try to mitigate threats and try to protect giant armadillos. In the Pantanal, the main threat we are trying to mitigate against is our fires. You might have heard that 2 years ago, in 2020, we had terrible fires throughout the Pantanal, where over a third of the biome was burnt to the ground. And we have documented that in the past 12 years, we have lost 3 giant armadillos due to fires. In the neighboring park in the Cerrado in Emas, 2 giant armadillos were also documented dead. So we built a community fire brigade where we have joined 10 ranches together, fundraised for materials to provide them with all the materials they need so that they can combat fires themselves. And so here we are with the fire brigade. This fire brigade was trained last year. In July this year, we're training another more. We're giving the opportunity to people who are already trained last year to be trained again, but we're also branching out, inviting more neighbors. And so we're really protecting a huge area, over 1,000 to 1,500 kilometers with these community fire brigades to prevent fires. And that's really important. In the Cerrado, the main threat we're focusing on is both habitat loss and a human-wildlife conflict. Habitat loss, we started since 2015 trying to model habitat distribution of giant armadillos. And you can see all these little dots. These are the places where we visited micro watersheds, and the red ones is where giant armadillos were not present. The blue ones is where giant armadillos were present. And we use that to try to model the probability of occurrence of giant armadillos. And you can see that here, in dark brown, that's where giant armadillos can occur. And what is terrible is these results showed us that, and you can see here in green, fragments where giant armadillos, there's adequate habitat for giant armadillos. And what these results showed us are terrible. What they showed us is that the habitat is completely fragmented and isolated. And here we only have 69 fragments of more than 25 square kilometers that are adequate for giant armadillos in the Cerrado of Mato Grosso do Sul. Only one protected area that is more than 100 square kilometers, and only 4 fragments that are more than 100 square kilometers. And when I said 25 square kilometers, that's because that's the average home range of a giant armadillo. So only 69 fragments, more than 25 square kilometers of suitable habitat for giant armadillos in the Cerrado of Mato Grosso do Sul. And so the project we're trying to focus on now, and we just started this year, is creating corridors, working with local people to try to create private corridors to try to link these different islands of native habitat which support giant armadillos. Another threat that we're trying to work on is human-wildlife conflict. You see beekeepers, they place their hives near the fragments where, near these native fragments, right? They try to get the bees to use the flowers of the native Cerrado. Well, that's also where the giant armadillos live. And now the giant armadillos have learned to destroy beehives. And you can see in this video, here's a male giant armadillo, very interested in toppling the beehive. And what it will do is it will topple the beehive, rip it open, and eat the larvae. And so what we have created is now, we've created a manual guidelines to coexist with giant armadillos. And so there are lots of different methodologies that can be used to prevent these attacks. And the beekeepers that prevent the attacks and use that, they will gain a certification of giant armadillo friendly. And so you can see here's our first honey. You can see it has the certificate on it. And I have several jars here. You can see here is giant armadillo friendly honey. You can see we now have this for sale. There are about 200 beekeepers that live in the giant armadillo distribution area in the Cerrado Mato Grosso do Sul. We just started now certifying beekeepers. We already have 50 certified beekeepers, many more interested. And what we're excited is that this honey is now being purchased more expensive. So it has higher value. Giant armadillo honey, friendly honey has more value. So that's a really exciting project that's being developed to try to save the last giant armadillos in the Cerrado and help beekeepers to coexist with them. In the Atlantic forest, the Atlantic forest is one of the most threatened biomes in South America. Less than 10% of the biome is left. We've surveyed several areas of the Atlantic forest, and we have found that only in the Parque Estadual do Rio Doce in Minas Gerais there are still giant armadillos, a viable population of giant armadillos left. And so we have now been working since 2020 in this park to try to save the last population of giant armadillos of the Atlantic forest. And this is a very critical situation. We believe there are at least 60 individuals left. We have a huge camera trap grid. Here's Lucas Barreto, who is coordinating the work there. We have several camera trap grids monitoring the population. We've been able to estimate the population. And now we're working the surrounding fragments to try to reconnect them with the park. And we're working hand in hand with the park to make giant armadillos, one of the ambassadors of the park, so that the park is not only preserved, but expanded. And that the different threats, we have several threats of encroachment. They want to open a road in the park and everything. So we're now working with other species conservation projects to try to prevent that from happening. So save this important habitat and save this last population of giant armadillos. Education outreach is something that is key to our work, and we collaborate with the FUBA, which is environmental education company. Andrea is the one who coordinates this work, and we have a specific action plan for education and communication. We work in over 200 schools in Mato Grosso do Sul. We work with lots of different types of local community. From as you can see, the Cerrado has a huge diversity. We work with lots of different communities, and of course, our state government. And here we are with the governor of our state discussing giant armadillo conservation. All our education, due to the pandemic, we've had to now bring all our education work online. And so if you go onto our website, you can find more about our education work. And last but not least, I want to just introduce the team of ICAS, the Wild Animal Conservation Institute. We have 16 full-time staff, and everybody works extremely hard. And it's a very diverse group of biologists, veterinarians, social scientists, public relations communicators, educators, public policy specialists. And so this diverse group forms our NGO. And I want to point out that 9 members of our staff are now sponsored by zoos, and that is really key. I myself am sponsored by the Royal Zoological Society of Scotland, which owns the Edinburgh Zoo and Highland Wildlife Park. And so zoos are a huge part are a huge collaborative and important part of our work. And last but not least, I cannot help but mention that Gabriel Massucato, which is who coordinates the work in the Pantanal and is one of our biologists. He just won the Future for Nature Award just 2 weeks ago. And he was in Holland receiving this wonderful award, and he's already back now into the field. And here, we have our full-time staff, but we also have volunteers, students, and lots of different collaborators, 10 students, and about 40 different collaborators and institutions that we work with. And here are the donors that work, lots of different zoos, especially from North America and Europe, reminding you that there are no giant armadillos in captivity in North America or Europe, and yet these zoos fund our project, and we're very, very, very grateful to them. So, this has been the last 12 years of my life have been dedicated to giant armadillo conservation. It all started with this one picture. It's been a crazy adventure. It has taken me through different biomes of Brazil, in all kinds of situations. And this is just an amazing species that, even today, when I see a giant armadillo, which is rare, which when we capture them, it's still such a huge, impactful event. These are dinosaur-like creatures. They're huge. And even though they look so big and strong, they're actually such a fragile species that really need our protection. Remember, this is an animal that has a low population growth rate. We discovered, I forgot to mention, that they reach sexual maturity only when they're 7 to 9 years of age. Then they only have one pup every 3 years. And just if you look at compared to the 9-banded armadillo, which is the armadillo most people are familiar with, they have 4 pups every year. So in 3 years, the giant armadillo has 1 pup. The 9-banded armadillo has 12. The nine banded reaches sexual maturity at 1.5, 2 years. This species 7 to 9 years. So these are really fragile species that, habitat loss, fires, roadkill, any removal of any individual has a huge impact on this species and can lead to local extinctions, because this is a species that occurs at low density, that is extremely rare, that spends all day underground and only comes out at night. People don't even know it exists. And when it goes extinct locally, people don't even know it goes locally extinct. And that's what I find really heartbreaking with this species. It can disappear without anybody noticing. So hopefully we are trying, my team and I are working as hard as we can to give a future to this important species, which also gives homes to other animals, and it plays a key role in the ecosystem. So thank you so much. And if you are interested in learning more about our work, you can please get into our social media. We have, or get directly in touch with me. I left my email right here. So Instagram, Facebook, Twitter, we're LinkedIn, YouTube. We have media and all of those. Thank you so much.
Arnaud, what a spectacular presentation. And I think this is a theme that we're going to keep coming back to throughout the entire Biodiversity Festival, is that a lot of these charismatic and really amazing animals are facing a wide array of threats. It's not usually just one thing, it's a lot of different things that impact them. But I go back to the beginning of your talk and your sheer joy and passion seeing the baby, which I've never seen a baby in any footage I've ever seen in my life. What a spectacular creature. And I think that's a testament to you and has led to this amazing recognition with the team around you and the amount of support that you've gotten from zoos around the world. So kudos to you for all of that. It's really quite wonderful and well deserved. You talked about building corridors and working with local community stakeholders to sort of make sure that there's passages that these amazing creatures can go through. Now, corridor development is a really popular conservation idea around the world. How are you finding that? Are people receptive to that idea? Do they leave the land? Do you have to fight with people to sort of convince them of the worth of this, or how's it going?
This is all volunteer, and so this is kind of the work is starting this year. We're working with different NGOs, and especially a new cellulose company who has made it its mission to reforest several areas. So thanks to this, this private company, it's really helping us. And so it's a question of finding the maps, linking the right things, and we're doing all the monitoring and try to show them what are the best places to do that.
Yeah. I'm really happy that you mentioned that. We talk a lot about government support for a lot of programs around the world, and even things like zoos where you sort of expect that their basis is education and conservation. But when private industry gets involved in a serious way, that's a really spectacular thing. And I think it can lead to a lot of great funding for programs like this. So kudos to you. With the bees again, another thing that we've seen with elephant conservation, we've seen around the world. Yes, I love your honey. I made you full screen when you were sharing that, so we'll have to make sure people have that link if they're keen to know. Here, I'll make you. We can see it a little bit closer for everybody. My toast is not the same without honey, and nor is my cereal, so I'll have to buy from you guys from now on. Now, you talked about these armadillo-proof things. There was a sort of a mock-up of it. How do you make something proof to a powerful creature with gigantic claws? What does that look like?
So one of the best solutions to prevent giant armadillo predation is to raise the hives up 1.30 meters. Okay. That means, because as you saw in the footage, that what they often do is they stand on their hind feet, they use their tail as a tripod, and they'll knock over the hive with their head. And so if the hives are higher up, that prevents them from doing that. However, then it's much harder for the beekeeper to work the hive. So if he has permanent hives, he can use fencing, both electric or just wire mesh. But sometimes it's important to dig underneath, so to prevent them from digging. And right now we're testing this new methodology where you actually have the hives up exactly 70 centimeters, which means it's a little bit high. It's too high for the animal to push it over, but it's too low for him to raise on its hind legs. And so that's something we're testing.
Wow.
And so we're looking at these different methodologies because giant armadillo do cause a lot of damage in our state. And so but in general, beekeepers are really great stakeholders in the sense that they are people that are usually love nature. They get their honey from nature, and so they're very sensitive to that cause, and so that has been working really well. And the good thing is now we're able to purchase this. This honey is now worth more. The factory that purchased the giant armadillo funding that has a certificate is paying a little bit above market price, and so that's also another incentive. So we worked with a North American NGO called the Wildlife. What is it? It's Wildlife Friendly Network Enterprises. They helped us to develop the norms. We have a contract, a 3-year contract with the beekeepers, and includes not just giant armadillo friendly, but practice no deforestation, careful fire management, no child labor. It's all these other things, so that it's really to international standards.
Yeah. Well, I love this because, I mean, what I find in a lot of the education work we do with conservation groups is that people want to be able to buy products that help support local wildlife. It's very difficult to do that research on their own. And so any organization that partners with a group that can vet that and make sure that it's at the highest standard for people, for wildlife, for the habitat, it really goes a long way to helping people make those informed decisions. So what a spectacular program. For you've been active in media, you have this team, you partner with zoos, you get this education word out there. How effective do you find that in terms of supporting your efforts? Not just, you go to these community stakeholders, you talk to them, you engage them in the work that you're doing. When some of your work gets featured on PBS or BBC or something like Hotel Armadillo, do you see that feedback into your work? Do you see more interest, more people reaching out? How does that play out?
Well, it works well when it's local media. That works really well. The BBC and PBS was not very helpful to us because when you work with these broadcasters, for it to be broadcasted in Brazil, Brazilian television would have to purchase the rights, and so it was never broadcasted in Brazil. So, which is something we didn't understand. We were really excited when the BBC and PBS wanted to film our work. Yeah. We had no idea that wouldn't help our cause here in Brazil. That's been really disappointing. Yeah. Meeting David Attenborough and having him, talk about our project, that was the highlight, of course.
Yeah. I'm happy for you and jealous at the same time. It's a spectacular opportunity. But I did bring up, for anyone who does want to check out the PBS Series 2, Hotel Armadillo, looks fantastic. I had this up and it's a gorgeous trailer if people want to check it out online. And you had the most intensive of all our final slides for any of our speakers so far with all these ways people can get in touch. But iGuessConservation.org.br is a really fantastic tool. GiantArmadillo.org. You have all the sites. You have more sites and ways of contacting you than I think any 10 people. So I hope our viewers today, live and after the fact, take the opportunity to do so. Arnaud, I'm inspired. I'm so excited. I've been waiting for this program ever since I heard I was getting the chance to host it. So thank you for sharing your passion.
Thank you so much, Jesse. Thank you so much for having me, and have a wonderful evening.
Thank you so much.
Such a fun night. I'm loving all the talks. I'm really enjoying watching all of them. It's
Stick around. We've got many more to come. I think we got like 7 more before we wrap up the night.
I look forward to seeing and watching them. This is great. Thank you so much.
Have a wonderful evening. Thank you again. And we look forward. Hopefully our audience takes a chance to learn more about you and your work. Again, iGuessConservation.org.br. If you guys want to check that out, it is your one-stop shop for their education work, their conservation work, and so much more. Now, right now, to keep the fun rolling, we are bringing in one of my favorite speakers, a person I've had the pleasure of hosting, I think, 5-6 times now, more than almost any other individual conservationist in the world, and that is Rosa Mia Gayen. So I don't want to steal her thunder because her personal story in leading to her role in protecting a really amazing species in the cotton-top tamarin is incredible. So we're going to dive in with her. We're going to head from Brazil up to the northern coast in Colombia to learn a little bit about her work protecting these really charismatic and beautiful and delightful monkeys. So Rosa Mia, thank you so much for joining us this evening, and welcome in. Nice to see you again.
Hi, Jesse. Good to see you. Amazing to be here with you again and sharing with everybody who's connecting today and through the weekend to this amazing event. I think it's the 3rd year, right?
3rd year, and I think by the time we'll finish this one, it'll be over like 250 total events with conservationists around the globe. So it's like we just don't sleep. We have a great time. We have an audience from around the world, speakers from around the world. It's so much fun.
You're amazing, guys. Thank you for putting this together and giving all of us the opportunity to share our stories. And I enjoyed very much seeing Arnaud. It has been a while since we met, and admire his work as well.
Hey, networking is half our fun. After the talk, we can get you guys all together. We'll have a virtual get-together. But please do. We'd love to hear about your work with the Tamarins. And so if you want to dive in with your presentation.
I will. And let me get this rolling. Yes. So
Look at that.
This is our beloved little cotton tops. They look big on this screen probably, but they're just about the size of a squirrel. And they live in these amazing forests in northern Colombia, the tropical dry forest. It's called the tropical dry, as opposed to a tropical rainforest, because it's got, 6 months out of the year is very dry and then lots of rain. And that's where our beautiful, crazy hair monkey lives. Even though he's just 1 pound monkey, size of a squirrel, it's very similar to us humans. They live in family groups like we do. They're very, very territorial, just as we are humans, right? Juvenile cotton tops leave their home, or sometimes they get kicked out of the house, just like we do, to go and find our own territories and make our own families. They have a very sophisticated vocalization system, just like a language for us humans. And they feed from fruits, and you just saw one of those eating a little insect there, and nectar and sap from trees. And they have an amazing role in the forest where they live, and it is that they're seed dispersers. They eat so many different fruits that grow in the trees in the forest. And then when they move around their territory and they're pooping, all of these seeds are coming out down to the ground and, becoming new trees. So, aside from the size difference, they're very much like humans, it's primates after all, right? But I want you to take with you 2 main facts about cotton top tamarins today. One, the fact that they're only found in that little red dot in our huge planet. Is the country of Colombia, and not even in the whole country, just on that little black stripe of land that you see in the northwest region of our country in Colombia. That's it. You don't find them anywhere else in the wild, in Colombia or in anywhere in the world. So they're very unique and very special little monkeys. And the other fact is that, unfortunately, like many other wildlife species that you'll see about or you hear about during the festival this weekend, they're highly threatened. And it's mostly, in the case of cotton tops, it's mostly habitat loss. And that habitat loss happens for development, cattle ranching, agriculture, large scale, but also for domestic uses like selective logging or firewood, fences, construction. So usually farmers in local communities that are facing poverty, they go into the forest and cut trees. And they also hunt cotton tops for the pet trade, which is something that really, really breaks our heart. That is why cotton tops are considered a critically endangered species. We estimated that there's about 7,000 left in the wild, and the number doesn't say that much. It may seem a lot, but it's only in less than 8% of the forests that used to be in that little black stripe in northern Colombia. And not even all of that 8% is good for cotton tops, because you need to have the trees that provide food and the trees that provide shelter, and not all of the forests that are left have that. And they're very fragmented as well. So I am the director of Proyecto Titi, which is a nonprofit organization based here in Colombia. It was founded by Dr. Ann Savage about 30 years ago. And we have 4 pillars of our work that support our mission to guarantee a long-term future for cotton top tamarins. Just avoid extinction, give them a chance. And we do this by, conducting field research, which is actually the beginnings of Proyecto Titi were as researchers, but then given a lot of emphasis to forest conservation, of course, because it's the main threat. And that's what I'm going to focus most of my presentation today, but also with a big social component on environmental education and providing people in local communities with alternatives to generate an income and try or reduce that dependence on cutting trees and hunting animals for subsistence. So We work in 3 different areas in Northern Colombia within that black stripe. And you see all those green dots that in this map, this is what's left of the forest that is home to cotton-top tamarins. So we work around protected areas in those blue circles just to keep or protect whatever's left and try to increase that forest size and connect it as well, and work with communities around it as the focus of our conservation work. Our research is done with telemetry. So we put a little backpack, like this one you see in the picture, to the dominant male of each family group. So the daddy carries the backpack, and that sends a signal that helps us find the monkeys in the forest. Because we're talking about a 1-pound monkey that hangs out 30 to 40 feet above the ground, and it's very elusive, doesn't like humans that much. So it helps us find them and study them. Our team, biologists and assistants, go in, pick up the signal from the animal, and take notes about everything they do. Just as I summarized in a minute, we have learned all of that about cotton tops by observing them day after day for many years now. So all of that information is so valuable, not only for sharing on publications and scientific journals, but for using it for protecting the animal. And that is one of the things we're doing as we move forward. Right now, we are conducting the 3rd population census that we have done. The last one was 10 years ago. And we're going to confirm that 7,000 number and that 8% of forest through this work that is very intensive. We'll have some updated results after 10 years of working very hard to protect the species. And we're hoping that at least the populations are stable and, hopefully, or we're dreaming that it increases, but we'll see. That's part of the research work applied to conservation. And we have used all of that information in protecting forests, and which is the focus of my presentation today, being the main threat for cotton tops. And we are doing or have done this, using 3 different strategies. One is working with environmental authorities to create protected areas, public protected areas. Working with them to date, over the last 10 years, we've been able to secure about 5,400 hectares in 4 public protected areas that probably protect just about 5% of what's left of cotton tops in this region, which is a significant number when you don't have that much left for the species and when deforestation is your main threat. But we're also using a second strategy, which is purchasing land. And we've been very successful over the last 3 years. We started with 70 hectares, and 3 years after, we have almost 500 hectares. Hopefully this year we will get there, of protecting forests for cotton-top tamarins and, of course, for all wildlife, but forest that helps connect protected areas and increase the habitat size and restore as well, because some of these properties were used for cattle ranching and agriculture. And then our third strategy is connectivity, because cotton tops always hang up in the trees and they never come down to the ground. So when they have this kind of landscape, then they get confined into islands, and that can certainly hurt their long-term viability, their genetic viability. So that work is what I really, really love the most, because it gives us a chance to work with local communities in creating these forest corridors, like green highways, for the cotton tops to move around and find resources and be able to mix up genetically, if so to speak. And we would love to buy much more land to protect, but it's unrealistic. So we work with farmers and create conservation agreements in partnership with other organizations in the area. And, we tell them, okay, what if we set aside the land within those yellow lines and we plant trees in it to create forests? And in turn, you get support for increasing productivity on your land using environmentally friendly practices, of course. And we provide seeds, we provide poultry, we provide wood trees, fruit trees, honey production as well. And that way they get benefits and we get benefits. And we have created today this network that protects about 1,000 hectares of forest corridors. You can see our reserve on green in this map and how it's shaping up to connect the National Park Los Colorados to many other corridors and protected areas in the region. So we're creating basically a spider web of forest corridors that help monkeys have opportunities to move around and find resources and grow, of course, and grow in populations. And we do this by, all of this forest restoration work, this corridor restoration work is done by our team on the ground while they patrol our reserve. They collect seeds from the trees that we propagate in our nursery. And it's mostly trees that provide food and shelter for cottontops. So all of that scientific information we have picked up over the years is being used to restore the forest and to create forest that is good and viable for cottontop tamarins. And then we work with the farmers to plant those trees in these areas. Some of these areas are not easy to get to, so we have to, use their burros. We hire them to help us in this labor, and also, digging the holes and planting the trees and helping us, bring this forest back in areas that previously were used for cattle ranching and agriculture. This is very exciting for us because all of these people that you see in this picture are part of our staff, and they're all hired from the local community. So what a better way to engage your community than providing jobs. We wish we could do much more, but these are our permanent staff, but we hire farmers that are participating in the conservation agreements, and they help us plant those trees, and they help us monitor, they help us transport, they help us, propagate, so it's a network of people in local communities that are helping us put this restoration work together. And we have found that it's helping their economies, helping their families, and they're getting engaged really emotionally and, and also professionally with cottontops and all of the efforts to restore the forest. And you would think that that's where it ends, but then we have to monitor these saplings as they grow to make sure that they are surviving and they are being healthy. So we monitor about 30% of the saplings we plant, and we go back every 3 months for the first year, and then every 6 months, and then once a year up to 5 years. And you can see our colleague Aldair there with the machete, kind of checking out the height of one tree that grows really fast. It's a ceiba tree. After a year, it was three times the size, and that's what it does. The tropics, the heat and the humidity. That's a good thing. It makes your it makes your work in the field really hard and tough with the humidity and the mud and everything, but everything grows really fast, which is very good. And this is what we want. We want cottontops to be able to move around, to be connected, in this network, and be able to find the resources they need to survive. And even though we talk about cottontops all the time, we know that all of these efforts to protect the forest for these little monkeys, it's protecting it for many, many other wildlife species of the Colombian biodiversity. So many amazing species that share a home with cottontop tamarins. So while this forest works not only for cottontops, but for every species that shares a home here, and also for people, because we do benefit from ecosystem services. We benefit from the water that the forests protect. We benefit from the wood and from food and resources if we use them sustainably. And that is where our forest conservation work and our research work connect with communities. And just as we involve farmers in our forest restoration work, we work with local communities and the ladies that live in there. They're family members of the farmers and the kids. And we have other projects just as this one, the eco mochilas we call them, but they're made with, crocheted with recycled plastic bag. Really amazing work that they can do in their homes while they care for their families, and a work that has been recognized internationally. And now it's in the runway of some of the fashion designers here in Colombia that are putting them on their shelves. And that provides an income to these ladies and their families, and also connects them with cottontop tamarin conservation. And look at these beautiful little guys. These are the group of women in local communities, make them all sizes. Actually, if you would love to support these smiling women, you can go into our website and purchase online all of these products. It really helps them make it through. We're now resuming our ecotourism activity where you can come to Colombia, actually check it out on our website, and you can come and visit the cotton tops, see the cotton tops, visit the forest, interact with the farmers, interact with the artisans, and really have a great day in the field. And that also connects to the school programs that we develop to engage kids with cotton top tamarin conservation. They go through a semester program. They go to the forest and see the monkeys. Well, this was pre-COVID. We're hoping, keeping our fingers crossed, that we can resume this year, these field visits, have a lot of fun in the forest, connect with the monkeys, and then do something about it. With everything you learn, do something about it. Share it, share with your family, with your friends, and just make it fun for the kids and for the communities to understand the importance of protecting cotton tops, how that benefits us, and how can I help. And, my regards to my team here in Colombia, very passionate individuals working in all of these programs to secure a long-term future for the very cute cotton top tamarins, the cutest monkey in the world. Thank you so much.
I mean, you're hopelessly biased with cutest monkey in the world, but I think that you have a good case to make there.
I do, right? I know. Look at this guy. How can you not fall in love with this guy?
Yeah, I know. You sell me every year. Every year I go in all jaded, being like, no, I think I can make it. Anyway, what a spectacular program. And again, just like Arnaud and for all our programs, the amazing team behind you, the enthusiasm of local community, it's just such a special thing to engage people that way. Community conservation has become a buzzword of sorts, but it's so exciting to see when people really get people of all kinds, all ages, volunteering their time, their effort, their passion. And so, again, such a huge kudos to you for all your incredible work. Yes. With the ecotourism coming back, so ecotourism has, again, become a really big way of getting people interested and engaged in seeing these animals in the wild. Is there an impact on the monkeys? With the research work that you do, with more people coming into the forest to see them, are they acclimatized to people? Do you see them sort of going away? Do you keep your distance? I know this is a big thing with gorilla conservation in Central Africa. How do you find that?
No, I think we are trying to do this the best way possible. So there's a lot of control in the sizes of the groups and the frequencies of the visits and, the protocols, just to make sure we don't create any disturbance. We usually bring people or our visitors to groups that we have been studying for a while. So they're somehow habituated to our presence. So coming in would not generate, a big stress or any major disturbance. They're used to us coming after them, following them for a while, taking notes on what they're doing. And some of them are more comfortable than others. We still study animals that keep their distance, and then they kind of curiously look at us when we're taking notes in the field. And then, it makes our work harder, but it gives us an opportunity to guarantee that you can come and see the monkeys with the telemetry we use. When we are walking into the forest and run into a group that is not habituated, then it's very hard to see them. I mean, you can walk for hours and not see a monkey. So yes, so the fact that we have this telemetry system that we work with allows us to show our visitors the monkeys, and also to do our day-to-day, because this is a day-to-day thing. So these groups are visited daily. Basically the forest is the team's office every day, with mosquitoes and all included. And that's what they do. Just make sure the animals are okay, monitor them, document everything, because it's very valuable information for us. It's been amazing to have so much data, and I think it's every biologist's dream to have so much data to process and understand for these animals, and use it for protecting them and securing a long-term future for them.
Yeah, I love that answer. And honestly, I think it's such a special opportunity for anyone who's tuning into this broadcast, wherever you are in the world. You've had the chance to interact and see one of these creatures that we're featuring in the Global Biodiversity Festival in person. It changes your life, whether that's a monkey or an elephant or a manatee or a stingray. I mean, it's just so special to see something live that is different than anything you can experience, even virtually. And I love the opportunity to share this virtually, but I hope our audience gets the chance to come to Colombia and see the tamarins in person. What a special thing. Natasha has been our go-to all-star audience member. She's been here since the beginning of the festival today with Bobby to kick us off a few hours ago with Joe. And so she wants to know a technical question about the monkeys. What's the difference between the dry forest cotton-top tamarins and the ones endemic to the rainforest like the golden mantle tamarin? Are there physiological differences, anything behavioral, anything you can share with us?
Yeah, some of them have the same genus, so they're just different species. There is, I believe, if I'm not wrong, it's about 16 different species of tamarins or small monkeys in South America. Colombia, there's at least 8 different species. And this one that we work with, which is the cotton-top tamarins, are endemic from Colombia, from the north region of Colombia. And there's another one endemic that lives in the center of the country. And basically, it's their adaptation to the local weather. So cotton-top tamarins go through a long span of not rain and very scarce food in the forest, because if there's no rain, most of the trees drop their leaves. There's nothing to eat fruit-wise until the first rains in the year start coming and the trees start blooming again. So they are smart. So they learn to eat insects, and they feed from insects during the dry season, and also from sap from the trees. And that gives them a lot of energy and a lot of calories, so they can kind of get by. Usually animals in the tropical rainforest don't go through that. So they have, kind of a different diet and different adaptations to their environment. But that would be the difference. In one of our locations is six months out of the year with no rain, and 6 months with a lot of rain. In some other locations in our work sites, it's every 3 months. You get 3 months of rain, 3 months of non-rain, and animals adapt very well to that, especially for their diet.
Yeah. Thanks, Rosemira. Speaking of adapting, one of the pictures you showed very early in your presentation was this little monkey with a big backpack. And we've done programs for kids before, and I'm curious, does that impact them as they're moving through the forest? Do you see a monkey go for a leap and he's just too heavy and he goes a bit lower, or what happens?
No, actually that is if the weight of that transmitter, it looks heavy, but it's not. It's just 15 grams. It's very, very light. And actually this system was designed based on their biology, because they are used to carrying the babies on the back, like you saw in the first image of the male carrying 2 babies in the back. They do for more than 10 weeks carry the babies in the back, and so they're used to having some weight on their back. And 30 years that we have been studying cotton-top tamarins in the wild, we have never seen any impact or any aspect that affects their behavior, their moving through the forest, their interactions with other species. So it's been an amazing tool for us to be able to study the animals, learn from them, and use all of that information to protect the species. So there's no evidence that, or any documentation that it creates any issues for us.
Well, I love that you sort of took inspiration from their ecology with the babies
Right.
in the back. We have, again, Natasha's been mentioning the design background and art. It really helps a lot when you're planning these things and how to interact with animals, how to put things on them. We've had Critter Cam, of course, is something that we've seen a lot in our programs. And there's a lot of thought that goes into that before you do it with a particular species. So
yeah, I think that's a challenge for every biologist. Just try to track the animals with no, making sure that their welfare is above everything. And that's, we certainly played that role. Yeah.
Outstanding. Now, I love this question again, Natasha. If anyone else in the chat wants to share questions, please feel free. Don't be shy. But Natasha's killing it. So I always love, I'm an educator by background, so any question related to education I think is really important. You spend so much of your talk at the end emphasizing that. So more about the way you communicate these species to children. When you're talking to classrooms, you have puppets, you have stuffies, you have coloring activities. How do you plan that? What's the response you get from kids? Anything you can share would be great.
Yeah, we have learned to communicate with different audiences, trying to have a relevant speech to them. So something that they care about. So when we talk with farmers, we talk about water conservation for their cattle. We talk about food production sustainably for their families. When we talk with kids, it's all about fun. So it needs to be storytelling. You know, we use a lot of storytelling. We use a lot of role play. We use a lot of games. And the puppets are one of the most successful and popular strategy we have used, especially with kids between probably 5 to 10 years, so elementary school mostly. And we now, because of the COVID, we had to adjust, like Arnaud was telling us, we all had to adjust to something. So we created this traveling puppet show that has become very, very popular. So we usually focus on a couple of things. The fact that cotton tops are only found in Colombia, so we go straight to the heart and say, hey, we need to be proud that we have this animal. We are the only ones in the world that have this animal here, right? And that is like, wow, really? Oh, that means I'm special and they're special. Yes, they are special. And then, you know, it's just connecting the culture and the arts and the dancing and the painting. That has been the best tools that we have found to be very effective with kids. And we are evaluating pre and post all of our education programs to have evidence that is working. So they're retaining information. They are committing to their age level, of course, to not having cotton tops as pests, which is our main message for all of our education programs, and to become active, to share the information, to share with their friends, and to be just, you know, cotton top friendly communities. That's our goal. And, you know, we're making a huge effort in evaluating everything because it's we need to make sure that it's working. And thankfully, you know, we are seeing good evidence that it is.
You really do epitomize this use of research to prove the stuff that you're doing is effective in everything that you do. And I think it's so important. We're seeing this more and more from conservationists around the globe. It's one thing to dive in and have a passion and start working towards the conservation of a species. But once you have that data, you get funding, you get support, you get people that want to work with you. And I think that's such an important element to this. Rosamira, every time I chat with you, my face hurts from smiling, which is a good problem to have in conservation. I think everyone leaves our broadcasts like that. But I just want to thank you so much for sharing everything today. Again, this is going to live online forever, and your website is just a spectacular tool to learn about that education work, the conservation work, so much more. You're active in all sorts of areas, just like Arnaud was before you. So I really appreciate that, and I hope all our audience.
Thank you, Jesse. And our social media is at Proyecto Titi. Very easy to find. You can contact us through there, and you know we'll be happy to share more information if any of you wants to know more about evaluation or education programs and research for us. And, you know, hope we've motivated you to support our work. Thank you, Jesse, and thank you, Joe, and have a great evening and a great weekend.
Thank you so much, Rosamira. I hope you can tune in for more. I think we've got some more Colombian broadcasting soon.
Of course I will.
Have a wonderful day, and
you too.
Guys, time flies and you're having fun. We're nearing the end of the evening. We're going into our final stretch of a few amazing speakers. And before we dive in with my last person I'll be hosting for the night, I do want to give you guys a little bit of a spoiler for some things that are happening in the days to come. So I'm going to play a little clip to get you guys excited about the days to come. Check this out, and then we'll dive in with some epic wildlife rehabilitation and reintroduction work. For those people who are keen, that is our kickoff to Sunday's presentations at 8 a.m. Eastern. So if you want to check out Stella and her amazing work with whale sharks, I really encourage you to tune in. I must say, I get asked a lot in my job, what's my favorite animal? Now, certainly our speakers do in all the programs that we run. And so it's really tough when you're a biologist, when you love wildlife, when you love wild creatures. They're all pretty spectacular, but I know I've never seen one in the wild. But if I had to pick, whale sharks are pretty much at the top of that list. They're just such a special creature. Now, we have dove in over the last hour with giant armadillos, one of the coolest, freakiest, strangest creatures, ecosystem engineers in Brazil. We just wrapped up with Rosemary Gayen talking about cotton-top tamarins, and I am so excited to wrap up my session today for our Global BioFest Day 1 with Yvonne Kassane. Now, she is a wildlife veterinarian who has worked over 20 years in translocation, reintroduction, and conservation programs around the globe. And so I don't know if there's any more incredible moment in conservation than that moment when you re-wild or reintroduce a species into an area. We featured bison programs on this. We featured a wide array of species on this. And seeing, I mean, the looks on the people's faces, certainly excited about the culmination in many cases of years of effort to bring these species back to wild, but even the animals' faces. I know bison don't have the most expressive faces in the world, but I can tell you in the programs you've done in the past, they look like they're like happy as could possibly be, smiling ear to ear to get back out into the wild. And so I'm excited to dive in with Yvonne today to tell us a little bit about her amazing work, introducing a wide variety of species, and especially some predatory ones and some of the conservation work associated with that, because it can be a little more complicated when you have things that can have a real impact on people's livelihoods in the area. So Yvonne, thank you so much for joining us today. So nice to have you here with us.
Hi, thank you. Thank you, Jesse. I'm really glad to be here.
I'm going to turn it over to you. I don't want to steal your thunder. I want to hear all about this amazing work you've been doing and dive in with a great Q&A after. So if you want to bring up your screen share, let's head into it.
Okay, here it goes.
Here it goes.
Let's go. Yeah, here we are. So
it wants to work.
Okay, how about that? That is working?
That is working. That looks beautiful.
Oh, perfect. Okay, perfect. Yeah, I know. It did that the last time, but
yeah, some people's computers like to do that, and it freaks out a little bit.
Yeah, where did it go? Okay, here we are.
If you want, instead of pressing hide, you can just drag that banner right to the side of the screen.
Yeah, probably I'll do that. Okay, perfect. Yeah, that's perfect. Okay, so sorry. Hang on a second. Sorry about that.
Sorry, take your time. We have time for time.
Sorry. Okay, here we go. Okay, so yes, one of the things that I've done with really nice success, and it's not only me, it's about lots of people involved in these programs of reintroduction. So I want to share with you about the bighorn sheep and pronghorn reintroduction, and also a little bit of the jaguar conservation things that we have done in Mexico. So let me start with just a little bit of the background historic about bighorn sheep in Mexico. Well, they used to be in all the north from Mexico. And if you can see the difference between over here, this is the actual distribution of the bighorn sheep. And this was how it was historically. So really, it got extirpated from Coahuila and from Chihuahua and Coahuila. It got extirpated by the 1950s and 1960s. So it's not really that long ago, but we lost all population over there. So these ones over here are the ones that we reintroduced. So this is how we did it. So well, first thing, so if all those populations disappeared over there, what we then worked on was trying to get some of the animals that were in the populations that were still healthy to bring them to this land that was lost from the bighorn sheep. So we use these kind of nets that are drop nets, and they use some baits to attract the animals over there. These can be either wild animals, or in maybe some really, really huge areas that are fenced, but anyway, they don't have contact with humans. So we use these, and well, the net obviously drops on top of the bighorn sheep, and then we work over there. You have to have a lot of people to be working with each one of the animals, so they don't get stressed under the net until someone comes and lifts the net, and then we work on other kind of issues for their health, like checking them if they are okay. We use sometimes some vaccinations or deworming or other things that we need to make them feel okay, or that we can check that his health is going to be okay. So that's what we do. But we can also use net guns that are used by helicopters. So they chase the animal, and then they net gun the animal, and then they put them like that, 3 or 4, and then they put them on top of the helicopter, and that's how it looks. They put them in nice bags, so they take a nice ride by flying, and then they arrive, and we have these people already there to try to take the animals in a safe way, and give a look to these people that are over there. It looks like they are part of the crew, but they are really part of the community. So this is the community in which we were working with. These are native people from Sonora. And so they helped us, and they are also participating in all the management of the bighorn sheep. So they take part in all the process, and we set these kind of stations for working with the animals on the shade, giving them water in baths, to cool them down if they are overheating. And we use oxygen to provide them also. If they were chased, they need some kind of oxygen, because they get they can go into sometimes into shock. So we take care of all these things. And that's, for example, one of the bighorn sheep receiving the oxygen. And then we mark them so they can be transported. And once they are transported, we have to take off their blindfolds and everything. So they go calm, either they go by car or they go by airplane, again, with all the precautions for doing that. And this is like a really, really resumed way to say what it cost on several trips of several groups of bighorn sheep that we were moving from Sonora to Coahuila, and to restart those populations that were extirpated. And I can say they were very, very successful. It did take some years. We don't release them just back to the wild like that. Most of the times they are released like this in you can see it's almost wild, because the fences are really, really far. So at least we can have certain control over them if in case they got any injuries or anything. Plus, we want them to settle, to breed, and then increase that population, and then release the animals to the wild. So without almost any contact, sometimes not even providing more food if the area is enough big. And if it's not, we provide some food, but then we release them. And the numbers are very important. The more animals you release, the more success you can have in reestablishing a wild population. So this was a program done, and right now there are wild population in Coahuila from bighorn sheep successfully reintroduced. So we put some collars, satellite collars, just like the Rosamir was talking about before, about putting those backpacks. Well, we don't put the backpacks, but we put the collars, the satellite collars that give us the information where they are moving and if they are surviving. So that's about the bighorn sheep. And now I'm going to talk a little bit about the pronghorn. And the pronghorn is, it was also the same case. Some of our populations got extirpated, but a lot more than the bighorn sheep. You can see that it used to be all the way to the center part of Mexico, and those were extirpated. And now it's only like those darker points that you can see over there are just the remnant populations that we still have, and some of them are also from the reintroduced animals that we have to Coahuila. So in Coahuila, they were also extirpated, so we decided to bring pronghorn. But in this case, we didn't actually have any healthy populations in Mexico from which we could bring the pronghorn to the other states. So we started a binational program with the United States and brought actually the animals from the United States to Mexico. So we participated in a program also in which we were giving some of the pronghorn that are a different subspecies from the rest of most of the animals in the United States are from a certain subspecies. And there's a particular one in Arizona that shares with Sonora. So there was a program in which we were exchanging some animals from Sonora to Arizona. And that was one of the programs. And then in another program, the United States actually gave us from the other subspecies back some pronghorn to bring to Coahuila. So we actually brought 300 pronghorn that were going to be distributed in 3 different parts in Coahuila. And each one of us had different trailers to bring all the animals, but don't think we were putting 100 in each trailer. We actually took 4 years to bring all the 300 pronghorn. So we actually, this is a very, very interesting way in which they do it. We participate in making this funnel trap that it's more or less like this. So the helicopter finds a wild group of pronghorn, and they manage to make them go into this funnel, which is really open and wide in the front. And then it starts shrinking until it gets to the last part, which is really just a small room where you can start working with each of the pronghorn. So once they are in the funnel, they use people to actually push them. So they get kind of like a little bit scared. So they move to the other side of the people. And that's how they start going to the inside of the funnel. And then they close the door, and then they just start bringing them by small numbers into the last part. And from there, you can work with them. And again, we use certain vaccinations or anti-inflammatories or things like that, just to have them properly taken care of and put them in the trailers. And then the process also starts with putting some collars. These are VHF collars. And the issues about going through the border with all the permits and everything to go through, while the pronghorn are still there, but under the shade, we put them some fans, we put some water, food, etc. And we even have some trailers with air conditioner for them, so they don't get overheated. So to try to get them in the best condition. And so really, we had a very, very successful also movement of all these animals. As you can see, they were really resting and not really stressed. And until they went back all the way to Coahuila and got released over there. So this is the story of really the pronghorn. And again, I'm just putting it into a few slides for you to see all what it really took, like 4 years for the pronghorn. In the case of bighorn sheep, when they were released back to the wild, it took about 8 years, but it was also very successful. So these are kind of like really, really good stories about what can it be done to kind of compensate the damage that we have done by extirpating these animals. Of course, when you work in these reintroductions programs, the most important part is working with the people that are going to receive the animals, and that the cost for the extirpation is not there anymore. So in this case, for example, the bighorn sheep and the pronghorn, between diseases and overhunting were the main reasons and loss of habitat why they were extirpated. So we worked with the people in a habitat that was restored and with people that wanted them back. So we were sure that the animals would prevail, because if not, the reintroduction program wouldn't work. So anyway, that was a very successful story, but now we go to the harder part, just like Jesse was saying. The hard part about working with predators is that not many people exactly like them as much as the pronghorn and the bighorn sheep, right? So the predators have this problem that many people do not like them, because they predate the animals from which the cattle owner lives. So it's a different issue. With the bighorn sheep and the pronghorn, is like, oh yeah, I do want them back, no problem. But with these animals, we're not translocating them, but we're working to protect them and not lose more of the populations that we can lose, or the individuals even, that every animal counts. So these are just some of the pictures that we have from people that retaliate on jaguars for predating on their cattle. So the main thing here is working with the people, trying to change their minds, working with the ranchers, also understand their problems and try to work with them the solutions. So one of the things we started working with was kind of a contract, an agreement with ranchers in which they wouldn't kill the jaguars, or in this case also the pumas, because it affects them both. If they put a trap for pumas, it does affect the jaguars. So we work with them. No more killing of predators. It is illegal to kill predators, but nonetheless, they still kill them if they kill their cattle, even if it's illegal. So we just wanted to make sure that, they understood that on our agreement, we were going to help them, but, in change of not killing the animals. And in our part, we were going to actually work with them on the solutions to decrease the predation problem of their cattle. So that's, just an example of the contracts we made. And what we were going to help them was, okay, what do you need? What are your needs? One of the problems is that they have the cattle all dispersed and they don't really manage the cattle. So in order to establish a breeding program, or to have areas in which they could have the calves and not in the same areas where regularly the jaguars are, they need certain infrastructure. We're talking about the north in Mexico, which is really dry. So there's, one source of water, and then they don't have a way to pump the water to other places. So, okay, you need a pump. We'll put solar panels, have you a pump, and start making the changes that you need to decrease the depredation. So then we can really work on the solution together. So water tanks also so they can distribute them in other areas for their cows. So they were really happy about it, and they kind of understand then, the importance also of the predator, that we were working with them, but also we were tending their needs. So in another part of the program, we also brought peccaries, which are a natural prey for the jaguars, because of the poaching and the overhunting, the prey is also reduced. So the jaguar doesn't have something to eat. So that's also a reason why they go and eat the cows. So we also brought peccaries to them in different parts of the ranches. And we also assisted them in changing and switching to the breeding control program for their cows. So we brought a veterinarian also specializing one for the cows, and started making those changes. And this is what we want to see also, like with them, working with them, we can have jaguars and we can have cows. If we have proper management and we have good prey, their predation really decreases a lot. So they were really happy about it. And this is what we really like, like a person that is satisfied with the changes we're doing. And look at this guy. This is one of the ranchers that we were working with, and it's his livelihood. He wants to protect it, but now he's not killing jaguars anymore because he doesn't feel as threatened. He feels like he's supported, and he's doing the changes to decrease the depredation. So that's the most important part, to solve the problem, change mentalities, change the way we can solve the problem and make a coexistence with the predators, even having the cows. And that's also like another case from the father and son. It's very, very important also to try to always make all the community part of it, just like we were saying before. I mentioned the other community with the bighorn sheep, and here's the same: is the cattle owner, but also this guy, for example, he already liked the jaguars, but now we're working with him, and his son is also participating in conservation. I mean, really great people. And not only them, it comes the fisherman from the area and the cowboy from the neighbor. Everyone wants to be there, and we invite everyone to be there, because once they see this, once they can touch the animal and they can make a connection, then it's easier to make any more changes. And then they don't fear the animal, as much as when they haven't even seen it. So, so working with the people is really one of the most important things in any kind of conservation program, I think. And just to finish this part, although I'm going to show you a video, which kind of resumes everything I just talk to you about, is these main things. Like everyone who watches all these talks and everything maybe feels like, yeah, but what can I do about it? And like you show in the video from the whale shark, it's get involved, right? And you can get involved also, even with small wild animals that live in the area, or if they don't, you do have a lot of power, because we have to decrease the human population growth, because we're taking the space of all these animals. We have to decrease all the consumption ways that we have, because every time we buy a cell phone or anything, then we're taking minerals, we're taking everything that has an impact on the lives of all the biodiversity, on all the animals. And even if you live in a very crowded city, it's very important your decision about who you vote, in political issues, because we have to consider the environment. And those things, if every person would do it, really setting their mind about all the impact we have in the environment and in wildlife, we would make really huge changes. So we all count, every action counts. And of course, the more passion we get about it, much better. But definitely wildlife needs us. So the more we get involved, the better, right? So let me just then put our video. So let me see. Okay, let me see where I am.
Take your time.
Sorry.
We got tons of time. You have you're the fastest speaker of anyone so far.
I know, it's just because my video is 6 minutes. So, yeah, so I was just checking all the time. Okay, so Beautiful. Can you see the video over there?
Yes, that's great.
Okay, perfect. So we're gonna start this. I'm gonna unplug this. So let's see.
You hear it? You see it? That's great. Lovely.
Okay, I'm gonna just decrease a little bit over here. So this is part of what I just told you about. It is the helicopter using the net gun for the bighorn sheep, and there it comes all the crew. And these animals are not anesthetized, so the fastest we work is the less stress we're gonna put the animal on. So we have a team in which everyone knows what they are doing. So we split tasks, and this guy's taking temperature, someone else takes blood, or put some injections, while the other ones are working on putting a satellite collar, so we can decrease the time. So there they go. Release. That area is not completely wild. It does have a fence, but you never see really the fence until then they get released to the wild. And those are the pronghorn. That's the helicopter starting to push the animals to the funnel trap. And there's all the people ready on the other side. And that's also using the net gun also in pronghorn to put some satellite collars and study the ones also that were in Mexico already, since there were just a few that was part of the binational program. And then we skip to the jaguar part. We work with the ranchers, but we also place collars, satellite collars on the jaguars, to actually make some research about if the things we're doing to reduce predation actually work. Like bringing more prey and changing the cattle management, if it really makes reducing the predation. So we put the collars and we can find out more about the jaguars, where they are moving, etc. This one already had the collar, was not working. We had to replace it. And when we put to sedate one of these jaguars, we protect their ears, protect their eyes because of the anesthesia. Now, these ones you cannot work without anesthesia. And we also give them oxygen, which always increases, makes better all their physiological parameters during the anesthesia. So she's getting a new collar. And like you see, everyone knows what they are doing. We're also split. We put IV fluids also to help her metabolizing the anesthetics better. And we reverse them. And until she's fine and she can go, then we can feel okay. We can still follow her, all the movements because of the collar, until we know she's killing again and being okay. That's a male. And we have also worked with kittens, although this kitten is from a puma, but we can also put collars on them to see how long they stay with the mom and where they move, etc. And that's also another project we have from bears, also collaring bears to know how many they are, and make some estimations on their density and how they move, and which is their best habitat that we need to protect if we want to conserve also the black bears. So those are a few things that we can do as a wildlife vets in the conservation projects from not only me, but several researchers. Okay, so that was the video. Let me see. Okay.
Yvonne, what a spectacular program. And by the way, the idea of bringing in the whole community, the children of people that you're working with, the local fishermen, cowboys. So I'm in Canada, we have wolf predation of some of the livestock here, and that's a big deal, is getting local ranchers that are really resistant to the ideas at the get-go to get their friends and colleagues involved. Because once you have that community support, it builds off itself in such a beautiful way and leads to such amazing outcomes. That was a spectacular program. Yvonne, Natasha has the same question that I had at the beginning of your talk, and that is, how long does it take to train everyone involved? Do you do rehearsals? I mean, you make it sound so easy to be in a helicopter flying beside something and shoot a net gun at it, but that sounds very difficult. How do you get this expertise?
Yes, no, I'm actually not the one net gunning. I have been in the helicopter. When they net gun them, I go down with them, and do all the vet stuff. But yeah, net gunning does take some training, special training, and most of the people that work like that, start really young and probably take about, I would say, like 6 months training. It's not really like that difficult. The only thing is that you have to aim and know how to shoot, and also the security part, because it's very important. All these people know that, even a pen can be a problem if you're in a helicopter. A thing where you are writing your notes, if that thing goes off and hits the blades from the helicopter, you can even crash. So it's that important. So everyone does have to know all the security issues when you get into the helicopter.
Again, you continue to make everything that's pretty amazing sound very casual, but that's a testament to your expertise in all these amazing places. A question I've always wanted to know, so you have these animals, you have them come out, and you fly away, or you drive away. What do the animals do next? I mean, they're in this completely new habitat, this new country in some cases, and do they start acting normally? Do they start grazing within minutes? Or are they freaked out for like a day or a week? How do you monitor that with these creatures that are in this entirely novel situation?
regularly they are freak over for at least one day, like the pronghorn and the bighorn sheep. That's why it's so important to also release them in groups. So if you're going to release like you just capture like 10, right? And it's much better if you can still have other 10 and then release at least 20 all together in a group. And pronghorn, for example, when we moved them from the United States to Mexico, some of the animals got scattered from other groups. And the family group is very important. So the first group that we released was all family group, and they stayed together. And then we had some individuals that came from another group, and then they just, couldn't catch up, couldn't really get along with the other animals. Even you would think like, oh, they are pronghorn. But no, they have family ties. So it's much better to always capture all the family and not from, pick from different groups, because they may not get along. And the problem is that they will stay alone, and then they can be easily predated if they are not in a fence. And even in a fence area, we had these animals that took them about 6 months to actually get with the other group. So yes, that's a very important observation. Yeah. They do, you have to take care of their family group, and they do get stressed, very stressed. And it might take weeks also for the animals to get into the new environment.
Yvonne, time flies and you're having fun, so I want to leave off with one final question. I'm a fan of yours now. I'm a big fan, and I want to follow your work. How do I do that? Is there a website, social media pages? Where do I go to learn more?
Yeah, we do have one for the jaguar stuff. Is on primeroconservation.org. That's the one from there. And I think it does have an email over there in which you can contact me. That would be the easiest way. Fantastic. Or you can also, of course, you can find me on Facebook. But
Facebook's the go-to for everybody, but we'll make sure that our audience has that link for the website at the end. And thank you so much for joining us today. It's such a special opportunity to hear your story, and I really appreciate you taking the time.
Thanks, GC, and thanks to Joe also as well. Thank you so much.
Thank you to Joe. Well, Yvonne, have a wonderful evening. I'm going to introduce Joe, speaking of Joe, in just a minute. For those who've been tuning in since 6:30 this afternoon, thank you so much for sticking around. We've had an audience now for over 4 and a bit hours, so welcome in. We're wrapping up with 2 more programs that my colleague Joe is going to be hosting. I think a lot of our speakers have made note of this, but Joe Grabowski is the founder and the CEO of all of this effort, bringing in conservationists from around the globe, not just today, but over the next 2 days of really special programming. And so I'm excited to bring him back in to wrap up our meeting today. Joe, nice to see you again, man. Come on in.
Hey, Jesse, how's it going?
I mean, it's been 3 incredible programs back to back to back, and I was watching with popcorn for most of the rest of the festival. So I'm like, again, I work with you all the time, but I'm also in awe of this thing that you put together. It's amazing.
Well, it's I haven't left the screen. It's great to see so many amazing scientists, conservationists, filmmakers from around the world joining us and sharing their stories. And it's so important that we're sharing those biodiversity stories now.
Yeah, absolutely. Well, I don't want to take up any more time that we could be spending with some of those amazing speakers, so I'll leave you to it to introduce our next speaker, keeping this jaguar story going, and have a wonderful wrap-up of BioFest Day 1.
All right. Thanks, Jesse. We'll see you tomorrow. All right. Well, let's keep things moving right along. Let's stay on schedule. I'm really excited for our next speaker. We have Yara Barrows joining us. She is a biologist who has worked with conservation of endangered species all of her life. She's a coordinator of the Jaguars of Iguazu project, working for conservation of jaguars in Atlantic Forest in Brazil. She's also a member of the Conservation Planning Specialist Group, 9CPSG of the IUCN. So I'm going to bring her in live with us right now. Here we go. Hey, Yara, how are we doing tonight?
Hi, Joe. How are you? Thank you so much for inviting me. I'm watching the lectures. It's amazing. I love it.
Oh, it's so great to have you joining us. It seems like such a long time since we connected and we had a test call together, and now it's so great to have you live in the festival, ready to share your story with us.
Okay, it's my pleasure. Thank you.
Excellent. Well, let's let you take over for a little bit. We're excited to dive into your world, and then of course we'll have a little Q&A time.
Okay, I'll share my screen. Let's see.
All right. Always a bit of an adventure with the share screen. You never know what technology is going to do, but I see it coming up, so that looks good.
How is it? Can you see it?
Perfect. We're ready.
Okay. So, hi everyone. Thank you for having me. I'm always loving to talk about jaguars. It's always a pleasure. I am the executive coordinator of the project Jaguars of Iguaçu. Jaguars is onça in Portuguese. So our mission, this is Kaluana, an amazing male that we have in the forest, is the conservation of jaguar as a key species for the conservation of the whole national park biodiversity, actually. Because as a key species, we used to say, if there are jaguars, there is life. So if the jungle is good enough to have, in a long term, a jaguar population, it means that it's doing well. And where we are, well, we are in Brazil, in the south of Brazil, here in the state of Paraná, here in the border of Argentina and Paraguay, and here is the national park. As you can see, I used to say it's an island of life in the middle of a very deforested state, and a very important place for many endangered species, including jaguars. And when we talk about Iguaçu National Park, everyone thinks about that, the falls. Of course, the falls are there. They are absolutely amazing and stunning, but the national park is much more than that. If you see here, all this is the national park, and here are the falls. So it's a minimum, what you can see when you visit, it's a minimum area. The park has 185,000 hectares. There are 10 cities around. Here is Argentina and Brazil. So we will not save jaguars by working only inside the national park. As Yvonne was saying, the work with the community is very important. So we work in the 10 cities around the national park. This is India, one amazing female with her cub, Mitacoré. So people say, why do you focus on jaguars? Well, because it's an apex predator. It's an umbrella species. What means that if the jungle is okay if the place is okay for jaguars, many other species will benefit of that. So by protecting jaguars, you end up protecting lots of other species. We have two species of big cats in the region. We have pumas also, or cougars. And the jaguar distribution, jaguar has a problem. It's an endangered species. In the country, it's endangered. It lost basically half of its historical distribution. The dark green is the actual one, and the light green, the historical one. As you can see, it lost half of its area. And from what is left, 50% is in Brazil. So we have a lot of responsibility in keeping this species going. And we work basically in three, no, don't do this to me. Let's see. This is Peter. We work in three pillars, basically. We work with awareness, we work with research, and coexistence. We always say, people say, is this a research project? I say, no, it is a conservation project that has research as one of its pillars. This is Kaluana. In terms of awareness, we have lots of products to work with the community. We call products. We have Jaguar Chat, we have Jaguar at School, Jaguar on the Road, a kind of pint of science with jaguars. We go to this Chagalini, we go to schools or bring kids to the national park, and we talk about jaguars, about the animals in the region, about security issues. So we talk and talk and talk to children. I used to say that if a child will baptize his doll, we are there talking about jaguars. We are always talking about jaguars the most we can. Of course, all the activities with the community we have to stop a little bit during the pandemics, the worst phase of the pandemics, but now we resume it after my team is vaccinated. We are resuming them. Jaguar Talk, we go to communities in the end of the day with people who live on the border of the national park and talk to them about security, about good management practices, how they can manage their cattle so they are less vulnerable to big cat predation. This is our version of Pint of Science. Many people doesn't want to come to formal lectures or stuff, so we go to a bar and talk to them about jaguars. And we drink, obviously, which is very nice. And this is Jaguar on the Road. Basically, we get our stuff, and to whatever there are people, we are there. Fairs, exhibitions. So this is with Boy Scouts here in many different towns. We just go there, put our booth, our Jaguar booth, and then we talk and make activities with children, adults, and we have a very good response from the public. We created a citizen science program called Team Pantera. So at the moment we have only 9 people on Team Pantera from 4 cities. We train them. They spend 2 days being trained by us, and we visit them regularly. So they are our focal points in the cities where they live. 2 of them even created a trail inside the national park called Jaguar Trail. They keep it. It's an amazing experience. And this is basically what we want. We want to replace the fear that people have about jaguars for enchantment. This is something that Jeff Swanagan said, touch the heart to teach the mind. We want people to know that jaguars are not there in the border of the forest and waiting to jump on your neck and kill you. They just want to be jaguars and be in the forest and do their jaguar stuff. Because this thing of thinking that jaguars are always ready to kill you is what feeds this fear, and you tend to want to stay away from what you're afraid of. You want it dead or you want it away from you. And we want to try to change this with information. And I always say that I like to use enchantment as a tool. Because I think that only knowledge by itself doesn't have a transforming power, but when enchantment opens the door, and knowledge comes in a more transformative way. So we work a lot to make people fall in love with jaguars, to make them see jaguars through our eyes because they're full of love. And I like to use this example. This was many, many years ago when I was young. And I didn't even dream about working with jaguars. I was working with macaws at the time in this is Amazon. And in town, we saw this. This is a very young jaguar. And I said, why the hell did you kill this animal? And they said, well, because maybe one day when he goes grow up, he will kill our cows. Just maybe. So they kill just in case. So the fear that people has kill animals. And not only jaguars, everything they're afraid of. So this thing, no, this cannot be like this. So we work to change this. This is Thiago. We work a lot with coexistence. If you can see, here is the national park. Here are people living. So we want them to share the land in a peaceful and harmonic way. So we are there all the time. Last year, we made like 250 visits to 78 properties around the park. We are there all the time. We don't go there and say, oh, okay, good luck. We are there all the time. We talk to them. We see them. And basically people who are excluded somehow, they are fed up of being visible. So when you see them, when you talk to them, understand the fears, understand what they think, it's amazing. And this close contact that we have allow us to diminish the fear that people has, build a confidence relationship. And there are papers, articles that show that, because with jaguars, the perceived risk is actually bigger than the real risk. And when people trust who is in charge of managing this animal, it decreases the perceived risk and increases the tolerance. So our constant presence increases the tolerance, and it's an amazing opportunity to exchange knowledge with these people because they are amazing. And we produce a lot of material also to support these activities, from to how to dispose carcass disposal, because here it is a problem, how to coexist with jaguars, what are the animals of the national park, so people can connect not only with jaguars but with the national park. Jaguars exist, they need a national park that is very well and kicking, alive and kicking, so we also want them to connect with the national park. And we work with predations. Everyone who works with big cats will face this. This is C3. This is an old male. As you can see, he's blind now with one eye. And when someone, this is on predation event, and when someone tells us, oh, they phone us, oh, I have a dead cow, or I saw some footprints, or we try to go immediately, although the area is huge and we are only 5 people or 4 people. We go immediately as soon as possible, and we notice that this makes a difference because they don't have to shoot first and ask later. They know if they call, we'll go. We evaluate the situation. We identify the predator by fixing the carcass in the floor and putting camera traps so we can know exactly which predator was. Even if you by the carcass you see it's a jaguar or a cougar or dogs, we want to identify which jaguar. We install lots of preventive measures like electric fences, blinking lights, bells with blinking lights, painting the butt of the cows with big eyes. So every possible protection device available, we try, and we are having success with many of them in many properties. For a year, whenever the mirrors of predation, we go there, do everything, install cameras and the devices to prevent predation, and we monitor this place for a year at least. It's a continuous monitoring. At least once a month, we're there, sometimes more. And we try to identify alternative sources of income. Because when you get there, the first question is, you're gonna capture this jaguar and take it away. Okay? We said, no, well, there's no way. There's nowhere else to take this animal. And second question, you're gonna pay for this cow. Okay? We said, no, we will not. First, we don't have money, which is a very good reason. But we also think that only reimbursing doesn't solve the problem of bad management. If you just pay them, they will feel they will not be stimulated to change the management many times, because we also provide guidelines about how to manage your cattle to avoid predation. Many times you don't need big devices or electric fences, just small adjustments in management helps. And we help them through this process of changing the management. And regarding alternative sources of income, what we try to do is, if we have a talent, we identify a talent that is already there to produce a product or a service, we help them develop these talents. So they generate or a product or a service to which the jaguar can aggregate value. And this aggregates value in keeping the jaguars alive. This is Marcus. These are Marcus and Susie. They had in 2018 3 cows killed by a puma. And they were super angry when we got there. And we implemented lots of protective measures, the protection devices, and then we saw he fabricated cheese. We said, well, where do you sell this cheese? No, just here in the community. So we started to advertise about this cheese. We named it Jaguar cheese, queijo da onça. And he started to sell a lot. And now he developed into much more than that. He created, behind the national park, his area, he created this kind of kiosk called Jaguarete Ranch. Jaguarete is jaguar in Spanish. And now he has a bar, he sells products, he built a pool, he built a camping area. He's receiving lots of bikers, lots of visitors, and he's making a lot of money. This area became a super success. And now he built this. It's a Jaguar, Toca da Onça is like a Jaguar Airbnb, and he rents to people. So he's making money because of the jaguars and cougars without us having to give him money. And also he was like invisible, because he used to say when he wanted help from the city, no one wanted to help him. And now he gives interviews on TVs. He tells people the stories of jaguars and cougars. He's respected. He's admired for helping us. So he's a very good partner. He and Susie used to say they never thought a puma could bring him so much happiness. And he used to say, if I knew things would get that good, I would have been given 3 cows more for the cougar. I don't think he would, but it's good to see that he associates the improvement and his financial improvement to cougars and jaguars and to the work he does with the project. We also give the signs of property, jaguar-friendly property. So people who work with us, this is Jorge from Other Property. Properties who work with us, help us, implement the measures we propose, receive us well, let us install cameras in their property. They receive this. It's not a certification, it's not an ISO, but it is we recognizing your importance, we recognizing your help, your value. And we did 4 properties so far. It started last year and it's going really well. And this is croissant. We also work with research, and the main lines of research as we know we have jaguars, but how is the prey base? Do they have food available? So we are conducting a long-term study on prey base together with other researchers in all the Atlantic forest. We work with diet, not like Pantanal, when you see jaguars jumping in the water hunting caimans in the forest. Very, very rare that you see a jaguar. So we collect feces and they are evaluated to check the content. We have lots of camera traps installed. This is Vanya, who works with research. We have many camera traps installed in the park. And we also make a general monitoring of fauna in the park. So we get amazing, we have biodiversity, anteaters, peccaries, deers. This is all jaguar prey. Tapirs. We had one jaguar that predated one huge tapir. Ocelots. Jaguarundi. And a kind of egret called hirsocó. We'll call it cutia. It's kind of a guti. And Tyra. So this way we also monitor. We evaluate continuously the biodiversity in the park, and now we started to sampling the corridors, the vegetation corridors outside the park. Here is the national park. Here are some vegetation corridors. Each yellow dot is a camera. So we want to see if big cats are using these areas and their potential to be developed as connectivity corridors to other areas. We also capture and monitor jaguars. For us, it's very important to understand their movements, the size of their territory, because help us understand their need and how they use both countries, because we are in the border of Argentina. We work very closely with Proyecto Yaguareté in Argentina. We used to say that we are projects divided by a river, united by the jaguars. So the GPS collars, they help us to monitor them via satellite that we receive the points where the animals are. But sometimes when you have problems with the GPS, we can fly over the park and try to get a VHF sign, and then we could go walking in the forest and find the collar or some points where there is a cluster of points and find maybe a prey, and it's an amazing view. This is croissant that we captured in 2018. Just to show the importance, here are all the points that croissant sent. Here is Brazil. Here is Argentina. Croissant is one of the 7 jaguars already identified that cross between Brazil and Argentina all the time. So as you can see here, he has a home range of 264 square kilometers and a territory of around 66 kilometers. So this is a very important data for us. And we do a census of the animals. This is the longest and biggest effort to census jaguars. Every 2 years, together with Proyecto Yaguareté, we sample an area of 600,000 hectares in the 2 countries. It's simultaneous. And in the 90s, they estimated that inside the national park in Brazil, we had 765 jaguars, and it dropped dramatically in 2009 from 9 to 11 animals. So the population was almost extinct. They were being killing to extinction, local extinction. And then conservation work, I work with the police to try to impede the activity of illegal hunters. Make the population starting to increase. It more than doubled in 10 years. We are super happy. And the last census was 2020. This is an estimation, not an exact number, but the carrying capacity is 50. So we still have a long time to go. This year is a census year. In 2 months, we're going to start we put camera traps all over the park, and they stay there for 3 months. So we have around 24 animals in Brazil, but if you consider all the green corridor considering Brazil and Argentina, we have an estimation of 90 jaguars. If you consider all the Atlantic forest here, that is Brazil, Argentina, and Paraguay, the Atlantic forest has 250 to 300 jaguars. So this region where we are has basically 1/3 of all the jaguars in the Atlantic forest, and is the only population in the Atlantic forest that is increasing. So it's our pride, but also a huge responsibility. And we have amazing, and on this very hard work, because it's a very hard work, convincing people that an animal that they fear has the right to survive is not a very simple task. We have this amazing thing. This is Indira, which means pure beauty. Her with 5 months old crossing the road inside the national park. And here, when she was already 3 years old in 2021, so reached sexual maturity. And this amazing thing. This is Indira with her cub, Aritana, already now he's almost 2 years old. So it's so difficult that a jaguar reaches the adult age. It can die for so many reasons. And see Indira, that we basically saw her as a baby, as a cub, and now be a mother, it's an amazing experience. And we used to say, I know it's kind of cheesy, but I used to say that to save jaguars, you have to talk about love. To say anything, you have to talk about love. People will not help us save jaguars because they are endangered. For many people, this doesn't mean a thing. So they will help us save jaguars if they don't want to live in a world where this golden eyes doesn't exist anymore. They have to fall in love with these eyes and believe, no, I will be part of it. I always used to say that to work with conservation, you have to transform people from spectators to actors of the process. We always say to people, let's take care of our jaguars together. So we don't say it's our jaguars, it's the jaguars of the national park. We say it's our jaguars. It belongs to all of us. So this is how we work. So we work a lot in Facebook, Instagram, and we use a very simple language, a language that people can connect to. This is we have a very small team. We are 4 people working like crazy to work in this huge area. And we are absolutely, I'm very lucky. We are 5 people with me. I am very lucky to have this very motivated and passionate team. And as Arnaud said, conservation is you don't do it alone. You always need help. So we are very thankful to our many collaborators and sponsors that make possible for us to work. I think I talked too much. So very quickly and briefly, this is the work we do here to protect the jaguars.
All right. Well, Yara, thank you so much. That is, I mean, you said you thought you talked too much, but it was just not enough time to share all the amazing work that you're doing. And the results, I mean, speak for themselves. That story that you shared of that couple who have kind of completely flipped 180 on their opinion on jaguars, and they're they're champions now. They're doing so much. They do so much, and they're doing so much, it sounds like, to help protect them as well. So I'm wondering, are they, people like them, are they good assets to you? Will they speak to the community? Will they talk to other people about what they've experienced?
Yeah, they talk to other people, they talk to neighbors, and they talk, especially Marcus and Susie, because they receive like hundreds and hundreds of people on the weekends now. So and even during the pandemic, it happened because it's a very open area. So people who couldn't stay indoors anymore went there. So yes, they talk to people and they talk about the importance of jaguars, the importance of cougars, and mainly they say how they can survive, how they can coexist, because he continues to have jaguars and cougars basically on his backyard. But now he knows that his area is protected, the management is good, and he's making money off them. So he not only knows today the intrinsic value of them, but also he can make money. This is our success case. Not all the cases we work are like this. Not many people are easily convinced, but it is possible. We believe it's possible. And they are like, Marcus is like our ambassador. And this is what these people become, ambassadors. It's amazing.
Yeah. So speaking of the successes, you work with kids, you work with adults. Do you find one group easier to get on board, to change their minds, to get them, between the kids and the adults?
Oh, the kids are very open because the kids still don't have kids are kind of afraid, but they still don't have the years of being afraid that an adult have, because in adults the fear is very deeply inside and you have to work to take it out. With kids it's easier because kids are still easily touched by enchantment. Thiago and Aline, two of my staff, they did this small play where Thiago is the hunter and Aline is the jaguar, and the public, the children, they can interfere if they want. And it's amazing how kids attack the hunter and totally they protect the jaguar, say, no, go that way, the hunter is coming, go hide. So it's like in 5 minutes kids are completely defending the jaguar and like hunting the hunter. It's very easy and it's a pleasure to see.
All right. So you mentioned there's 10 cities around the park. Are the jaguars fairly cautious, or will they venture right into those cities?
Jaguars are most stay inside the jungle. Eventually they go to properties and predate, but properties that are very close to the national park.
Yeah.
Cougars are they have more plasticity, ecological plasticity. So cougars go a little bit more. They go under sugarcane plantations, corn plantations. So it's easier to see cougars outside the forest than jaguars, but it happens eventually. But it's more difficult than seeing cougars.
Yeah.
Or pumas.
So just kind of on that question as well, in the map that you showed, there was lots of roads. Are roads an issue with the jaguars, so crossing the roads? And is anything like underpasses or overpasses being used?
No, we have no passages for fauna in the region, what is terrible. Inside the national park, when jaguar was killed in 2009. After that, never, but 2018, a female was killed by a car in Argentina on the other side. It was a female, she was pregnant with 2 babies, and so it was terrible. And we have a big issue here because the national park, in the past, there was a road that cut it in half that connected to cities, and it's closed now since 2003. And there's eventually this political pressure comes and goes to open this road. And now at this moment, we are facing this threat again. So if it is open, it's terrible for animals being hit by car, for hunters to get in, for illegal activities that can bring people and kill jaguars. So yeah, road is a problem. We don't have here since 2009 any jaguar killed by car, but we just released a cougar that was hurt by a car 3 years ago, and another institution made a rehabilitation for 3 years. So yeah, roads are always an issue near national parks or where we have animals.
Yeah. All right. Well, I mean, that graph you shared was scary to see the numbers fall so much, but it's good to see that it's trending in the other direction. I have no doubt it's because of the amazing work that you and your team are doing, changing minds in the community, and like you said, doing it with love.
I hope so. No, I believe so. I believe that there's love is the way. And I know it doesn't sound scientific, but really you have to do that. We have to touch people's hearts, otherwise you will not change the minds. For me, it's very, in 30 years working with conservation of other species, this is very clear for me, I guess.
Yeah, people protect what they love.
Yeah.
Well, Yara, thank you so much. Thank you so much for joining us tonight on day 1 of the Global Biodiversity Festival. Thank you for the work you're doing and being here with us to share it with our festival attendees.
Thank you. Thank you so much for inviting me. It was a pleasure, and I wish you all a great festival.
All right. You have a great weekend, and I hope you're able to tune in a little bit more. Thanks, Yara.
Okay. Bye-bye.
See ya. All right, we are moving to our final stage of the evening. So we'll be back in action tomorrow, bright and early, starting at 8 o'clock a.m. Eastern. And we have a full day of events from 8 Eastern straight through till midnight, actually 11:30 p.m. tomorrow. So starting at 8 a.m. and going right through to 11:30. Of course, we know Many won't be able to sit the entire time, which is why we're going to put up a playlist of all of the events. You can check out the events you weren't able to make live, but do join us tomorrow as we get started. It's a great lineup tomorrow, from speakers to trips out in the field, virtual field trips, to places like Costa Rica and Gabon. It's going to be a great day tomorrow. You don't want to miss it. So this will be our fourth continent we're going to visit tonight. I'm so excited. We're going to head to India, and we have Aishwarya Sridhar joining us. She is a 2022 National Geographic Explorer. She's an award-winning wildlife photographer and filmmaker. She's an emerging fellow at the International League of Conservation Photographers, and she's been working towards policy protection for wetlands using the visual medium. And she was recently selected as one of the Explorers Club 50, which celebrates global exploration leaders around the world. So I'm going to bring her in live with us right now. Aishwarya, how you doing?
I'm doing good. How are you, Joe? Thank you so much for the kind introduction.
Oh, of course. And congratulations on the grant with National Geographic. You must be so excited.
Oh, yes, I really am. Really looking forward to going on the field and working on that project.
Excellent. Well, this is day one of the festival. You're like the headliner at the festival, the headliner in the evening when the big act comes out at the end of the night. So thank you so much for being with us. And I'm going to let you take over for a little bit.
Thank you so much, Joe, for inviting me to be a part of this amazing festival. So I'm going to quickly go ahead and share my screen, and I really hope that works.
I'm sure it will. I know we practiced not too long ago, so I bet we'll be good.
I hope you can see the screen, right?
It's loading up. I can see that the share started. You just have to bring the presentation up to the front.
Can you see it?
Not yet. I still have it. It's still on the. Let me just double check. I'm going to bring it in. Oh, it disappeared. Let's try that share again using the entire screen option. And then, yeah, if you pop the presentation to the front, I think we'll be in business. There we go. Perfect.
I don't know. Why is that happening?
Oh, well, yeah. There you go. Perfect. We're good.
Perfect?
Yep.
All good? Okay, great. So now I jump into my session for today. So basically, I am from India, a land which most people know as being the second most populous region in the world. Now, there's actually more to India than just slums or people or palaces. It's one of the 17 mega biodiverse regions of the world and home to 8% of the world's recorded species. I grew up in the countryside along the coastline. This is not very far from where I live. And I had wildlife in my own backyard. I was lucky, by the way, and that's one of the reasons why I fell so deeply in love with nature. But on the flip side, coming from a nation which is developing, I also got to witness this constant tussle that exists between ecology and economy. And let me tell you, 99% of the times, it's economy that ends up winning. I saw the landscape literally transform over a decade from lush green to dull gray. Mangroves, natural carbon sinks, got replaced with carbon-intensive industries. Wildlife corridors transformed into bustling national highways. And seeing this change, at an early age, I understood the extractive relationship that we humans share with our planet. And I wanted to do something to change that. And that's why I picked up the camera. To tell stories that evoke emotions and inspire action that leads to concrete change on the ground. Right now, the pressure on our planet is intensifying every single day. I'm sure all of you all know that. And conservation and sustainability need to be the top priority of not just you and me. It has to be of world leaders, of corporates, of policymakers, and people from all across the globe. And that's where actually a storyteller comes in. Using the visual medium, we can really inspire action. It's a very powerful medium, and I'm going to be talking you through today about the power of storytelling and how I have harnessed it in my life over here in India. So there are two main focus areas on which my documentaries are usually based on. I dabble mostly in creating documentaries and clicking impactful photographs. So the first focus area I would say is blue chip natural history. This is all about spectacular images of wildlife in their pristine habitat, or really you get to see a sneak peek into the lives of these majestic creatures. And the second part is human interest and conservation based stories. So, hard hitting films that delve deep into an issue and also present tangible solutions. Through both of these and a combination of them, I want people to feel a reverence towards nature, to care for it, and to protect it fiercely. Now, going into blue chip, when I first, started working on my blue chip, I was in college, and I literally had no idea how to do it. Blue chip one is these are documentaries which are mounted on a grand scale. You have really amazing cinematography of visuals of wildlife, and these are films that entertain, educate, and inspire. They take you into the deepest parts of the jungle and bring out the best of the natural world. Now, when I started, all I had was a vision to bring the life of one of the most enigmatic tigresses from the forests of central India in Taroba. She's named Maya, she's on screen right now. And she's although one of my favorite big cats, but she's also one of the few tigers who have really redefined the word wild, I would say. So I first met Maya when I was 15 in 2012. She and her three siblings were at a waterhole. It was peak summer in central India, and the four of these subadult cubs were playing. And suddenly, you had a giant Indian gaur enter the scene. Now, it's a bovine. They're huge. So compared to a subadult tiger cub, the gaur is almost three times the size. And this big guy, he was a bull. He was weighing over a ton, I would say. And this guy entered. The other three siblings smelt danger and they literally ran off. But not Maya. She stood her ground. And she walked up to the gaur and tried to intimidate him. He realized what she was trying to do and he did a mock charge. And that's when she knew that, oh my God, this big guy is not someone that I should be messing around with. And she scooted. But that moment of courage, of confidence that she displayed really captivated my heart. And there was an unspeakable bond that you can say that I began to share with her. Since then, whenever I've visited Taruba, I've made sure that I actively take time out to track her, to follow her, and photograph and document her journey as such. This started off actually as a passion project, something because I really love big cats. But after a certain point of time, there came an incident where she began to display strategizing abilities like that of a human. And that's when I knew that her story definitely had to be told on celluloid. She was a mother of three cubs, and there was an intruding male that came, and the male splintered her family when she'd gone away on a hunt. And what happened was, after that, when she came back, she found no cubs. So instead of submitting to the dominating male, what she did was she went outside her territory, brought back the father of her cubs, and used him to kick out the intruding male. Now, when this incident played out, I felt as if I was watching a Bollywood masala entertainment movie. Because for a big cat to display such level of thinking and of strategizing is really not documented in the wild before. People perceive big cats to be hardcore predators, carnivores, primal creatures who have innate killing instinct, are not capable of definitely feeling emotions or thinking for that matter. But here she was literally redefining her wild genetics. So I wanted to change that perception of big cats as well in people's minds. And that's why I decided to make a film on her. Now, I portrayed her as a Bengal tigress who was a mother who felt the grief and loss of her cubs when they died for the first time, when she became a mother, or when she decided to train her cubs when they were very young, at about 9 months of age, because she lost her mother as a cub and she didn't want her cubs to remain dependent on her for a long time, because you never know what happens in the wild, or for that matter, when she brought back her former mate to overthrow the intruder. So, by portraying her as an individual, as someone who is a survivor, a ruler of one of the largest territories of Tadoba, or even as a mother who would do anything, I mean, literally anything, to protect her cubs, I wanted people to really connect with Maya and to root for her and every single point of the story. And took me 6 years actually to put together the film. And finally, the film is airing on Nat Geo Wild right now. It premiered last year during Big Cat Week. So I'd like to show you all an excerpt from the documentary.
Big cats will go to great lengths to claim their territories and prolong their bloodlines. In this next film, made by aspiring young wildlife filmmaker Aishwarya Sridhar, we journey into the heart of India for a powerful portrait of a Bengal tigress that will do anything to protect her family's future. As part of Big Cat Week on Wild, this is Queen of Taru.
Matkasar. He's pushing closer. Following Maya's scent. He's not impressed by the cubs still hanging around. He makes his intentions clear. He wants queen and kingdom. Maya is not aware of the approaching threat. She's off to patrol her territory and get more food for her demanding cubs. She leaves them behind. Matkasar moves in. Bola is the biggest threat. The two female cubs retreat. But Bola has his mother's fighting spirit. Thankfully, it's just a spat. Bola keeps his life, but has to leave. Matkasar achieves his first goal. The cubs are on the run. He makes himself at home. When Maya returns, home is empty. Again. All that's left is the scent of an intruder. Maya's second chance at motherhood was better in some ways, but her plan to keep her bloodline in this kingdom has failed. Again. She's not receptive to this new male. But Matkasur is not one to give up easily. He's already staking his claim to the territory, marking with scent as far as he goes. He chased off the cubs. Half the job is done. Now he has to wait for Maya to accept him. She's aware of Matkasur, but seems neither interested in fighting him off, nor letting him in.
So that was a small sneak peek into Tiger Queen of Taru. The film really brought about a change in the way people look and see big cats. Now, when we talk about blue chip documentaries and films that are, you have amazing cinematography. You give an audience literally a sneak peek into the animal's world, and by doing so and capturing unusual behavior, you make them kind of fall in love with wildlife. And at the end of the day, when you love something, you would definitely want to protect it. That being said, for blue chip, the next way or another way in which you can light the fire in the bellies of people to do something and make them act is by presenting hard hitting stories of conservation, by digging deep into an environmental issue and then presenting tangible solutions to overcome it. Now, conservation filmmaking and photography is something that I deeply passionate about. Both of them have an innate ability to transcend the psychological barriers that actually exist between an individual and an issue. At the end of the day, we are visual animals, right? I mean, our sense of sight is the most powerful, and we tend to connect a lot with visuals. So, among all the different conservation projects that I've worked on till date, one remains very close to my heart, and that is Panjay. It's protecting the last remaining wetlands of Uran. Now, I grew up in Panvel, and Uran is about 30 kilometers away from my house. And in the initial years, that it used to be a birder's paradise. Vast stretches of marshes and mangroves, the air reverberating with bird calls. It was really magical to be there. But over the last decade, I've seen almost the largest tracts of mangroves and marshes and wetlands being taken up for development because Uran got the entire region of Uran was selected to become the hub of a special economic zone way back in about, I think, 2008. So after that, as you can see on screen, the landscape literally changed. And today, all that remains is one wetland, and that is Panjay. Panjay is about 239 hectares, and it's a beautiful wetland, like all wetlands, which are ubiquitous ecosystems, providing a host of ecosystem services like uh, maintaining the groundwater table, flood control mechanism, a climate change mitigation tool, as well as providing a habitat for incredible biodiversity. Panjay is no less. It sees about 200,000 birds, migratory birds, coming in every year during the winter season. And it also supports the local economy with 2,500 fishermen depending on the wetland for their daily income and livelihood. But sadly, Panjay is also a prime property in terms of development because it overlooks the Mumbai shoreline, as well as you can see the entire sea from there. So Panjay was allocated, despite it being a CRZ1 land, which is Coastal Regulation Zone Level 1, which has the highest level of protection for a coastal land. But despite that status, Panjay was allocated to become a residential complex. And construction work was about to begin. So that's when I decided to make a documentary detailing the plight of this wetland. And the documentary aired on DD National, which is India's public broadcaster. And once the film became viral, I took a couple of media spokesperson there to just showcase the wetland and to do a media story as such. And what we discovered was shocking because the entire wetland had completely dried up. It was bone dry. What looked like the top picture, during the peak of summers, had turned to to look like this, the bottom picture, completely dry after monsoons in October. Now, this was really something which is shocking. So, investigations revealed that a dam with 71 sluice gates had been built at the main inlet of the tidal waters. Now, these sluice gates operated in such a way that during high tide they shut, which meant that water could never enter Panjay, but during low tide they opened, which led to all the water that was still standing at Panjay to recede out of the wetland. And finally, the wetland dried up. Now, this was definitely a way to destroy the biodiversity, to kill the wetland, and construction to start. So I clicked pictures with GPS data of the sluice gate of the dry wetland, and I sent them to all the authorities, filed complaints. I submitted them as proof to a couple of litigations that were filed by some non-governmental organizations at the Bombay High Court. And finally, after about 2 weeks of a struggle, the High Court ruled in our favor and they opened the gates and the Panjay wetland was restored. But this was a very small battle that was won in a long-standing war, because I've been associated with this campaign for a couple of years now. My role has largely been to collect photographic evidence and give it for litigation to support that. And during these years, I've had to work undercover. I've worked with local government organizations, with the state government, with NGOs, with the local fishing community, as well as the media. And together with all of us contributing our bit, we've built a campaign to protect the Panjay wetlands. But once these pictures went viral and a lot of media attention fell on Panjay, what happened was the developmental agency, they installed illegal security cabins, as you can see on screen. And these people literally stopped us, all photographers, researchers, and even birding enthusiasts from entering the wetland, because they realized that with the camera and with the technology improving, you can actually click GPS tagged photographs. And that was proving to be very detrimental for them, because these photographs could act as proof in the High Court and for any litigation for that matter. And even when I went there after this security cabins were installed, these people threatened me, they've abused me. I've also been threatened with dire consequences if I continue working for it. But that never actually dampened my spirits, because I wanted to truly protect this wetland. This is a wetland that I have grown up at. I have some wonderful memories with my father birding there. So I really didn't want to lose all of that. And this is such a thriving ecosystem, not just for the biodiversity, but also for the local fishing community. And there's been multiple incidences after that. Firecrackers have been burst at Panjay to scare away the birds. Mangroves have been hacked mercilessly by these security guards. And every time we've filed with photographic proof. And finally, in 2022, victory came in the form of the National Green Tribunal upholding the status of Panjay as a wetland and dismissing the developmental authorities and telling them that all illegal activities have to stop, the land has to be protected, and the security cabins have to be removed. Not just that, another victory came in the form of the National Wetland Decadal Change Atlas, which is prepared by the Ministry of Environment, Forest and Climate Change, that listed Panjay as a wetland of importance and a wetland on the Central Asian Flyway that requires protection. And finally, now the pressure on the state government is very, very high to declare Panjay as a bird sanctuary. And I really hope that the verdict comes very soon, and the wetland is not just protected for its biodiversity, but also for the fishing community who really depend on this piece of land for their income. So this entire campaign, I would say a large part of the success came from the photographs. They acted as a catalyst in getting the attention on the issue, and as well as allowing, like kind of inspiring the government to act quickly, because otherwise it takes a really long time for litigations to be heard and given a verdict in India. That said, photographs cannot be ignored. The visual medium is too strong for that. The storytelling has the capacity to not just bring about environmental conservation at the grassroots level, but also it can bring about environmental protection at the highest policy level. So I feel together, uh, with all the people across the globe, each one of us, if we decide to put our heart and soul into it through photographs and through films, we can make this world a better place, a greener and a more sustainable world. That's it. I think, yeah, that's. Thank you so much, Joe, for inviting me.
All right. Isharia, thank you so much for that great presentation. And that clip you shared from the movie was just incredible. And that's actually where I want to start is 6 years. Wow, that is a huge commitment to bringing that story to life, bringing it together. What keeps you going? Because I know there's probably days, maybe even weeks, where you can be out in the field and you don't get any good footage. What keeps you going?
I would say every time I am in nature, that kind of really charges me up. Each moment that I spend, there's something new to film. Not necessarily that I see the big cat or the subject of my documentary, but there could be other moments with other animals or even with plants or anything. Being in nature that kind of gives you a sneak peek into the world as a storyteller when you're that close, and that really gives you time to think about your work, and also to it's just cathartic, I would say. So that's what keeps me going.
Yeah.
I know it's
All right.
frustrating because there have been days, especially on a recent shoot of mine, where we were at it for about 3 months continuously, wanting to click a particular behavior of a big cat, but that just didn't happen. I mean, luck wasn't on our side. But we also got to witness a lot of other moments which were otherwise not visible to other people. And kind of the story compensates that way then.
Okay. And then you talked about those two types of films, the blue chip and the human interest, the conservation. Do you lean more towards one or the other?
No, actually not. I try to maintain a balance between both because both of them are different means, different vehicles of hitting different audiences. And through that, you can achieve impact at different levels using both these mediums, using both the type of films. So, yeah, I try to maintain a balance between both.
All right. Let's grab a question here from YouTube. Have you ever thought about, or do you use drones to try and document some of that, maybe illegal activity?
Using drones, I've not thought about documenting illegal activity till now, because at Panje, drone flying is prohibited because it's close to the JNPT airport. The JNPT port, and it's a little dicey place to actually fly a drone because there's a lot of Navy attention, there is the port authorities attention. So you can, if caught, I'll end up in jail for something which I'm trying to do for a greater good.
Sure.
So I haven't thought about flying drones yet for that, but for another project of mine that's coming up, yes, definitely flying drones is on the question for capturing the illegal activity.
All right. So I know that there's when you're in the wild and you're filming, you get attached to subjects or you get attached to a story, and sometimes there might be that that urge to intervene. Sometimes, is that something you've ever felt and had to push down?
Yes, I have, because especially while filming Maya, there was the time when there was another, so Matkasur's litter from another female, that male cub that entered Maya's territory. And we were filming Maya, and Maya had two small cubs, a third litter. She'd already lost her first. The second one, yes, she rejoined with them and they split up. But the third litter was very small cubs. And we had just got an intimate moment with Maya and her two cubs. And then we see this huge male walking, and the cubs coming on the road and crying and fleeing the area. And then Maya confronts with this male. And that time Matkasur was nowhere around. And this litter was from Matkasur. Her third litter, she mated with him. And at that point, I was literally in tears, because I was seeing these two small cubs crying for their mother. The mother's fighting an intruder off who's almost twice her size. We don't know what will happen, whether she'll be grievously injured or not. And you have these little tiny ones crying on the road next to the vehicle. That was a heart-wrenching moment. I really felt if I had the power, maybe I could go ahead and, break that fight off. But at the end of the day, that's nature. That's the law of the jungle.
Yep.
So you really can't intervene in that. So that urge to, that conflict, yes, definitely does arise. But it's something you have to work on in the field.
All right. So as a woman in your field, are there any challenges or issues that you find yourself overcoming or dealing with?
Yes, I do. I do find a lot of issues, especially when it comes to filming in areas where there's a lot of perception that women are only meant to be running the households and not be doing a field job. So I've come across sexism. I've come across people men especially telling me that, oh, you're a girl, you shouldn't be doing all this. Go back to your house and learn to cook, or do a desk job. And even on the field, when I'm working with other male colleagues, some of them are very open, but a few of them I have come across people who don't want to take orders or don't want to listen to my creative perspective. For them, they feel that as guys, we already know how this is done. You're a girl, so your knowledge on this subject is not as much as what I have. And they're not even willing to lend a ear to my suggestions or my opinions. So that sometimes does hurt, that just because of your gender, you're judged.
Oh yeah, of course. And I think you're definitely proving them wrong with what you've been doing and the success you've been having. And you paved the way for a future generation of female filmmakers, and you're inspirational. So
I hope so. I really hope so.
Absolutely. It's awesome what you're doing. It's amazing.
Thank you so much for giving me a chance to present my work at this amazing festival.
Absolutely. Aishwarya, it was an absolute pleasure to have you join us for the first time this year. And we can't wait to continue following your work.
Thank you. Thank you so much. And I really wish the best of luck for this festival. I hope it becomes a grand success.
All right. Well, thank you. I hope you enjoy the rest of your day. And hopefully we talk again soon.
Yes, I hope to connect with you soon as well, Joe. Thank you so much.
All right. Thank you.
Bye.
Okay, we've reached that point. Day one is in the books. A couple quick reminders before we wrap up for today. First, we're at it bright and early tomorrow, 8 a.m. Eastern. We look forward to seeing your groups joining us. Tomorrow is split into 3 parts. There's a morning, an afternoon, and an evening. And there's no pause in between. It just has to be split into 3 because YouTube can only do the 6-hour chunks at a time. So starting first thing, 8 a.m. in Eastern in the morning, we hope to see you joining us. head over to the website and you can see the schedule and see what's coming up. You can tune in live on the website. If you registered for the festival, you have all of the links. If you've just registered recently, we'll send out another update with links in the morning. And I want to wrap up with just a big thank you to everybody for tuning in today. Thank you for being with us. Thank you for the great questions. Thank you for supporting this festival and spending some time learning about biodiversity and hearing the stories of these amazing scientists, conservationists, filmmakers, and more from all over the world. I do want to wrap up by just a shout out to Lenovo EDU. They've supported this festival for the last couple years now, and they are building an incredible education community. And it's not just for educators, it's for kids, it's for scientists, for explorers to find resources, to share ideas and conversation and things that work and things that don't. So do take a moment and visit, register for the community, find out what it's all about. There's some really cool blogs and video pieces. And of course, they share some of this festival and some of the other festivals that we do as well. And so I'm going to leave off tonight. We're going to play out with a little sizzle reel from Lenovo EDU, and we cannot wait to see everybody with us first thing tomorrow as we dive into day 2. Field trips, guest speakers, multiple continents. It's going to be a blast. So signing off for tonight, and we will see everybody in the morning.